I'll push forward as you bring your rotator up. And sonar. Oh, there we are. As expected. 30 centimeters out.
It's always nice when you find the seafloor at the bottom where you expect it.
Yeah. Okay, looks like you got your camera coming up.
Yeah, 60 degrees.
We'll push forward, and then we'll come down together.
Yeah. All right, 45 degrees, and I'm pushing out, pushing down.
Copy, pushing down. Down we go. Try to pull some tether down here. Pilot has eyes on bottom. All right.
There it is, everyone. It's the bottom, and I'm super excited to be here on our final dive of our Deep Connections Expedition 2019, and we are currently in Box Canyon between Block and Alvin Canyon.
And Megan is super amped up because she had 3 espressos this morning.
I sure am. So this is Megan Putz, your biology
yeah.
lead from the University of Hawaii, and our first sight is the cutest octopus ever.
This is a good start for the last dive. If we're
Bridge
starting
nav.
off with an octopus, then it can only get better from here.
Well, they just knew that I was so sad that we missed out on octopus yesterday for Octopus Friday, but I did represent, was wearing my octopus leggings, my octopus shirt, my octopus headband, and my octopus earrings.
This is a supplemental octopus Saturday, I believe.
Do you have a bailout plan?
Yeah, let me just double check that. I'll call back.
And this is Jeff Obeltz, your geology lead from the Naval Research Lab. And we are starting the dive with a little octopus that is Bathypolypus?
What?
Bathynomus, right? No, that's a giant ice pod. Never mind. Okay, I'm going to stop talking.
Yeah, that's right.
What's it called?
It's called Granodoni.
Oh, same thing.
Yeah, okay.
And also is that a...
It's an eel.
Could you come in?
Starts with S.
The dragon one.
Nope.
No?
It's the one that has the funky gill slits.
Ah, cutthroat.
Yeah, it was
Okay.
like,
I thought it wasn't
bailout
because it had the
southeast.
fins coming out on the side.
I mean, a lot of fishes have that. But the halosaurs that you were thinking about have the fins further up on the body.
Yeah. So once we get
slowly
all
creep
settled
in
out,
this
we will
guy.
do a nice zoom on our little octopus here, who is just entirely apathetic to our presence, thankfully.
Stand by.
I do love octopuses. So adorable.
It doesn't get much better than landing on bottom and immediately seeing an octopus.
It definitely doesn't.
You can bring a lower outboard, yeah.
Look at this little octopus friend.
Okay.
With his little
Come in
cute
video.
curly
Try to hold this.
tentacles.
Will you get lasers too when you get a chance?
Yeah.
Thank you.
The other thing I like about
video.
seeing these octopuses is we
Yeah.
can actually see a reflection in the octopus's eye. So all those little lights are all the lights from Deep Discoverer, the ROV.
And they have really well developed eyes, right?
They do. Octopus are also very intelligent.
Yep.
Yeah, they are way too smart for their own good in terms of solving puzzles. They pass the mirror test. They can recognize their own reflection as a reflection, not as another creature. They're infamous escape artists from aquariums, and at this point I'm half convinced that if they had opposable thumbs, they would be our overlords by now.
It's funny you bring that up, Jeff. In my invertebrate zoology class in college, one of the other students did a presentation about cephalopods, and that was his final remark was, if they had longer lives, they would probably have taken over the world by now.
And they'll invent that before too long.
We'll get splash lasers on it. Copilot, can you turn my lasers on?
Lasers going on.
It also looks like this octopus has closed its eyelid. You see the small membrane that's come over its eye?
Yeah, you can zoom in on the eye. Oh,
Laser. We're going for lasers.
It looks like it's considering movement. And if I recall correctly as well, they have kind of little independent neurological substations for each of their tentacles. Is that right?
That's right. So their tentacles are sort of have their own mini brain.
I didn't know that.
That's pretty awesome.
It is pretty awesome.
What isn't awesome about these things?
I can't think of one thing that isn't awesome about an octopus. They're full of curiosity.
They're out there
They can
somewhere.
fit through any opening as long as their beak or their mouth can fit through it.
Yeah, and this is a little one. Those lasers are scale lasers. They are 10 centimeters apart. So this little guy isn't too much bigger than 10 centimeters. And it's just gradually unfurling, like doing a languid stretch on Saturday morning.
I think it's decided that we've been staring at it too long.
Yep, and maybe that's our cue to begin the rest of our dive and start to make our way up the western wall of this little box canyon in between Block and Alvin canyons.
He's like, I'm not sure about.
I really want to watch one swim away from us. Maybe he'll swim, but it looks like more of a slow meander.
Yeah, this one doesn't quite look like the Minister of Silly Walks that we saw the other day. The octopus that had this really...
I'll take off.
Yeah, there's no other way to describe it than just a silly walk.
It was a silly walk, but it was,
screen.
a very long stride and very swagger kind of walk.
Yeah.
Well, it bodes well that we might see some more octopus along the way.
And we also, as soon as we got onto bottom, we saw some debris here in the access of the canyon, which is generally associated with relatively steep slopes.
Have a nice day.
That indicates that there's probably some sort of gravitational slope failures or mass wasting happening
You
here.
want to get a move underway now?
Because especially during sea level high stands,
Yeah, true. What do you guys
at this location
thinking?
along the continental shelf, there aren't too much in
little
the
south
way
of west?
of triggers of slope
265,
failure
2.
that cause
255?
moderate to large size events
Okay.
in other locations.
Kind of
And
range
those
you looking at?
triggers can
30?
include seismicity
Start at 20.
or
Let's see what,
earthquakes.
we approach.
They can
I think
be
that's a good idea.
active
Okay.
river
20
flushing
meters,
events or really large river
255 on the
discharge
way.
for submarine canyons that are on active margins, so the continental shelf is really narrow and the rivers are still connected to submarine canyons during the sea level high stand that we're currently in. Or one of the other big triggers for slope failure, more in really heavily sedimented river delta areas, are the passage of hurricanes or large storms that create differential peak trough pressures on the seabed that can destabilize sediments and cause them to fail downslope. But in this area, just due to the depth, the distance from rivers, and also the
Are we a little hot in the middle
general
of the video?
lack of seismicity or earthquakes, most stuff, if there's going to be
There's lighting.
slope failures that are happening in this area, it's just kind of stuff failing under its own weight
Yeah, try
after
to put it
time.
out, see if it gets any better. Let us know video. I'll let you know when we're...
We might have our first coral of the
You
dive.
can zoom in on the coral video.
This sort of
See,
brambly looking
just
bit
lights
could
are-
be a coral.
You can come
So
in.
that's also a good sign. As we zoom in, it doesn't look very coral-like to me.
Yeah, what is this?
Might be a massive hydrozoa. But there are a lot of animals hanging out on this little bramble. I think there's a nudibranchs You
Yeah,
see
I was looking at them, and that's what they
Oh,
look
yeah,
like.
let me
Like,
get
not the
first.
really bright colored
Yeah.
ones that you're used to seeing in shallow water, but the whole body plan certainly looks like it. That was going to be my guess, but I was afraid of being wrong.
It's okay to throw out guesses. I'll always,
Yep.
correct you if I
You ain't
can.
coming.
But
think I'm
yeah,
looking at the right
I'm
thing, yeah.
pretty sure that those little animals
Yeah.
you're seeing are a type of sea slug called a nudibranch. It has all of its gills, those little appendages on the top, outside of its body.
Tyler, can we get a zoom on the sea slug?
Yeah.
I think another common name
you.
is sea hare.
I'm gonna
Because
try to
they
get closer.
usually have
You
two
can come out.
longer projections that
I'll hop
look like
over there.
ears,
Let's
like a rabbit. But those are very, very small and super adorable.
I'll get
as
you a
well,
closer look. Watch it. Hold on.
Just getting so excited about this biology.
Come back in. Okay. All right. Copy. Coming out.
The other things that we're seeing in the branches of this bramble are some amphipods, and those are definitely sea hares or nudibranchs.
Okay. Come in more if you got it.
Yeah, I'd always
on
heard
these
of those
guys.
associated with kind of
Alright.
tropical shallow areas.
They're small.
Is their range pretty wide then?
Yeah, you can find them in a lot of places, but it's those brightly colored tropical ones that usually get the most attention. But we see them in the deep sea quite often.
I'll actually just frame it up for a second, and I'm gonna hand off.
There are quite a number of really kind of interesting colored nudibranchs that we have in our deep sea animal guide. One of my favorites is this one that has these little balls on the ends of their branchiae. I'll show you a picture. And everyone at home, if you'd like to check out those images, you can look at the Deep Sea Animal Guide. Go into the mollusca and the gastropoda. And that'll show you a number of different animals that we've found throughout a number of dives in the Pacific, and we'll be also adding some
All right,
Atlantic
co-pilot.
animals coming up soon.
Here we're under a move.
Yes. Ship
This is the one I'm talking about. It has
Carrying on.
little yellow balls at the ends.
Ship's stationary right now, right?
Oh yeah, that
It's
one's a
like
little
red,
more
red spotted. It's super cute.
ship
a little more festooned in color.
was moving. Let's see.
Ready for a
looks
deep
like
sea
they made
festival.
about 20 meters, so they might be about done.
Let's get a quick zoom here, just for ID.
Is that trash or a coral skeleton?
Looks like trash.
Yes. It kind of looks like trash. It's not coral.
When in doubt, assume trash.
All right, let's come on. All right.
Go ahead, Nev.
All right, come back up.
All right, pilots. Let's see. Last move went in about 6-7 minutes ago. How do you feel about another 20 meters?
And if you're recently joining
Oh,
us,
it's okay.
one other
You can
thing
go on fish if you'd like.
of note is that This
Good.
is our first dive somehow. This is dive number 12, and we've dove on almost
much
all
distance
submarine
is that to the
canyons, and for
first
some reason this
significant
is our
contour
first
there?
dive on the
At the
western wall of the
1240?
canyon. I was thinking that
65 meters.
the Okeanos on this expedition
like a
has
30
kind of
meter
a
boat
tragic
together, 2
Zoolander
hops?
affliction
Yeah,
where we're
even
just
then it's only like a
incapable
20
of
degree slope.
diving
Alright,
on
let's
the
leave
west wall of canyons and only on the eastern
Yeah, let's do
walls.
30
But
meters.
we've
Just gonna
overcome
do west.
this
How's that sound?
this
Yeah, do
270.
west
possible
first.
affliction,
I'll,
and we are ascending the
over this
western
tiny
wall
bit.
today.
275. Or 270, excuse me.
Sounds good. And I'll put it in.
This is Alvin now.
Go ahead now.
Okay, you ready for another move?
I'm ready.
30 meters at 270 degrees at decimal 2 knots.
Yes, 30 meters, bearing 270, speed decimal 2 knots.
Good copy.
And we are expected to see fairly steep slopes today, but I would hazard to guess,
Ivy there,
and our
Delta,
mapping lead Mike White
Yeah,
told
you
us
seem
that
a little high up.
probably shouldn't expect
I thought it was a little
the
low.
Oh,
stunning
really?
geology that we
Okay.
saw
I'd
the other
like to
day
stay
in
around
Beach
12 to
Canyon,
15.
where we
Good
saw
enough.
a
I can come down if you'd
continuous
like.
succession of over 100 vertical
Oh, you're-
meters of carbonate material, limestone and
We
chalk.
have a Delta
And
10 right
after
now.
the dive, I looked it up, and the height of that carbonate
Yeah, with
sequence was
the
actually
slope, though,
taller
our
than the
deltas
cliffs of
will have
Dover,
to change,
which is pretty cool.
right?
Yep.
As I get it out.
And it was really cool to look at
On the
as
slope,
you
we'll need to
panned
maintain a
around
smaller
and
delta.
as we took a look at
Yeah,
the
okay.
Sirius view, which gives us a wider view of the geology in the area. It just was a huge expanse of white sheer rock face.
Also, I just want to reiterate that.
Yeah,
if you
and
come
I was
across
doing
corals,
some remedial
excellent.
geology after the
Nice
dive
and long
regarding
shots.
carbonate stratigraphy.
Thank you.
Carbonates aren't particularly an area that I'm too familiar with. I mostly focus on sedimentary or land-derived rocks instead of carbonates, which are made out of either corals or the fossils or the shells of little tiny animals like pteropods and coccolithophores, or gastropods as well, you know, mollusks,
This
your
one looks different. Let's
bivalves
go
and your
in on it.
snails and whatnot. And
Let me try
apparently,
to bring us closer.
carbonate rocks tend to hold their slopes really well because they get very cemented and consolidated, chemically cemented together, and so form kind of stable bricks and buttresses.
Right now we're taking a look at a hake. This is a fish in the family Moridae. It's pretty distinctive with its long dorsal fin right at the top of its body and the long filaments. And this one has a very pointed nose.
Yeah, you can go tighter if you'd like. I don't
So,
know that I've seen one
I'm
with
not
a
familiar
snout
with
like
this
that.
particular fish. I thought it was going to be Antemora. Might still be in the genus Antemora, but not the same fish that we've seen
Yeah,
on previous
go ahead.
dives. Maybe it'll cooperate with us for a nice, good close-up.
Yeah, it doesn't seem exactly terrified of us, but also doesn't want to stay still. And also this crab in the background with its defensive posture, I think, with the claws straight upwards.
Yeah, this fish probably doesn't like looking straight at us because we do have lots of bright lights
Go ahead.
on our ROV.
Fish proceeds to stare directly at us.
You know, they like to do the opposite of whatever I say.
It's a really good mundane superpower, is to have marine life be able to do
Almost
the
looks like a
exact
duckbill
thing that
to
is
it.
not what you tell them to do.
Yep. It's sort of like when
gonna lose
you
him in the
pack a
cloud here.
raincoat, but then it
Yeah,
doesn't
let's come
rain,
out.
so you never get to use it. But the moment you forget
As
it,
I was
that's
lateraling,
the moment you just get torrential downpour. So
I
I'm
took
working
some of
on
the
this
sand with
reverse
me.
psychology with
Sorry,
these
a little-
animals.
Seems to be
Photo of
working
that
out.
pilot.
It's a shame we're not going to see any really cool corals or sponges on this dive.
Yeah, I guess not. No corals or sponges on really beautiful rock faces today.
I'm purposely trying to get some of this dirt off here. That I picked up.
Yeah, pardon the
Laterally over.
turbidity. Our pilot is just trying to shed some dirt and stuff off of Deep Discover. Just
All right, pushing back forward through the cloud.
Yeah, as
There
we touch down, the sediment is very soft and sticky, so it tends to stick to the tips of
there
our
doesn't seem to
toes.
be much
And
in the area,
we
so
usually
let's
like to put down so we can
see
get
if we
nice
can't
steady shots.
get some other stuff.
causing the least amount of disturbance to the area as we can.
I'm coming down and gonna tilt up.
Oh, that was a great view. This fish has a huge eyes. Huge eyes.
Lots of parasites, it looks like, too.
That too. We've been noticing a number of parasites on the fish we view. Most of them are copepod parasites.
It's coming in first close-up.
But we also see amphipods and isopods parasitizing fish as well. So most of the parasites
Yeah,
are
let's let them go.
crustaceans.
We probably got enough of them anyway. Let's
Does
push
anybody
out
have a good
beyond the
idea
dust.
on how long those parasites stay on the fish? Are they just on there
Should
for
have a
life,
little
or
more.
are they more like
Yeah,
ticks
should have
where they
enough
just
to get
their
through it.
fill and then fall off?
I believe they do fall off eventually when they, die or reproduce. But I'm sure it varies from species to species depending on the parasite. And I'm not an expert in fish parasites, so I wouldn't be able to tell you for sure. But I'm pretty sure that some of them do fall off eventually,
Go
but
ahead and
some
get another
others
one in.
are there for life.
Yeah, if you don't mind. It's still pretty spread out there. We're only 1,300 meters. I don't think we'll have too much of a swing.
And now that we've seen one octopus, any variation on the seafloor, I'm like, ooh, is that an octopus? Is that an octopus?
Oh, we saw one this morning.
But no octopus yet.
Sorry, say again?
Yeah, one of those trademark Megan
great.
Putz roctopuses that we've seen.
Yep.
Roctopi?
Roctopods. Now we're taking a look at a suggested shrimp swimming through the water. They're pretty obvious with their extremely long antenna. We often see those in the water column.
All right, pilots.
I'll never get tired of those because they
sonar looks
just always
pretty
remind me
clear
of the
ahead.
shrimp on a treadmill video.
That
Why
move
was
did just
that
finish a
shrimp on a
couple minutes
treadmill,
ago, but I think we look good
I
to
don't know.
do
Just
another
goes
30.
in the water
You'd
column.
have to ask the
I
shrimp.
agree.
And a lot of the fish that you see swimming around those small fish are bristlemouth fish. Their scientific name is cyclothone.
It looks like one of those sea cucumbers that does crunches to move.
Yes, this is one of the swimming sea cucumbers, and it's extremely small. So they flail their body in a very awkward way in order to swim through the water column. But most of the time, these animals are on the seabed, and they eat sediment and remove all the nice, tasty, organic matter from the sediment and, remove that sediment, cleaning it for everybody else. They're the vacuum cleaners of the deep. I always like how they swim. It's just so awkward and awesome.
Are you good with that video, or do we want
Yeah,
to try
that
to
was a
get some
pretty
more?
good sighting. I wasn't sure what we were looking at until it came into focus.
I think we're probably.
Yeah, viewed from afar, you'd think it might be a jelly or
Alright,
something,
Pat's gonna
but
put
it's
that move in
actually
it for all
a
set.
type of sea
All set.
cucumber, which
Go
is
ahead.
an echinoderm related to other animals like sea urchins and
0
sea
meters
stars.
at 270 degrees, decimal 2 knots.
Yes, 30 meters, bearing 270, speed decimal 2 knots.
Cap.
Oh, I think I spotted a sea pen. It's middle center screen, pilot.
Yep. We'll
There's
take
a
a
small
look.
stalk
Might have a little bit.
a little
till I
bit
get
lower
there.
down on your screen.
Standby video.
So sea pens are a type of octocoral, and these animals usually grow in the sediment, though there are some sea pens that have a modified base that can hold
Start
onto
going.
rocks. But this one has a long peduncle that goes down into the sediment that helps it anchor there. And so if we were to pluck this sea pen from the sediment, it would have this long, very pen-like
Pretty
base.
far out at the end of my tether, so I'm
And
getting
here you can
yanked
see
around.
the tentacles of this coral are
Trying to stabilize
very
for you.
translucent, so it's hard to see.
All right,
But the polyp
think I'm there.
body is actually a dark, more opaque white color,
of course, just as I said
and
that.
the axis of the coral is red. That's actually really interesting. Normally, when we see these animals, they tend to be either brightly colored all over, or it's sort of like a tricolor polyp coral. But that white part of the animal is the stomach of the polyp, and it can pull those tentacles in really close to that main stalk. So this coral is likely Protoptilum.
We saw these, not this exact species I don't think, but sea pens in general, in huge densities in our one exclusively soft sediment dive site, which was an Intercanyon in Canadian waters where we were looking for Russian hat sponges. And we didn't find Russian hat sponges, but we sure found a lot of sea pens and also a whole lot of short-fin squid,
Alright,
if I recall.
yeah, let's come wide.
That was the squid dive.
Yes,
Squid Cafe.
it was a squid cafe.
If you want to see all those squid, you can go check that out at
Coming
the
back to center
highlight
compartment.
videos from this
Copy that.
expedition at oceanexplorer.noaa.gov.
Up a little bit as you did.
Oh, it's
It
my
came down
squid.
quite a bit.
Oh yes,
Go ahead.
this is Magneteuthis magna.
I love these things. They're super cool. They're just really majestic how motionless they remain and just kind of glide at really neutral buoyancy. That's scuba diving goals right there, that neutral buoyancy.
I like how they swim in an L shape. Normally when we see the squid swimming, their tentacles are in line with the rest of their body, but this one always seems to be swimming in this
Still
L shape,
working with us. Yeah.
just sort of floating by.
Now that he's coming up, this will help. Yeah, go ahead.
Yeah, that's a great look at it right there.
That's a really beautiful squid. And it blinked its eyes at us.
I don't think we've seen one do that so far.
No, I don't think I've ever seen that. But it's probably because our lights are bright. Sort of like when you turn on the lights too suddenly in the morning and you're like, whoa, bright.
Yeah, this squid is used to the only light it observing being from bioluminescence, and now it's got pretty bright ones on it, but we'll be gone soon, and it'll be back to normal.
Yep. Another super cute, charismatic cephalopod for this dive. Only a few minutes in, and I've got my cephalopod fix.
If you want them
I
with
haven't
the
gotten my
black
fix.
background,
I
this is
want
our
more.
final chance.
Well, I'm not saying I couldn't have more. I'm just glad we got at least 2 observations.
Squid box checked and octopus box checked. Not bad for a half
Otherwise,
hour
I
of
have to
that
let
of
'em
bottom
go.
time.
Here. Looks like one of those mucus.
Yeah, I just have your light bar to my starboard.
Copy.
I can turn my head.
I'm just
if you like.
really enjoying this squid. It's so beautiful.
Is that your favorite mucus house, Bob?
Start rotating my head.
Yeah, it is. All right, I think it's time to leave them.
Yep, let's move on. We
I
have
you
a
today.
long
I
way to
came
go in such a
15
short
degrees to
time.
starboard. As you come over,
Hello, Okeanos.
Hi, Mike.
Now, Brady,
I skipped calling in on the octopod, but I couldn't resist on the squid.
The squid's pretty awesome.
Oh, you shouldn't have resisted calling in
yeah,
on the
that's
octopod.
Well, you've seen a bunch of granulidini
270.
before. That's a Magneteuthis magna, which is a really typical squid of the deep sea. And we often see them drifting along just above the bottom. They actually have, their tentacles have microscopic suckers on them. They're really, really tiny, but they're really, really sticky. So they keep the tentacles in the sheaths in the ventral arms, and then spread out their ventral arms with the tentacles dangling down underneath them and they basically just drift along waiting for copepods and things like that to bump into the tentacles.
So they have a
You wanna
pretty
go ahead and get
similar
another one started?
feeding strategy as the siphonophores that we see a lot as
Yeah, you
well.
can put one in. How do you feel about moving at
That's correct.
260 for the next move?
Or actually more,
Yeah,
it's
you can do
it's
that.
more like that of
Copy
a high-dipid
sonar still looks pretty
ctenophore, because
clear. So I think
the
we're still good for
ctenophores
30
have
meters if
sticky
you're okay with that.
tentacles rather than
Copy
stinging cells.
Okay, I'm
And
lined up at
these tentacles on the
265.
squids are really, really sticky.
You want 260 then?
Yeah, if you're
They're
good with
super
that.
cool looking squids. I think they might be my
260 there,
favorite squid after this expedition.
Go ahead, Nav.
Hey, you ready for another move?
Well, good.
30 meters at 260 degrees, decimal 2 knots.
Okay.
Do you have any questions about them? Otherwise, I'll get
Good copy.
back on the road.
Just, I don't think we've talked about this dive yet. One of the things that just makes them so cool is just that magnificent red color.
This is a shrimp.
And why are they red?
Looks like it.
Well, lots of things in the deep sea are red because red light gets absorbed by seawater really quickly, so it doesn't penetrate very well through water, which is why the light that you do see as you get deeper and deeper in the water is blue, and blue light won't reflect off of red pigment, so it's basically like absorbing all of the light that bounces or that hits it, so the light doesn't bounce off and give them away to predators.
All right, go ahead.
Yeah,
Video.
that makes a lot of sense. That, the ctenophores, and also this shrimp that we're looking at right now. Pretty cool.
Just as an aside, there are fishes that have red headlights to foil that form of camouflage.
Adaptation and counter-adaptation, right?
I'm sorry?
Adaptation and counter-adaptation. Everything trying to stay one step in front of the other one. Pretty cool.
Evolutionary arms race.
Cool. Thanks for calling in, Mike.
There we
Always
go.
good to have the cephalopod 2 cents.
Well, you've seen a couple of good ones already.
It's been a great dive for that so far.
Okay, I'm going to hang up and get back on the road.
Awesome. Thanks, Mike.
All right. Okay, bye.
And we're on your call video. Copy. Moving on.
So that shrimp we were just looking at was Nematocarsanus, a type of long-legged shrimp, very common in the deep sea. We often see them ambling along the sediment or along rocks. We're also seeing a number of fish in this area. So fish often use these areas of flat, soft sediment as feeding grounds. So they'll come here looking for tasty snacks. I think that fish found one.
As demonstrated.
I believe that fish is a halosaur. So what you thought the first eel was.
If you want to snap in on fish, yeah, just for ID.
Yep, that was definitely a halosaur, and showing us how it can swim really quickly
Sounds
away.
like we got the idea anyway.
That halosaur started its morning with
Let's
3
come wide.
espressos just like you did, Megan.
Probably.
And we also see, this is the second crab that we've seen, and they've been orange instead of the red color that we've seen most of our crabs so far during this expedition. Do you think that's a different species, Megan?
No, I think it's the same crab. It just, sometimes they are slightly different variations, but most of the crabs we've seen are in the darker orange. This one is a little bit lighter. Well, I think I spotted a Coca-Cola can.
Oh, I wasn't sure whether that was Beerus canis that you pointed
The
out during
Beerus
the last dive.
canicus?
Yeah.
No, I think it's a Coca-Cola.
Another, unfortunately, more common than we would like species in these relatively far from land canyons.
Pilot, can we snap this fish in the foreground?
We can. Go ahead, video.
So I think this might be a type of cusk eel. Nope. Yep. Yes. Okay. I think this is one of those live-bearing cusk eels. Oh, it got a snack. It threw me off because it opened up its pectoral fins and they were just so beautiful and long. I was like, wait, what kind of fish is this? Possibly Barotherides. I think that's the fish. I'm going to check my animal guide real quick. But this is a type of cusk eel. And they're pretty distinctive with their dorsal and anal fins coming together. They're very long along the body. So to find this fish, you go into the chordates, the vertebrates, the ray fin fishes, and then the eel-like fishes.
And it seems a pretty common adaptation for these bottom-dwelling fish to have a lot of barbels and long little tendrils on their face, which
nice
they
job
use
there,
to root through the sediment and find stuff that they can eat.
Coming back up.
Nav grid move complete.
Okay, found it. So these fish are in the Ophidiidae family, and this one might be Dicroline. According to my animal guide, they have a wide array of tendrils that you saw coming from the little feelers that were underneath the head, and very long pectoral fins.
Yeah, it looks like a pretty close match to me.
This looks different. Snap.
Yeah, I think that is different. I think that's a Liparid. So a fish in the family Liparidae. I believe the common name is snailfish.
Yep.
And they're a common deep sea fish, but we don't often see them. And some of the first observations of this fish alive were made by the Deep Discoverer, I believe. In the past, many times these fish have come up in deep sea trawling nets. And so what we do know about them is with museum specimens. And museum specimens, you lose a lot of the color and the shape of these fish when they're alive. And the way we tell these fish from the different species is by counting things like fin rays and teeth and small characters within the body that are preserved when the specimen is preserved. So
I'm
if we get
drifting way out from them,
nice
but I can't.
side body view of this fish, that would be really good for identification. But they are very small.
I'm way far away. That was my problem.
There might be some experts out there, people like Bruce Mundy, who's an expert in deep sea fishes, might be able to see a picture posted on social media and
let's try again,
provide
just
a
partial.
nice identification
See if we can
for
get
this
a better
fish.
lit. Few seconds. There we go.
Thank you, Pilot.
Much happier
that's
with
great.
that. Go ahead. There we go. Much better.
What's always interesting about this fish is how transparent the skin is. So you can actually see inside of this fish, and has a very rounded snout.
I'm going to lose him in some dust soon.
I believe
so
this is part of the group or the family of fishes that is the deepest living fishes that we know of, observed within the Marianas Trench.
Let's go full if you got it, because I'm going to lose him soon.
Thank you, Pilot. That was great. We're seeing some really awesome fish today. So far, we've seen some halosaurs, cusk eels, rat-tail fish, the snailfish, hake. It's very fishy.
Yeah, and we've also reached the toe of the slope here, and now we are ascending up the canyon wall, and we notice right away a pretty dramatic difference in substrate
Right, it's
between
starting to get
this
into a
and
little
Beach
bit more
Canyon, the last
slope
canyon
here.
we were diving in. Where on Beach, we almost
Sonar still
immediately
looks
came
pretty
into
good. How do you feel about keeping 30 meters at
rock
260?
walls, relatively high gradient. When here,
Could
what
also
we're
bump it
probably
down to
seeing
20
is
meter
more like
mirrors.
a
Yeah, we just don't
buttressed
want to get
toe
too
of the
many
slope, where
in
this
line,
is
but
a
I think our
field
timing's
of
good.
talus
So.
Yeah, we've been
or
paused for
debris
enough.
that's
Yeah.
kind of
You guys
formed
swing.
To a 30
a
meter
triangle-shaped
move is good.
deposit
good, I'll
and
put
has
it
been
in.
mantled with hemipelagic sedimentation that covers everything in the deep sea.
Go ahead now.
Hey, ready for another move?
I'm ready.
30 meters at 260 degrees, decimal 2 knots.
That's 30 meters, bearing 260, speed decimal 2 knots.
Good copy.
These small fish you're seeing are bristlemouth fish, known as cyclothony. They are the most common vertebrate on the planet, and they're very common in the deep ocean and are one of the fish that we come across quite often during our midwater dives.
Is this a bivalve?
Looks like it.
I think this might be a piece of
Seems
trash.
So big to be a bivalve, though.
Oh, yeah, it might be a
I'm gonna get up
bivalve
close
shell.
to it.
You're right, pilot.
You have enough
I think
time.
so.
You have enough tether.
I just maybe my perspective is off with all the sand, but that seems larger than usual.
Yeah, can we get lasers? Then we can see
We
how big
will.
it is.
Absolutely, watch me. Just setting up, getting a toe down, and then we're gonna get some nice images. All right.
So I'm thinking that
Start
this
with
shell has
lasers.
come from shallow water and made its way down slope. Obviously, this animal is no longer alive. It's just its shell, just as you would probably find one on the beach. So it's kind of like going to the beach.
There you go. All right, go ahead, full zoom at will. Oh, that's my rotator going a bit longer than I'd like.
It would be interesting if this was sourced from the continental shelf, both for the size of this particular shell. It was over 10 centimeters long based on the lasers, and also just that this canyon that we're in right now, this box canyon, a box canyon is a generic term for the shape of the canyon and its location along the continental slope, are much, not much, but a decent amount farther and less connected to the continental shelf than the shelf-incised or shelf-sourced canyons that we've been diving on mostly during this expedition. It would have probably taken a fairly energetic event to move this shell and deposit it within the axis of this canyon, but not unheard of.
We're good here when video is.
All right. Let's come wide then.
See another fish up to your upper starboard.
I've also been noticing these scooting traces along
Let's go
the
fish.
in the sediment. So it looks like probably a
Another
fish.
duckbill?
As it scooted along on the bottom, you see these little ripples along the outside of the trail. And so fish like this one can just swim near the bottom or scoot near the bottom, and you're seeing the marks of its fins as it's moved itself along.
Let's come wide. Leave that one go. I think we got ID.
So that's another thing to keep an eye out. A number of people who study these animals in the deep sea are also looking out for traces of animals and animal activity. Even if you don't actually make an observation of an animal, the way the sediment has been disturbed, known as bioturbation, can tell us a lot about who's living here and what they're doing in this area. So we see a number of holes or burrows where animals are living. You see a number of traces that could belong to a number of different animals, like sea cucumbers making trails across the sediment, fish making trails across the sediment. different types of worms make trails alongside their burrows or in a raid pattern along their burrows.
A nice Tino 4 drifting past, not one of the red ones that we saw in the descent, clear one.
Some off left we'll go look at. Just wanted to see what these
Yeah,
three
after
things
reviewing the
sticking up.
animal guide, I do think that that fish that we're seeing, that hake, is an Antamora,
All right, go,
but
let's
I'm
go look
not
at
sure
this
of the species.
flatfish off left.
Ah, flounder.
Oh, yes, we have a flounder. So many fish.
Yep. Flat-fished with right-left kind of squishiness to the point that both of its eyes are on the, if I'm looking at this right, the right side of its body, I believe.
Yeah, go ahead, video.
Yeah, it's easier
Should
to
pass-
tell when you can
pass
see its
soon.
mouth.
There we go.
And then you have to like sort of think about it. I don't know what it is. Some people are like, oh yeah, right side, left side. And I'm like, which side is my right side?
Yeah.
I always have to think about it.
I think it is right side. And this one looks to be decent size, probably around 10 centimeters. Nowhere near as large as the halibut that we saw earlier in this expedition, one of the
Back up
highlights
slight.
for me
There we go.
in the minor canyon north of Kinlan Canyon.
Now,
Those
Braden, with the
halibut,
bait-
which were well over 1 meter, close to 2 meters in length, that we saw 3 of them, endangered species. Very cool.
So these fish are in the family Pleuronectidae. And that's the group of fish known as the
Pilot's ready
flatfishes.
when
Though
you are.
there are other types of flatfishes, like we saw a tonguefish earlier in this expedition. That has more of a, doesn't have a caudal fin like we see on this one, that end tail, but their fins come together, and it kind of looks a little bit like a tongue. Tend to be very small.
Yep, and now with the lasers on, we can see that it's about 10 centimeters wide and 20 or 30 centimeters long. So decent sized,
Moving on.
obviously commercial
There at 30,
species.
pilot.
Copy.
Looks like there may be something out here to your port. It's been pretty steady.
On Sirius sonar or your sonar?
Serious sonar.
I think it might just be a false artifact. I've been keeping an eye on it, and I don't think it's been moving. But yeah, still good to keep an eye
So
on
I'm the
it.
real bright target
Yeah,
that's
it's-
slowly moving. So I'm heading towards it.
Yeah, this. Laura, is you thinking like this one and this one?
Yeah,
So,
that's
yeah.
what I was looking at.
They've been kind of there, I think, since I've sat. They kind of come and go, but they seem to, kind of stay there. But Pies, if you are ready, it's been about 5 minutes. Ready for another 30 meters, 260?
Yep.
Copy that. Coming up.
Go ahead, Nev.
Hey, ready for another move?
I'm ready.
30 meters at 260.
That's
degrees,
another one of those
decimal
little
knots.
sea cucumbers.
That's
They're
30
in
meters,
pretty
bearing
decent
260,
density
speed
down
decimal
here,
2 knots.
doing their ab crunches throughout the water column to move around. And of course, the ones that we see in the water column are probably a tiny fraction of the amount that are living on the substrate, and we just can't really see them because they're almost translucent. So until you zoom in, you don't really know they're there. But we've talked a lot about this during this expedition, is that the field of view that you have right now, while it might look relatively featureless, if we zoomed into any area of the seabed, you would just see an enormous amount of creatures living on the seabed And of course, if you went even further and dug into the sediment, you would see an even further diversity of life as in
Getting
fauna
something out there on my
or
sonar.
organisms that live within the substrate instead of on top of it.
Yeah, that's weird, huh?
That's new.
And a lot of these organisms are
23
very,
meters
very
out.
small. So people who study these animals will often take
There's
course
quite the return
and
as
sift
well.
through the
Keep
sediment
our
in
eyes
order to
open,
find them.
So you're
And
looking at it.
even though they're
Yeah, it's
small, they
kinda
can
in
be
line with
extremely
track.
beautiful and very fascinating to study. So when I was first getting my start
Yeah,
in my
ship will
career as a
stop well
marine
before that, but
scientist, I
yeah,
actually
we'll
did a
approach it
lot
slow.
of
Copy.
core sample taxonomy, so sorting through
Coming back down.
mud and sand,
pretty
looking for these different
straight.
types of
Yeah,
invertebrates and identifying them.
Geometric, which is always concerning.
And that's where I learned a lot about
So we'll approach with caution.
what lives in these types of environments
Yeah,
and
20 meters out.
how
20
to
meters in front of D2.
identify organisms. So what kind of characters
This
are
new-
you
this
looking
new-
for
262.
this new
visually
should
in
put
order
you
to
with enough
identify an
cover to get there
organism?
to
And
inspect it.
that brought me to video annotation eventually, because I had
Let's see if there's
samples in
anything around
hand
here
and
in
I
the
knew I wanted
meantime.
to study the deep sea, and this technology that we have with the remotely operated vehicles is so great at being able to see very fine details, and that has made using the video as a tool to quantify the animals that live on in the benthos. So these, megafauna that we're seeing, like these crabs and sea
We'll look at the crab for a
cucumbers,
while.
fish.
I don't think
We've been
zoomed in on
actually
one
able to use that to gather data about what lives in the deep ocean.
While we're a little stationary, would you mind turning his sonar into a little higher resolution? See if we can get a better view of what that may be. If that's alright with you, pilot?
Yeah.
Yeah, this crab is probably one similar to responsible for the burrows that you can see to the left right here.
I'm going to slowly
we know,
move in
we
video,
know
but
they're
you can
happy to
zoom if
utilize
you'd
these
like.
burrows, even if it isn't necessarily them
No.
making them. We saw the other day
You could
I believe
sense
that
it.
was a codling trying to. What is this crab doing?
Run away!
No, but it's like doing a wheelie where it's only on one leg. I guess it's because of the sloped surface, but
time to let 'em
that
go.
was really funny.
Nope. Going back to port.
Anyway, there was, I think, a codling trying to get into one of the burrows, whether to hide or whether just to see there was
Seriously,
anything in
might
there that got a
Be
very,
getting that.
very
Not sure.
perturbed and angry crab chasing it
Definitely
out of
comes
its
up a lot
burrow.
better in D2 right
Do
now.
we expect any cables? Because it's starting to look like it comes out.
Yeah, we don't, but
Copy.
doesn't mean it's not there. So we'll.
I got a little more leash. I'm going to push out a little
Yeah.
closer to it.
Yeah, I'll keep an eye.
Yeah, you have it in yours. It's
There you go.
it's something.
Yeah.
meters.
Yeah.
You see something. Is that floating in the column?
Keep an eye on winch tension too,
Yeah,
co-pilot.
have an eye on it.
Whatever the white thing is in front of me doesn't look like it's moving. Okay, so that's on the slope still.
Okay, I think
So it's
I may
about-
see what we're
It's
looking
off
at.
to
There's
my
like an
starboard.
indent straight ahead of you.
15 meters in front of D2.
You see that indentation? Straight line in front of D2.
Yeah, yeah, I see it now. I see the base of it.
I don't see anything in the water column though.
Looks like another crab in the defensive posture.
There's
and a couple
definitely
There's
more
some white objects surrounding it.
blasts of
Continuation of that
rock,
trash we saw
probably
early
from
on.
upslope on the seabed.
Maybe about 10
In
meters
lieu
in front
of
of
these
D2.
rocks falling on this
Did
area
we,
along the toe of the slope, it just gets
It's
sediment
about 30
mantled
meters
onto
from
it. This is
Syria,
a
so
nice
still
view
well in front of
in the
Syria.
floodlights of Deep Discover right now, just showing all that really fine-grained fluffy stuff that just mantles everything down here if it's not being swept away by a current or if it's not having coarser material piled on
I
top
can't
of it.
tell. Am I seeing a point here at the end?
I think we're seeing our first hint of hard substrate.
Yeah, pies beware. Ship is stopped.
Yeah, I
So we'll
agree.
hang out here when we
So it
inspect.
looks like
Copy,
this is
pushing
what
in slow.
Mike was talking about, Mike White, our mapping lead, that there's probably interspersed steep walls. And this looks like a pretty different
pretty
substrate than what we
far.
were seeing in Beach Canyon. This looks like sandstone to me.
Yeah, let's
Fairly
look
coarse grained,
geological
although I
now.
have to
as we
Gonna
get
come
hold
up
my
closer.
a
breath
little bit.
because a lot of what I'm interpreting as pebbles within the matrix, the
Coming
rock
up.
is
Looks like a
probably
pipe.
actually encrusting biology.
I bet it's all sponges.
Yeah, they really throw me
I
off. They
can't see
always
over it.
just look like
Yeah, it looks
pebbles or coarser stuff. But yeah, it
Thank you,
definitely looks
video.
like sedimentary rock instead
Yeah, it looks
of
like
the
a step.
carbonate
Okay.
rock that we were seeing in Beach Canyon the other day.
Yep, and what we are seeing on this
Okay,
rock
all
are
looks okay here. I'm
a
gonna
lot of
head back
different
down
kinds of
to the bottom.
sponges. Probably mostly demasponges is what we're seeing. I thought we might see more corals, but we might see them as we
Let's
zoom
go
in
to
to some of
port.
these areas. Sometimes the corals that we see on these rock outcrops are small or translucent.
Got some fish stirring up the
And this is interesting as well. I'll have to double check
Wall
with the
forming.
exact depth ranges. I was looking at the dive planning template for Beach Canyon as
Alright,
opposed
Bart,
to
so
this
I feel
one,
set.
the
I
Intercanyon.
can put your
And it looks
sonars
like we're at
back
a
to
similar
low
depth
resolution.
range right now.
Yeah.
So we can
If you don't
work it.
As we were in Beach Canyon, and yet the lithology or the type of rock that we're seeing is completely different. And that goes to speak that even though over relatively short ranges you can expect to see the same kind of rocks because the depositional environment was broadly similar, we're a fair amount far from where we were at in Beach Canyon the other day. So probably the depositional environment has changed laterally in this case. And that's what is responsible for this different substrate that we're seeing
Yeah, it looks
right
like
now.
it kind of fades off here to the left. Let's look down it.
So we're just getting a nice view of this rock outcrop, looking
lower my
down the
gains a
slope,
little bit,
and
too?
it looks like the nice habitable area sort of goes away. It's just been, the sediment's been scoured away right in this location that we first came upon. So as we look back up
I know
slope,
you're catching
maybe
up
we'll
to me, but I'm
see
gonna go off
more
right
interesting
along it.
things.
I may steal your swims
Seeing,
if you
I
don't
think,
mind.
some orange
Looks like we're starting to
anemones
get into
spotted along this
Yeah,
rock, along
Your
with
call.
steeper
small encrusting
features.
demasponges.
Video, I'm gonna be still in
And
the
there's
light,
a
so it
small
may change.
overhang, and I do like overhangs because things tend to like to live underneath overhangs.
Pilot, could you scrape this wall?
Yeah, I can. With the vehicle.
Yeah, with the manipulator arm.
Stand by one.
And if you've been tuning in with us
Watch
for
the
a
pilot.
number of times,
Go ahead, pilot.
Be a little easier and I think quite a bit quicker if you would be okay with us just kind of scraping our toe of the vehicle down it
Oh yeah, that would work
as
too.
opposed to the manipulator.
For sure. Yeah, that would be great.
We can also get a little more weight on it than we can with the manipulator.
Even better. All right. Yeah. So
Your swing arms are still so you
right
should-
now
Copy.
I have requested to my
So,
pilots, and they
I got
are
my
indulging
left toe
me
down.
to look at this
And we're
rock
gonna
wall and
scrape.
kind of just scrape down it with the toe of Deep Discover. And what I kind of want to see is just the rock behind
great.
the hemipelagic drape, as well as any encrusting organisms that are on that, just to see if I can get a better idea of what type of rock it is that we're looking at here. If we were on land and we saw an outcrop like this that was covered with vegetation or something like that, you would want to scrape away a trench or something like that,
That was a good
or
spot. Dead center.
take a push core within the wall
Yeah, see
to
if
get an
I can't.
idea of what it is. So I'm kind of trying
We
to
could
do
always try and
the same thing at
exhale on it.
1,200 meters depth.
Section sampling. Blow the first layer away.
Geology is a hands-on science.
Yeah, it totally is, even if we can't actually get our hands on it down here, where we would be very, very flat if we tried.
I have good eyes
It
on.
seems to me that this substrate seems relatively soft.
Yeah, so you can see, watch lead, we're getting pretty decent force with the vehicle, and we're not really penetrating too much.
Interesting.
Almost,
Okay.
almost like clay.
Yeah, so
I'm going to try one more a little bit lower.
yeah, if you could get on the same place that you were at before, just to confirm that it's really that hard.
So that was actually my second scrape in that area. I'm trying one more just to the left here. A little more on the ridge. We'll see what it gives us. Yeah, kind of same results.
Very clay.
Okay, yeah, and if we could get a zoom of one of those
Yep.
exposed places.
Let's go ahead, video. We can turn fog lights on. That might help.
You ready for forward lights?
Yep.
Coming off.
Yeah, so what we're looking at here, based on just
I'm
the
going to go up to the other one.
hardness of it, as well as the texture of it, the relative lack of grains,
go ahead.
my initial thought was that it was some kind of sandstone. Now it's definitely looking more like a claystone or a mudstone, and it's also one that is extremely over-consolidated. And by over-consolidated, what I mean is that it's had all of the interstitial pore space or gaps between the sediment grains, as well as any water or fluid that would have been present, squished out of it, which results in it being just that really,
It's time to
really hard
carry on, I think.
material that maintains its shape and doesn't crumble away under
Right,
the
Pat.
10,000 pound weight
We
of
can
Deep
stop
Discover.
for a good amount
So that's
of
really
time.
interesting,
We're gonna transition to 20
to see that.
moves
It doesn't
now.
look
We're
like
gonna
it's
have
been
more slope.
entirely
I agree.
lithified or cemented,
Still
which is
260
something
looks
that
good for
Jason
me.
Chater of the USGS told us can happen, that sometimes
Off
the
to the left.
necessary chemical reactions required
Yeah.
to provide cementation and turn mudstone into something like
Okay.
shale doesn't necessarily
Squat
happen
lobster,
in
looks
deep
like.
sea, but at the
Yeah,
same
let's
time
snap
it
for
still
squat.
does get really consolidated to be able to form these
Good
really
eye.
steep slopes. That was interesting and a good little experiment to do. Thank you
Pilots
for
will get that
indulging me,
move on.
pilot.
Yeah, we're
Yeah, that's
happy
enough
happy
geology
to
for
watch
now.
lead.
And now we've got the cute
Go ahead, Nev.
little squat lobster. This is a Munidopsis squat lobster hanging out in a coal. We saw that on our dive 2 days ago at Beach Canyon. This squat lobster was in its hole, and
don't know that
apparently
we
they
need
like
to
to
set up.
have
What do you
dirty
think?
claws.
Video?
So we often see
Okay.
them with mud coating their pincing claws.
Okay, let me move them into center for you then.
Yeah, Copilot, you can kind of see it still kind of looks like it was
And
roughly
this
the
little
same
squat
artifacts.
lobster has found
Kinda
a nice
vary,
home. It could have even
they don't really
probably
change,
burrowed
Yeah, I think we
out
can
some
come
of this
wide.
mudstone. Looks like it would have been soft enough for it to actually
Not
create
sure
that
that's
burrow
the
on its own.
shot worth setting up for, yeah. Continuing uphill.
So probably what that mudstone was is just this kind of ambient hemipelagic drape. At some point when, during a low energy time, it could be similar to what we're experiencing right now, and then just really consolidated or packed down by the weight
Heavy there
of
at
sediment
30.
and rock being deposited on top of it. And I believe from reading that I was doing the other way, the main difference in whether that mudstone actually turns into something like shale and whether it doesn't is the presence of groundwater discharge
right.
moving through it. And that kind of brings the chemical ions that are needed to produce the chemical cementation between the grains and turn it into actual stone instead of just this really, really dense packed together clay particles that we were seeing there. So you wouldn't necessarily expect groundwater to be flushed through those materials at this relatively deep depth if this was something like mud being deposited in a shallower environment, like in something like a river delta, that would be present and that would eventually lithify and turn into your classic mudstone.
Starting to get off to my starboard. I only have light pool.
And we're definitely seeing the steep slopes that were really apparent when we were making this dive track and why we picked this, but the lithology is entirely different from what we were seeing in Beach Canyon. It's interesting.
Got you there at 2.5.
And one of the advantages and one of the appeals of submarine canyons to marine geologists is the same reason that
maybe it was
subaerial or terrestrial land-based canyons are interesting to our land-based counterparts. Is you get these kind of sheer wall outcrops that give you a nice cross-section of the geological record without having to dig trenches or cores to look at it in a vertical section. And doing that kind of stuff, digging trenches on land is laborious, but overall not too difficult. In the deep sea environment, it's very, very hard, and collecting cores is a really high effort operation.
Diving down and
there are specialized coring vessels, including the JOIDES Resolution from the International Ocean Discovery Program, that are set up to take boreholes hundreds and thousands of meters, actually, hundreds to thousands, not hundreds of thousands, miles below the seafloor to collect continuous rock records.
Sea star up there, upper right.
Ah, yeah, there it is.
It looks like another one of these similar outcrops to what we were seeing below.
And now that we're here and we've done our geology experiment, let's zoom some animals. Seeing some neat sponges, and there's a star, a sea star.
Have I had my 15 minutes of geology, Megan?
Yep. You can have more geology later, but right now it's biology time.
Standby video.
And pilots, we spaced out that last move.
So with the rate of sedimentation in this area, how much sediment we're seeing, it looks like this sponge on the rock here has gotten pretty covered with sediment, which is why we're probably not seeing too many corals in this area.
Go ahead now.
But as we zoom in, there are some. So this is a sea star neomorphaster. Chris Ma's called in a couple of times to talk about this sea star. He's a sea star expert at the Smithsonian, and he told us that this particular sea star is what people like to refer to as a living fossil, because we have seen this type of sea star within the fossil record.
Understood.
So it hasn't changed its body form and the way it looks in many, many generations. And it looks like this sea star has a shorter leg compared to all of its other legs, and that's likely because it lost that leg at some point in the near future, but has thus regrown that leg. So sea stars are pretty unique in having this ability to regrow their legs
got it.
on a
Go
whim
to the sponge.
if they lose one. So that's pretty cool. And one of my fun facts about sea stars that I enjoy
change
sharing.
of color in the Sirius upper left-hand side as you come up. You might want to head that way.
Yeah, that would be a pretty useful ability.
So you said you saw it.
And also,
upper left
as we're
significant.
zooming in, we're seeing.
So let's go to the larger sponge to the
despite
right.
the lack
so
of
come out partial.
charismatic
Sirius
corals
okay.
and all, we're
Lighter
seeing
coloring.
an interesting range
Not
of
bad.
encrusting organisms, gastropods, some demosponges, the ever-present
And I think we'll
not
want headlights for
xenophyophora,
this.
but still foraminifera, single-celled organisms,
Ready to come up?
and good-sized sponge.
Yeah, this is a
Coming up.
really good sized sponge. And it's in a very strange place. And it's also fully
Go ahead, video
like
when
covered in
you're
sediment
ready.
on the top of the sponge. So I'd like to get a nice good zoom of this. This is definitely a demo sponge, and it looks like it might be a massive gemma sponge, but I'm not sure if it's a geodia. I think I might want a sample of this. This might be something that the sponge experts that have been calling in a number of times about the sponges we've collected might be a new record, if not a new species.
Go ahead, Nav.
I'm trying to.
Easy stop, please.
Okay, easy stop.
But I don't think we've seen this particular demo sponge before, at least within our expedition. We've seen a number of different kinds of demo sponges. We've seen these orange small encrusting ones. We've seen the white encrusting ones. We saw that my sponge cake sponge that had a yellow frosting on the outside and interesting cake-like consistency on the inside. But this is definitely a different one. So if you're comfortable, pilot, we'd like to set up for a collection of this sponge.
Understood.
Bridge, we are stopped.
Thank you, Bridge.
All right, pilots. Sirius have some swing, but not too much. Ship stopped.
Thank you, video.
And I was actually kind of curious when we zoomed in if it was going to be a sponge or another piece of white rock that we see in that
Maybe
crevice on
Yeah.
the right side, but it
Just
was definitely a
realizing that
sponge, and
there's gonna
it's
be a
definitely an interesting
stretch.
one at that.
Let's go ahead and get rid of fog lights. Please.
See that coming off.
So pilot, just for your reference, this sponge will be pretty dense, so it should break off relatively easily. Might use a little bit more force than some of the delicate sponges we collect.
Roger that. Are we expecting it kind of to rip, or do we think the whole thing will just kind of pop off the base?
It should rip.
All right.
You're spun up. And I have the port.
It's sort of
file
like
box
a-
ready to go.
Copy.
Like a pound cake.
Arm coming live. Arms live. Let's stand by.
Yeah, we might even just grab the whole thing.
Let's go in on main Zeus. Let's go in on main Zeus too. Well, actually, did I hear you say you just kind of go for the whole thing?
Yeah, it's a lot smaller than I thought it was, so you might be able to just pick the whole thing off this rock pretty easily.
Okay. wide, please. We'll get that section. Co-pilot, can we come out on?
Coming out with a drawer. Bring mini down. Stand by one. Back out. Port side extend.
And we're looking on our mini Zeus camera, I believe, which
approaching.
isn't available on the live stream, but we can see that the short fin squid mafia, now they're on the main camera, have joined. They always seem to show up if they're in the area. They like D2's bright lights. I think they make it easier for them to feed. And they're here, and they're probably not going to leave. Not that we want them to.
I think the rest of that rolled underneath the vehicle.
Let's go partial on main Zeus, please. It's not.
Pilot, would it be possible to pick up the piece that fell, or did it go too far away?
No, we can go get that. That was going to be my next. I thought I could get a little bit more of the base, but we'll definitely go get what fell down below.
Thanks, that'd be great. Yeah, the stuff that's on the rock couldn't possibly regrow, so we can leave that there.
Okay.
Go ahead and switch it back if you're ready.
Yeah.
Bring it back up, Minnie.
cradle it.
So I think as a bonus to our piece of sponge that we're collecting here, looks like we also might get a very consumed piece of squid as well, the short fin squid that we've been seeing. So it's just what my heart has always desired. So
Can measure down.
nobody loses in this situation.
Are you gonna have some calamari then?
Yeah, it looks to be pre-chewed calamari, which takes out a lot of the work of the equation. So, oh no!
Holding there. Coming back in with a drawer.
Hold the drawer. Sorry about that, watch leads. We'll get it for you.
No worries.
Hold there.
We may push back.
Yep.
Copy. Continuing out. Closing. Oh.
Hello, squid. Yes, yes, we are taking a sponge sample. These squid have absolutely no fear of deep discover.
Sure, man, give you a view of
To the contrary, they really do seem to like, I can't think of another word, hanging out around us.
All right, watch lead. Sorry that took a few attempts and took a little time there, but
Oh, no worries, pilot. We're glad we got it. So we have collected a really interesting demo sponge sample, and there are a number of people working on
Yeah, nicely done.
hashing out the genetics of these sponges and identifying sponges from this region. So that's a really important sample for us.
that I understand what you mean now. I think you said sponge cake when you were describing it.
Yes, it's like dense, but like
Yeah.
cakey,
When
kind of
the
dense.
first time the jaws went into it, you could kind of see that it wasn't so much ripping as it was just sort of taking out
Breaking,
the fingers, the exact finger tracks of the jaws. And then when the jaws closed, it kind of split into two. So the second and third attempts there were trying to cradle the sponge so that it wouldn't continue to fall apart on us. So again, thank you for your patience.
Yeah, no problem. It's hard to know exactly how those sponges are going to feel and react when we try to pick them up. A lot of these animals are very delicate.
Now this is getting back into our Beach Canyon wall rocks. These are carbonates that we're looking at right here. And you can see the massive blocks indicate the kind of mechanical strength that those carbonates had to form the incredibly tall cliff walls that we were looking at yesterday. So I was a little surprised to not see this material at all during this dive so far, but I think what we might be looking at is just some regional variation in depositional rates that cause this sequence of carbonate rocks to be a
Just
little
out of
bit offset,
curiosity,
I
Nav,
think,
when did we call
from the
that
ones that
easy
we were
stop
seeing in
on the
Beach,
ship?
but I'm going to have to go back
Time
and
was easy stop
look
at
at
14:31.
the exact depth ranges
31,
just to
so
see how they
15 minutes,
correlate.
Yeah.
16 minutes.
It does look very similar, and this area is going to, if it keeps staying this rugged like this, we should be seeing some nice corals. I think I spotted a coral on one of these boulders already, and a couple of sponges as well. So some exciting territory. We're also getting to the steepest part of the slope. We've just approached where our map has indicated that steep slopes will start occurring, and now we're seeing this rugosity, so that's a good sign. Possibly if we keep seeing this type of rugosity in this area, it might be a good idea to lateral along it to see and characterize the communities here.
Camera's at 45.
Copy that. Thank you.
Yeah, this looks like we're in a talus field right now, sourced from further upslope. So we're probably not at the source of these rocks just yet. We'd have to continue traversing upslope to see that, but we're getting there.
I'm excited
Coral
about
there.
this coral.
Looks like two of them. Anthomastus down below.
And there's your Venus flytrap anemone.
There's always got to be one on every dive.
Tilted
There's
up.
actually a number of corals on this rock.
Yeah, you got one in the back there as well.
So I'm seeing a pink Anthomastus coral, some cup corals, and a bamboo coral. And then there's another coral in the upper right-hand corner,
video, we can
just
go
off
ahead and
screen,
come
that
in.
is something completely different.
We got quite a bit of things to look at here. Science video, feel free to just drive me around as you see fit.
Okay, yeah, so let's zoom on some of these cup corals and the Anthomastus and characterize those. And then just above this, there's going to be a bamboo coral. So let's just take a little tour of this area.
Copy that. Feel free to come in, video. Frame up this larger guy to start. Bring it a little more to center.
So the common name for this coral is mushroom coral, and its scientific name is Anthomastus. And it gets its common name because when this coral pulls in all its polyps, it looks like a little pink mushroom, sort of like from Super Mario Brothers. And there's a little black
Come
coral
down.
just to the right of that mushroom coral. Very
Coming over
small.
for the black.
So this could be a Bathopathes, a very small Bathopathes, a type of black
Holding
coral
there.
that's known for its sort of feather-like branching.
Is that full zoom video or do you have more?
Now you'll notice that the polyps on this
Copy.
coral look different compared to the Anthomastus. It doesn't have those pinnate tentacles, and that's because this coral is not an octocoral, but a hexacoral, meaning it only has 6 tentacles per polyp. And you're only seeing the
Copy that.
2
We'll
main
come down to
tentacles.
the
There are
lower
2 very
left.
large tentacles for each polyp, and the other
Spin
4
over.
are
Feel free to come in
smaller.
on that lower left. Or center now. Okay, I'll center
Now
up a
these
little
are
more.
hard corals that are a single polyp, often called cup corals.
Yeah, I'm just going to push it a little.
That's right. So our mapping lead, Mike White, has made the comment of these corals are growing on what appears to be fossilized corals. So in shallow water, you have reef-building corals that are hard corals, creating
Yes,
this
I can do that.
limestone.
Start heading that
And
way
then
now.
we're having hard corals also grow on top of that ancient fossilized coral.
Copy that. Come down a little more.
We're also seeing some yellow encrusting
Oh.
demosponges and
She's
white
running.
encrusting demosponges.
Okay, holding there.
And all those pores and channels within the sponge body help this animal pump water through its skeleton, and that brings food to the cells. Also brings oxygen.
Go ahead. I know there was something of interest, more of a stock. Up.
Yes, pilot,
Straight
it
ahead,
is
above.
just out of screen, up. Just pan up, and you should see it right there.
I can go ahead and get a little bit closer for this one if you'd like.
Oh, that'd be great.
Yeah, I'm going to come up. Video if you want to come full wide.
And then we have our customary photobombing squid.
See how much I can sneak in here without disturbing anything.
I don't think they realize that we're trying to look at corals right now. Another fun fact about hexacorals, that black coral that we were looking at, is they're the only group that actually has 6 tentacles for each polyp. Other hexacorals have multiples of 6. And that fun fact was supplied to us by Scott France from the University of Louisiana, Lafayette.
So are you saying that this is reef building shallow water corals that
Paths, just to
this
keep
rock
things
is made
moving.
of?
How do you feel about a 10
That
meter
that
move?
was my guess. I'm not a geologist.
What's your
Yeah,
heading?
well,
Go ahead and start coming
if,
in.
if
Bearing?
that's the case, and
260.
those ones did
about
require
250.
shallow water to survive, that
Seems
would
to
really
keep the two
push
center.
the
Either one
date
of those
of
is
this
fine.
material that we're looking back really, really, really far.
Go ahead and try
There's
fog
no
lights.
way that the area,
What's
the
going
depth that we're at right now, would have ever been exposed subaerially just due to glacial interglacial cycles. That's only about 100 or 120
Okay,
meter
feel free to
depth
take
change
as
when
much or
we're
as
in
little. I'll come
over
up a little.
1,000 meters water depth. So that would put these formation, if it is fossilized shallow water corals, towards a time when the Atlantic Ocean was still in the early stages of rifting, and this area would have been a shallow water environment. And I was trying to look up kind of the chronology of when that would
Alright,
be,
I
but
copy.
I
Coming
will
Move at
suffice
10
it to
meters,
say as
bearing
really
250
old.
at 0.2 knots.
Well over tens of millions of years, which wouldn't be unusual in these canyons. They tend to incise down into some really old rock, but that would be old even by old rock standards.
Yeah, sometimes when you see these rocks, you can actually see some
Lights
fossils
coming
in
off.
it, and like
Copy
see the
that.
fossil,
Three,
the
two,
preservation
1.
of
My
the
Z-bias
coral
is...
calyx.
No, that's not what I wanted to
Oh,
do.
there's a cool
can you check my,
hydrozoan on that rock.
Pressures real quick?
Yep. Verticals. You're looking okay on verticals.
Is that an octopus climbing up the
Yeah, I
rock
think you just
in the
got murdered for a
background?
second. You're all
Yes,
set.
it is.
Oh, we got good sight. I didn't see that. That's pretty cool. It looks like it's doing pull-ups.
Yeah, but I really want to see
There's
this
one more
coral on
coral
the
on the back.
backside of this boulder.
Sorry about that video.
But
Come over
But
that back
octopus.
corner.
It'll still be there.
Okay, I'm taking your word for it.
It looks like, you know, it's
Stand by.
trying out its octopus rock climbing skills. That's a pretty large rock, so hopefully it'll
You look
keep
at that
doing
lower
its
right
thing
there? Yeah,
while
what
we
is
take a
that?
good look at this wonderful coral.
What's
Come in
that
on
on the
this real
edge of
quick
the?
on the edge of this rock video.
Is that an octopus right there? No. Oh, that's the
No.
Venus
So
flytrap.
that's
there's
the
your
back end.
Venus
Yeah.
flytrap
Feel free to
anemone
come out.
on that small
I was
boulder.
thinking the same thing as well. when
That's
I-
been my personal Where's Waldo on this expedition, is finding the Venus flytrap on every dive. And there has been one on every single dive during this expedition.
They're pretty common down here,
Might try
so we
and
do
come up
see a
and
lot
look
of
down on
them.
it.
And I'm looking forward to seeing this pink coral that's coming up on this rock. I believe it's most likely a bubblegum coral, known as Paragorgia.
Your starboard is running a little hotter, so you might not fully have your verts back.
Copy. Should I try and flush them out a little? Or?
Yeah, if you can hold for the shot,
Yeah,
probably just go for that.
copy that.
So bubblegum corals, I believe, got their common name based on their very pink color and how the ends of the branches of this coral are rounded, sort of like bubblegum balls.
Yeah, she's definitely
ROV nav, bridge.
not fully there.
Position move complete.
Okay, come in quick video if you'd like. Coming over. Down. Understood.
And if you happen to notice the squid kind of running into rocks, that's because D2, as we were talking about before, these organisms aren't used to getting bright lights
She's,
at this depth, so they're just momentarily blinded by our lights, and their navigation suffers correspondingly. But they'll be okay once we leave.
Yes, so this is a bubblegum coral. And what you see wrapped around the branches is
coming
a
up a
snake
little.
star.
Yeah.
So we often see these snake
Yeah, I'm
stars
losing her
associated
fast.
with bubblegum
You're gonna come
corals.
I'm gonna pull back and then come up and down. That's alright with you, co-pilot?
Yeah, that was a
I
good
need to clear
view,
that
pilot.
out.
We can move on and check out that octopus, I think, that was climbing up the rock in the
Copy
background.
that. Stand by. One watch lead.
A true
So you don't need to
rocktopus,
back
that
up
one
too
is.
far. You can just run your verts as you're
Killing
heading
me.
towards the octopus. Yep.
How does that look?
Looks
Kinda
clear.
better. Copy that. Alright, I'm gonna push up into the starboard port.
Yeah, up to port.
Yep.
Kind of see them on the
Yeah.
sort of center on my screen.
Copy that.
Yeah, your verticals look clear.
Good. Yeah, I got a little inverted there.
Just a little. Should be in there somewhere.
Copy that. I see it right there.
I see it. It's right between the rocks, sort of
Go
splayed
ahead and
out.
start coming in video.
Yeah, it's not climbing anymore. It looks like,
I'll push it a little bit closer.
is that a defensive posture?
I'm not sure what this octopus is doing. Sort of like it climbed up and now it's trying to float back down.
What a graceful landing.
That's what's so good about having so many suckers on your tentacles.
Can we get lasers, please, pilot? Yeah, so now we've got the lasers on. It looks to be on the order of the same size as the ones
Starting to
that we
drift
had
pretty
before.
far away from
You might
Away?
be thinking about
Yeah.
these
Copy.
with other octopi that you might have seen in aquariums
about
and
a
stuff, but these
vehicle
ones
length
are
away.
a good bit
Understood.
smaller than ones I think that you're used to seeing,
Come
especially
forward now.
compared to something like the giant Pacific octopus, which, as the name implies, very large.
So,
And this one's definitely on the move.
could you come out video? Thank you.
Yeah, looking great.
I really enjoy watching octopus behavior. You never know what they're going to do.
Okay, should be steadier there if you want to start coming in again. Coming down. Copy. Come down, following them down.
Yeah, Scott says they've seen this behavior before where the octopus have been climbing up rock walls. They enjoy rock climbing just like people do, I guess.
Holding there.
Now we've got a screen full of cephalopods as our short-fin squid, our perpetual short-fin squid, move by because we haven't paid attention to them recently enough, and cephalopod envy is real.
Well, that's 3 cephalopod species for one dive so far.
So far, so good.
Pilot's clear when science and video is.
Yeah, I think we can let this one go. We got some good video of it doing its thing.
Copy that.
Let's see if what he does
Understood,
when he
thank you.
jumps off this rock.
You
And
want to
then
come
let's
back
move
in
on.
for the rock jump?
No, this is a good view. I think we zoom too far in, we lose some of the information of how it moves. It's just holding on with 2 tentacles right now. One big step for octopus, one small leap for D2.
I was going to say leap for cephalopods. Yep, looks like it's just looping
Yeah.
around and then
Looping around like, okay, that was it, end of the show.
Copy that.
All right, we're good. Thank you, pilot.
Feel free to come out video full wide, please. So, pilot, I'm gonna
Though I
take
could
off and
probably
push
watch the
forward.
octopus all day, if we're going to be honest.
oh, absolutely.
But I also want to see lots of corals, and we've only seen a couple so far, so gotta
Yeah.
hold out for my corals and sponges.
Gonna come back to center and come up over this feature.
And we might even see more octopods. I feel like this would be a great place to be an octopus. There's so many cool like holes to like hide in and like
Actually.
things to do in this area.
So it's like a
Yeah, this
coral view
is
over there to the
a really interesting a lot of rugosity that you're
What's
seeing
that
right there, a
stuff?
lot of holes and stuff like that.
Something yellow
That
there.
speaks to the way that these carbonate rocks evolve, which is a lot different
looking
from
to the,
sedimentary
on the
rocks,
report.
Not seeing
because
anything
these
for you.
aren't only mechanically weathered or kind of broken down by
You wanna do
physical
a quick zoom on
processes, which
this
might include
object over the
current
report?
undercutting or mass failures or anything like that. They're
Sponge?
also chemically
Sponge.
weathered
Copy.
because carbonate
Forward.
or calcite is a basic material and reacts
think I
with
saw that yellow that you were
acidic
talking about.
compounds or substances, which can include, sometimes seawater can be mildly acidic. It happens a lot more on ground, which is why you get lots of caves and caverns and stuff, for example, in
snap zoom
areas
on
like
this
Florida.
Yellow
But
feature.
these materials are being chemically and mechanically weathered at the same time, which is why you get a lot more kind of terrain.
Copy. Full wide. I'll set up for it.
So I spotted a yellow coral. Oh, and there's a little octopus in the crack. See, told you we'd see more. It's like an octopus's garden here. But we're not on an octopus watching field trip. We're on a coral and sponge characterization mission.
The octopuses do a lot more interesting things,
Corals do interesting things.
like sit there.
Just because you can't watch them move doesn't mean they aren't interesting.
That's what I tell people about rocks.
I was going to
You already
say.
come in? Gonna come over.
So we're getting a nice good look at this yellow coral. I think we actually have two different kinds of yellow coral. This bigger colony that we're
Take
seeing
as much as you'd like.
looks like a plexaurid coral, perhaps a paramuracea, with a snake star associate wrapped around the branches. And you can see where the polyps
Come
have
up a little more.
closed. They've been able to retract really close to the skeleton.
Rotate out to the edge. That's full zoom. Copy that.
So that's a really nice view of the polyps of this coral.
Coming up a little on the rotator.
And we often see these snake stars associated with plexaurid corals and paragorgic corals, and sometimes with
Holding there.
corallium pink corals,
And holding.
but less likely with bamboo corals. And also less likely with the canthagorgic corals, which is, I think, what I saw, a small colony of that, only a few polyps on the rock. So in addition to just cataloging what different kinds of corals and sponges we see, we also think it's very important to catalog what associations with other animals we see. So do we often see the
Video's
same
clear,
animals
pilot's clear.
associated with the same species of coral? Is there a
Copy that.
preference? And by making these observations, we can learn more
Continue
about the
forward.
community and the diversity within these communities.
Okay, coming up and off. If you want to do a snap zoom on the octopus in there, just so we see.
Oh yeah.
Sorry. It's a big eye.
Yeah, that octopus has a really large eye. So oftentimes octopods will find
Forward.
small caves like this to lay their eggs in, and they will protect and stay with their eggs until their young have hatched and moved
Got
on.
another one up here to the left.
There's
I was
another
gonna say,
octopus
looks
in the
like
hole.
there's-
So I was just thinking all these holes in the rocks would probably make good habitat
Looks like another coral
for
as
these
well.
octopuses.
Might be able to set up
This
for
is
that
like
one.
a octopus fortress.
And Patch, just to keep things moving, how about another 10
I mean, if
meters?
I was an octopus, this would be a
At,
great place to hang out.
Yeah, 260. How's that
I think
sound?
we're seeing another type
great.
of coral. This different yellow coral. It'd be a good to get a nice snap zoom of that pilot.
Copy that. I'll set up for it.
So this, I believe, is the Acanthogorgia coral,
So
the other
something
yellow
else in
coral
there.
that we often see.
Go ahead.
Is that a differently colored Venus flytrap in
Go
there,
ahead.
or are we just
Black
not
in the back
bright in the
there.
lights with it?
That's a completely different species. So the orange one is
Copy,
in the
10
family
meters,
Hormathiidae,
bearing
and
260
that red
at
one
0.2.
is an Actinoscyphia.
Okay, go ahead and comment on that video.
So as we come in, we're going to take a good look at these
Feel free to work
polyps
with
to see
Copilot
how
to
they're
get those
different
fog
from the
lights
one
on.
that we were just looking at. The color itself is different, which tells me that it's probably a different type of coral, and the bushiness of this colony tells me it's different. And those are the characteristics that are more like Acanthogorgia. Also, if we were able to see some of these polyps and the sclerites in the polyps, you'll see that they're sort of rough and spiky looking.
Copy.
There we go. And this coral has associated aplacophorans. So those are those
Coming up.
animals in the mollusk phylum that are sort of like
little
a
further.
snail without a shell. So they don't have a shell, and they tend to feed on corals and are associated in near corals. So this is definitely an Acanthogorgia. As you watch these polyps close up, you'll notice that they're not fully retracting in towards the skeleton, and you can see little spikes along the top of the polyp after it's pulled in its tentacles. And that's what they use for defense instead of being able to fully retract in, and that's a character of Acanthogorgia.
Copy that. Coming down, starting.
I think it would be a good idea to maybe get a sample with those associates.
I'll let the camera tell the story.
Yeah, in the chat room, Scott Francis saying that he's not sure that aplacophorans have been collected from this
Okay. Did I
area,
hear them
so
say
that
they
would
want a
be a
sample?
new record.
Alright. If you want to come full wide, I'll get set up. We can get a little bit closer for this.
Lights coming off.
Cover that. Thank you. So this may be get the sample, take off, put it in the bucket. I don't know if we'll have enough room for
RV
the
nav,
drawer.
bridge. Position move complete.
Do you agree with that? Copilot?
Yeah, unless you want to come around left to it, you might be able to get more drawer
There's
out.
a really big boulder
I think
that
I
we're
might
on
have a
top of
better
right now. If you
view
look
of
at
it
camera
coming
feed
kind
2,
of down.
you can see
yeah,
Deep
sounds
Discover in relation to the boulder. Deep
Looks like you have
Discover
a
is about
shark,
10 feet
I agree.
long,
small
and
shark
10,000
above you.
pounds.
above me.
This is this
not worth
boulder that we're
leaving the
looking
coral.
at right here in the scale of slope failures that we observe
Oh, yeah.
globally would be a relatively
Yeah, kind of
small
from
one.
this
This
angle,
would probably
come
be
down
on the
and slip
order
towards you,
of,
but
if I had to guess,
they do want the associates, so
a couple,
you want
maybe
to make sure you can still
10
see
or
that.
15 cubic meters of material, when landslides that are globally significant are measured in cubic kilometers instead of cubic meters, just to give you a sense of scale.
And by its should be a stop. Sirius may have a little bit of swing, but she will not swing past D2.
You want to come in a little bit closer? Video, please, on the main Zeus. They said associates. Do you see? Yeah.
Yeah, they do blend in relatively well. They were on
Or in the middle.
one of the center branches.
How big is it? Can you just grab
No,
it?
If you
that's
want to
an
come out,
amphipod.
yeah, I might be able to grab the whole thing. We'll get lasers
Yeah, they were
on it.
easier to see from the other side. It was
Understood.
right in the middle of the colony, about halfway down the colony that I believe we were seeing them.
That's pretty big, huh?
You'd be able to get your claw in there towards the base
I
and
agree.
take most of it.
Take most of it. All right. And then we might be able to kind of like.
It shouldn't.
Feather,
I think
yeah.
it'll fit in the box
Copy that.
without any issue.
Do you think we can open the box where we are?
Yeah.
All right. Do you want to ramp me up?
You're
Stable.
ramped.
Copy. Thank you. If you don't mind bringing up Mini on your screen.
Got it.
All right. Gonna approach from the right side. Come left. Cutters near the feature. Shot coming live.
Tilting down real quick.
Yeah, that she runs quite a bit.
Yeah, you'll probably want to give your jaws a little shake too.
Yeah,
See
a
if you
little
can. There you go. Tilting down.
Copy, thank you. Okay.
I turned your grip force down as well. So you might bump that back up.
Standby video, just right there is fine for now.
We're
Go
gonna
ahead and
take
take
lasers off.
lasers off.
Yep.
Exactly. I think something right around there is going to be
Do you want
decent.
to look with fog lights or anything?
Yeah, if you want to put fog lights on and come in just a little bit
Might
on
shine
Mini.
up your base a little.
Yeah, I think
Not
I
sure.
need to rotate the jaws slightly more.
I don't know if that helps or not. Maybe too many shadows.
Okay, come closed. Yeah, I'll wait for the ship roll. Oh, that was high. That was very high. Stand by.
Yeah, you might want to get it more into the palm of it too to get the snippers at the base.
Okay.
So I would hold that. I wouldn't release again.
Yep.
At this point, I would just tug on the base and see what comes.
Alright,
Fog lights coming off.
copy.
Assume going for port.
Yes.
Ready for drawer.
Ready for drawer.
Pushing the vehicle.
Copy.
Mini Zeus coming down. tilting up. I can give you a little
Can you
more
come
draw.
out? Yeah, if you can come out a little bit further on the drawer.
We're pushing. I'll try.
Yeah, that's fine.
Extending.
Okay, copy. Thank you. Okay.
Closing.
What do you think about reaching in there trying to grab that piece
Okay.
of fill?
Just
not-
got
I don't think you'll be able to.
I think you'll
sample
be able to get
of
that.
Yeah,
coral.
angle's gonna work
And
against you too.
Hopefully there's one of
Tilting up.
those chitons somewhere on that.
So chitons are the
Obscure.
polyplacophorans that have many plates. Aplacophorans have no plates, no shell. So these are sort of, they're in the same phylum, but they are different. I know. It's the biology. But you're doing so well for a geologist. I'm very proud of you, Jeff.
Coming out, joy lock, coming off.
So it
Yeah, I don't
does
think
look
I'm
like
gonna
we
be able to
lost
reach
the
it in
top bit of the colony that had the aplacophorans on it. So I counted this at the Acanthogorgia as our main specimen, just in case we didn't get any of those aplacophorans. But I really hope we did, because
sitting on that
Kevin
Venus flytrap.
Cocott at the
I
University
think
of
it's below
Alabama has been working on the
turn
genetics
90
and
and look down, you
taxonomy
might
of
be
these
able to see,
animals.
to
And there
starboard.
haven't been any known from this area, so it's a really great observation.
I'm going to leave you on HG2.
Copy. Thank you.
And watch your port
Yeah, that's a
side
pretty
pilot.
deep crevasse.
I'd stop there.
Oh, she is on there.
Oh, it's like resting
up
right on
sled.
top of the anemone. It made the anemone close.
I think that'd be
Sorry, an enemy.
pretty challenging.
I don't think that's, possible. I think it's outside our realm of.
Kind of rude to drop coral on its head.
I don't think
Yeah,
the risk.
it'll be
even
fine.
get
It'll just
into
open
that
back
through that
up and
crack.
that piece of coral will fall.
And it looks like this rock that we're looking at right now might be chalk, a subgroup within carbonates, instead of, I don't know whether, chalk might also be a limestone, but it's not a dolomite. I've been really doing some remedial carbonate
Anything else in
researching
this
yesterday.
immediate area?
Pilot, can we snap zoom on the wispy colony that is bottom center screen on this rock?
Would you like to come in quick?
I just wanted to confirm that this is that hydrozoan that we were seeing at Beach Canyon, and it definitely looks like it is. So
Copy
that's
that.
a good observation of this very large branching hydrozoan. There's also some cup corals and encrusting demisponges on this part of the rock.
Understood. Feel free to come out video. I believe it's just a...
Yeah, we're good with that. And we're seeing some more Paramuricea, that yellow, bright yellow coral with the associated snake star. And what are all these little brown spots on the top of this rock? You notice that
You wanna
those
do a
look like
snap zoom on the
biology
brown
to
spots,
me.
please?
Yeah, definitely.
They're foraminifera, so little gatherings of these single cell organisms, foraminifera, all on this rock.
For what?
I totally thought they were going to be like
Thank you.
little soft corals,
We will continue.
but you know, that's why we have to take a look at these things. There's a large red crab hanging out on this rock outcrop as well. And on a smaller boulder is probably another Paramuricea, that bright yellow coral. There's a couple red crabs hanging out.
Just gonna get over this feature first before I come back down.
Yeah, it actually looks like past that outcrop we were just on. Looks like it flattens a little bit.
Copy that. Go ahead and make some moves then.
Do we want to stay at this depth, or do we want to continue uphill?
Yeah, that's a question for watch leads.
Go ahead now.
I think it would be nice to go along the contour to the south, if we can. Yeah, I was just looking at the slope along this area, so we did end up landing a little more to the right of where we were going to go, and so I'm thinking if
the
we
first
head
one will need to pull us
south,
back off
we're
the
going
slope,
to
but
cross over what our original transect
that
was planned was. So
I got you.
that's a nice hot spot of steeper slopes, and hopefully we'll find more of these rock outcrops in this area.
Yeah, and this carbonate area that we're in isn't an outcrop, it's a debris field. So I think if we go along it, this is probably going to be just a big rubble field. It'll serve the same purpose substrate-wise, but I don't think what we're seeing is the same actual wall of the canyon that we were crawling up and down in Beach Canyon. So this material is probably sourced further upslope.
Lateraling over to port, and then I'll come back down. I guess the question is, how far back do we want to come?
I think back kind of where you were is a good starting point.
Okay. Back where we took the sample, or back where the octopus was?
Probably last sample, maybe. Start there.
Copy that.
Yeah, around there.
So Sean, I would make my first one, I don't know,
so we get a bird's eye view
150,
of this rubble field.
15 meters maybe?
15 meters,
one of our octopi.
And that'll take me off the ridge line and
directly
south.
into a pretty deep crevice on that rock.
Ready for it?
Ready to go.
Aim to get
probably a whole bunch of them living in this area.
Is this where you, I guess this...
Yeah, this is where So this is where I got the sample from. Yeah, because like this one comes, then it dips down. That's right where the top part of that one is.
All right, pilots, if you're all set, I'll call on that move.
Go ahead, Matt. A little bit. See if I can look up underneath these while we're moving.
One 5 meters, 150 degrees, decimal 2 knots.
Good copy.
When you're done here, pilot, I'd have you turn 180 and kind of see if you can look into what's coming our way.
Copy that.
So we're just reorienting our two ROVs, Deep Discoverer, which is camera feed 1,
there, so
and Sirius, which is camera feed 2. Sirius is smaller and further up the tether to start transiting along the canyon wall, oriented east-west instead of the north-south transect that we had originally planned.
We may have just gotten lucky with that rock feature. Not really seeing a whole lot in the sonar.
Yeah, we can do a couple moves this way, and if not, we can always revert back up slope.
I believe that ghostly looking stuff hanging in the water column right there is squid ink, or as Mike Vecchione apparently calls it.
It's a pseudomorph, which is a
you
squid
need to keep
ink
coming
and
apart.
mucus mixed together.
You want me
The
more to
squids use it as a
Yeah.
distraction
Sort of
uh,
I sort of
in
want
order
you
to get
just
away.
turn to port and drive kind of along.
And we've
slope.
certainly seen a lot of squid in
'Cause
this
that's the
area,
way we're
so
trying to go.
definitely
Yeah, I
not
was gonna
surprising
saddle
to
along
see ink from them.
Do that. Thought we were just heading west.
So we're looking
No, we're
along the
looking
slope
south
right
right
now,
now, trying to-
and
Yeah, we're gonna
it's,
move
as you
south.
can see, it's pretty similar to what we were
along
seeing
the contour for a
before.
little
A
bit.
lot of that
The first ship move was a little southeast.
just gently sloping substrate, and I think again that field of carbonate material that we were seeing, that we
So
just
I'm
spent
past south
a while
now.
looking at, is sourced from further upslope. This is a debris field as opposed to an outcrop of the variety that we were looking at in Beach Canyon the other day. And one of our fish experts on land, Ken Sulak, has provided a wealth of identification of fish seen so far into this dive, and also noted that just in general that even though this really fine bottom substrate may not be good for our charismatic sponges and encrusting corals, it is really good for fishes in terms of just all kinds of the
You're lined up on the direction of travel.
epifaunal and infaunal stuff, things that live on
not exactly.
or within the
last
sediment.
move was 150, but now I'm pointing south.
Yeah, you could probably cut like direction
I'm
of
sorry,
travel
I'm
on
just
slope.
interrupting myself and looking at this little red jelly thing drifting in the water column. I don't know whether we're going to be able to
after
get a
this.
chance to get a zoom
Yeah,
on
after
it,
this,
but
our move should be like 180.
these are the kind of
Yeah, yeah.
things that
Your nav bridge.
Mike Ford and
Hey, that
others
moves
on land, our water column experts, would probably be
chase it,
interested
you
in.
bet.
That's a pretty cool looking one. I'm going to assume
Plenty
some
of
kind of
tether and we can
cnidarian,
clear.
but I don't know beyond that.
So this looks like the Paralia medusa. So it's a scyphozoan, and more related to the jellyfish that you're used to seeing in shallow water. And it's pretty distinctive with its dusky red color. That's my best guess based on what I learned during the midwater dive a few days ago.
It doesn't look like it has any tentacles, though, or
I
if it
think we
does,
can
they're all
probably start
withdrawn.
heading south.
Yeah, it does. It's just
Copy that. You want
it's
to start with 20?
holding them in and floating away.
Sounds good.
So
20 meters due south.
if it just extend them, we would see them. But I can check my little drawing guide.
Go ahead.
Let's
We're
see
gonna
if
let 'em go here soon, but
that
yeah.
is what we think it
Yep,
is.
we're ready.
I think I drew something that looked a lot like that.
20 meters, bearing 180 degrees, speed decimal 2 knots.
Yeah, but don't take my word on it. My expertise in jellies has been limited to what I've learned and picked up from
180, I'm
all the
pointing that
midwater
way.
folks who have
Copy that.
called in
I'll
during
go
our dives and
perpendicular to
shared
you and
their
saddle along.
identifications in the chat room.
I'm gonna come down and get closer to the bottom as well. Which may significantly affect our delta.
Yeah, and as we're drifting back over this fine-grained benthic material, as I was saying, this is apparently more favorable substrate for bottom-dwelling fish in general because it provides a lot more faunal diversity for them to feed on as opposed to the hard grounds, which are typically food poor. And I thought I qualitatively noticed that during past dives, kind of a inverse relationship between the amount of hard substrate and the amount of fish we were seeing, it was really apparent on our couple seamount dives, but it's good to get that confirmed from Ken Sulak, one of our shore-based scientists. And he also makes a notion that has been kind of stuck in my head since he mentioned it a few dives ago, the need for a kind of maybe a benthic crawler or something like that that would be able to image the bottom in as much detail as we get of the really large, bright and colorful and frequently mobile organisms that this focuses on. Because while those might be the most conspicuous creatures that we see down here and the ones that really immediately get our attention, they might not be representative of the biomass and biodiversity overall observed in these environments. Kind of like an iceberg where what you see above the surface is only a small amount of what the total is. So that's a good note, and just another kind of general notion that the scientific community that we're serving on these dives is anybody who has any interest in this area in general. This looks like a mermaid's purse, as they're frequently called, which is a elasmobranch, either a shark or a skate or a ray, which are all in the same family, and I'm not using family as the taxonomic way because I don't know that stuff off the top of my head. Sure. But an egg case from one of those creatures, which isn't too surprising. We see a fair amount of rays and skates, as well as some lantern sharks at these depths, and I think that's the similar kind of reproductive strategy they use. So when we were at Bear Seamount a few dives ago, we saw an absolutely enormous pile of these mermaid's purses that from a distance fooled me into thinking it was igneous material because they're really dark color that's the same as rocks.
Something's coming. Rock, maybe.
This is a rat tail, I believe.
I'm now
which is one of the top
degrees,
predators in this abyssal
the
environment.
Despite
being not very large, they're very prolific, and we see a lot of them on these dives. That's another kind of keystone species that we observe in these benthic environments, particularly, as Ken Sulak was saying, the soft sediment environments. And here's another carbonate rock that we're looking at here, probably sourced from upslope of this area.
Nav bridge move complete.
Go ahead, Nev.
Ooh, here we go.
We're headed towards the orientation along the wall of our original waypoint. Is that correct?
I believe he's muted.
It's going to be very dependent on what we see along the wall. It's definitely along steep contours. It's going to take longer. It's hard to estimate. Once we do a couple moves, I can get a better
Okay. Yeah, I was just conferring with our navigator.
that
The
string-looking thing?
ROV team
You wanna
that
zoom in
works
real
with
quick?
us
Video? Maybe
to
fishing
pilot
line?
the ROV is 3. The
Yeah, it
pilot
looks like monofilament.
that controls the Discover,
Come out, please.
co-pilot, and nav. And we're looking in on this with very keen interest, not necessarily scientific, just to figure out if this is, it looks like fishing gear, it might be line.
Do you
Whenever
agree
we
with
see that,
Co-pilot?
we're always really keen to figure out whether it's suspended into the water
You're trailing
column
off on your
and
words.
whether
Oh,
it
do you
might
agree
be
with
a
that?
navigational
Yeah,
hazard.
It doesn't look like it's any harm, though. I don't see any up in the water column or.
We've seen
the
several
vehicle.
times during this expedition trawling gear or fishing gear suspended in the water column, and when we see that, we want to give it a good berth
And
because
Pilots, that last move
Deep Discover
is
and Sirius
nicely
are
spaced
directly
out if you're
tethered to
interested
the
in another one.
surface, so entanglement
Ready for another
is
20
a
meters due
hazard
south?
that we
Ready.
want to avoid.
Copy that. Coming up.
So it looks like we're back in this, what I think is that really highly consolidated but not cemented mudstone stratigraphy that we were looking
ahead,
at
Nav.
earlier that I had the pilot scrape to see what it was, unless it's just carbonate that's really heavily draped in sediment.
20 meters, 180 degrees, decimal 2 knots.
So when you're settling like this, you want to make sure. If you're
Pilot, could
facing
you blast the thrusters at this wall real fast?
if you're facing the wall, that you're constantly looking at your port rail or whichever direction you're going, looking at your rail cameras and looking at your manip cams to see if you're going to lateral into something. Otherwise, your other option is every once in a while, periodically turn heading to see what's ahead of you, and then turn back.
Copy. Watch the. I don't really think we should blow thrusters, but we might be able to get the suction sampler to blow some out in front of us so we get a view. Are you just trying to dust the top layer off, or?
Yeah, exactly.
Copy that.
Yeah, just make sure you point it away from the fishing line.
Yes, I'm just trying to get a closer look at this
Fishing
rock
line
just
should
to
be off to my starboard.
confirm that what we're looking at is that really highly
wanting to do
consolidated
suction?
clay.
Yeah, reverse suction.
It's interesting to
Draw
see
out.
this crab within an
yes, a little
outcropping
bit.
there.
Okay, extending drawer.
Copy that, coming up a little.
Hello, squid. Hi.
Copy. Go ahead and reverse section.
Stand by one.
Copy.
Okay. We are blowing.
Probably want a significant amount. There we go.
1500 RPM.
Copy that. I'm gonna come in a little bit tighter. Do we have any more?
I think that should be good. Thank you.
Let me try again.
Yeah, so we just used the suction tube in reverse to kind of blast
See if
away
I
some
can
of that
get you a little bit
hemipelagic
more.
sedimentation. What I just wanted to see was whether this material we're looking at is just that
It's
carbonate that we
2500.
were looking at before, that rubble field, but was just mantled
zoom
in
in
so
on
much
that
sediment
video?
that you couldn't actually see that distinctive white color. But it looks in this case that
Okay, feel free to cut section.
what we're looking at
Cut
is
section.
more of a
Off.
Copy that,
sedimentary
thank you.
material
Feel free to
instead
bring
of
the door
carbonate,
back in.
Door
I
coming in.
believe.
Yeah, come out. Did that satisfy your curiosity?
Yeah, thank you.
Excellent. Okay, pulling up, pushing away from the dust cloud.
I just wanted a quick check for what we're looking at is, I think what we're looking at right now is the actual canyon walls instead of
I think that,
just a debris field
kinda
that had
allows us to see it a
fallen
little
like
better
that carbonate
without
material
having a huge
below.
dust cloud
It would be
under
really
the way for it to
unusual
settle.
to be along the same depth contour, which is how we're moving right now. We're no longer moving upslope, we're moving along slope. So it'd be really unusual to
Irritating
see
something about settling.
canyon walls change their material so abruptly.
Looks like you got
But the
something
carbonate
white,
was probably sourced from above,
triangular,
which
in your
makes sense.
picture.
Different type of sea star as well.
Yep, and that's a carbonate rock that we're looking at right now. Tyler, can we get a zoom of that sea star, the fatter looking one to the right?
Sure. Do you want to just zoom in on this real quick video? Definitely make sure that it's a rock.
Yeah, so
Yep.
that is just a very starkly white piece of
Go ahead and
carbonate
come out.
material
Thank
sourced
you.
from upslope, really heavily cemented, and just a textural contrast to the mudstone that we're looking at right now that's predominant in this area. So we're going to come in close and look at this sea star right here. I'm not sure whether our onshore expert Chris
Nav
Ma
bridge move
is
is
watching
complete.
the dive. He might be enjoying himself on this Saturday morning instead, but having the video archive for identification purpose will help him. Go ahead, Nev.
Go ahead and start coming in video. And I don't know if we need any more lights or anything than that, but you can work with Copilot to get on fog lights if you'd like.
Yeah, so we're looking at a sea star right now. Megan has taught lunch. We do tech team lunch so that there's only one of us here as scientific advisors on the dive. And so I can't provide too much information.
I don't think I've seen this one before.
I know there's something different about the
Coming up.
got those little tendrils coming out from the ends of
Okay, coming to the one.
5 points and the lights of it.
Copy, coming up.
I seem to recall Chris Ma saying that
Coming over a little bit.
tendrils are extended. That the star isn't feeling for
I'm not hearing him now.
heating or moving position.
Coming up.
Yeah, sorry about that. I didn't have my Okeanos full out channel open. Yeah, so we're looking at the very corners of the sea star right now, and we see these tendrils. They have a name, as everything in biology does. I can't remember what it is right now. But apparently when those are extended, that means that it's kind of content and happy to feed and move. You could see that it's very slowly making its way downslope on those, I believe they're called polypodia, the little feet on the bottom of them. And they can actually move the star at fairly fast rates when they have to move, which is interesting. They don't, I think that, aha, is that
Hello.
Chris Ma here?
It is Chris Ma.
Hey, Chris, how's it going?
Saturday, and I'm just sitting back enjoying my Okeanos feed and working on some taxonomy in the lab.
Fantastic.
So, yes, so what you've got here is a sea star called Pteraster, and Pteraster is one of the sort of popularly known animals called slime stars. And these are called that because they emit a mucus slime defense. And if I were to burden you with a collection of this animal, which I'm not going to because I think it's an unknown species, its collection would no doubt be enhanced by the fact that when you brought it up, you would have a box full of slime, of mucus,
because it was probably agitated. In answer to your earlier question, the things that you were focusing on earlier, those are the tube feet, also called the podia. And the tube feet that are right on the arm tips are usually used in a sensory capacity. And so when they're moving, they're kind of feeling or possibly even sensing the sort of environment around them in anticipation of which direction to move. The arm tips are where the ocelli or eye spots are kept. So in these deep sea species, they probably don't do much because it's dark all the time. But the tube feet are, especially on the arm tips, are quite elongate, and so it's using them to sort of feel around its surroundings as it moves, among other sort of senses that sea stars have that we wouldn't, because they're pentameral rather than bilateral. Other species of pterosaur that I've seen generally feed on sponges and are thought to be detritivores. So it's probably just looking around for the next tasty thing, or sort of maybe even stopping to occasionally devour some, encrusting stuff on the surface as it moves along. So good. That's the second species that we've seen in this canyon, other than Neomorphaster. And so I'm always happy to see what else lives in these, because the Atlantic is generally considered to be not as rich an area for diversity as the Pacific. So as far as sea stars is concerned, but a lot of these deeper areas aren't as well known. So I'm always happy to be surprised.
All right, pilots, we've been stopped for
Awesome.
a while.
Thanks for
Wait another 20 minutes
the
out.
context and the elucidation, Chris.
Sure, happy to do it. Looking forward to whatever you come up with next.
Absolutely. All right. Thanks, Chris.
Have a good day.
RV Nav Bridge.
Yeah, so we're continuing down the walls of this box canyon in between Alvin and Block Canyon.
20
and
meters,
it's interesting
bearing
as well.
180 at
This
speed
is
.2.
probably the source of the steep slopes that Mike White, our mapping lead, identified to make this a good dive track. But it doesn't seem like this substrate is as ideal of a habitat for the corals and the sponges as the carbonate stuff that we were seeing the other day in Beach Canyon. So this is interesting. When we make these habitat predictive models to look for habitat suitability, slope always ends up being heavily weighted. Slope or steepness in the predictive skill location or predictive likelihood of seeing corals and sponges, particularly corals. But as we can see here, the lithology or the type of rock is not as conducive for the habitation of these organisms in this area as opposed to the carbonate stuff that we were seeing yesterday. So this is kind of an example of why biologists should take geology into context when they're doing this, because in this area, even though we're seeing similar slopes, we're not seeing the same type of diversity and density of corals that we were observing yesterday. And it's just an example of how pretty much everything in oceanography is a necessarily multidisciplinary endeavor, and really no field can kind of stand alone, and you're going to be having some serious blind spots if you're only studying, for example, the geology. If I study geology, it ignores bioturbation, which is a really important factor for the mixing of sediment and the ultimate lithification of the sediment in time.
Might be clear.
And Jeff, this is Mike White here. I totally agree with you. I think this is something. So I've been with OER now about 3 years. Done a number of these ROV cruises, and we love to target these steep slopes. And I remember a very specific dive on the western Florida escarpment. Dan Wagner was our expedition coordinator, was actually our science lead. Adam Skarkey was on as a geologist.
you
we
look
were
like
diving,
you might
and
be, clear
the three of them, well, two of them plus Scott
Your
France,
axial
we're
might
having
be
this
acting up
conversation
as well,
that,
your port
yep, we can
axial.
target these steep slopes, and yep, we can find hard bottom where hopefully these corals and sponges are settling, and they, of course, create the texture and the rugosity needed in order to increase biodiversity, right? That's where the trees of the deep sea, if you will. And it's not just the slope of the geology that's controlling this, right? And so now we have to think about, well, maybe there's some sort of physical oceanography, right? Or are they not having the right nutrients being delivered to them, or is the salinity not right? Is the temperature not right, or is there some sort of trace element that's limiting the sort of dense deep sea and diverse communities that we would be targeting? And it's tough to say, but it's so fascinating that in one area where we see similar rocks and similar geology and similar slopes and similar rugosity, similar multi-beam data, from one day to another, seeing different types of communities and how those communities are driven, right? It's not just what we have in front of us, right? We need to think about some other environmental, physical, and chemical factors that drive these communities.
Yeah, that's a great point, Mike, and that's why when they're doing these habitat models, they're not to throw too many 10-cent words, but multivariate models, as you were saying, that integrate a lot of those other factors, because if it was as simple as just steep slopes were all they would need, well, that would be a lot easier of a problem. But as you pointed out, there's a whole lot of other conditions that are required for them to live and thrive in these areas. Dissolved oxygen, you said current speed, all of these have to be taken into account and are weighted in different ways in these habitat suitability models to kind of find the Goldilocks zones where everything is right for them to thrive.
Not because something was stuck.
So speaking for somebody who works for NOAA's Office of Ocean Exploration and Research, one of the questions we continually ask ourselves is what sort of baseline observations, what sort of baseline data are we encompassing, and what does it take to characterize the seafloor? And I can speak for myself as an individual that I don't know I have those answers, and I think we have a number of groups within the office that are working to divine those answers. Right, but there's always a question of, are we capturing the right baseline information in order to do some sort of characterization of the seafloor, right? And we know we capture mapping data, we know we capture video data, and of course the RV has a CTD and we have a sampling program, right? But what other physical or chemical or biological data points from an exploration standpoint, do we need to capture in order to say, yep, we have some baseline fundamental observations of an area? And I think we as a program, often, look internally and say, okay, what are we looking? And then, of course, we in turn have to ask the community, right? What other information can we be capturing in order to assist in those, predictive modeling, right? What sort of factors do you need, and how do you weight them, right? What would our priorities be?
Yeah, those are all really good points, and speaks to how fundamentally different OER is as a program than almost any other research organization out there conducting oceanographic research, because what we're doing here isn't research, it's exploration. And basically, it may sound like a kind of branding thing, but it's a really, really fundamental difference in that what we're doing is going to areas that are unexplored and just getting, as you said, the baseline data that scientists can follow up on, have publicly available, all of our stuff mere months after these cruises become public domain and are accessible to any researchers. They can take these interesting observations that are made and follow up on them and design experiments around them and test their own hypotheses on later expeditions.
A big picture, if anything, the takeaway is when we do see these dense, diverse, deep sea coral and sponge, communities, and of course other communities out there, when we do see them, if anything, it just really speaks to how special they are, right? They don't occur everywhere that we have sea slopes. They don't occur everywhere we have these rock outcrops. You need a certain confluence of special, factors within the deep ocean in order for these communities to survive and thrive. And so when we do observe them, right, it's a recognition of how special they are when they are there.
Absolutely, and it's part of one of the first questions, again, is just kind of working on how how broadly distributed they are. And even that, we're still working off the habitat suitability models as just kind of placeholder data in lieu of us being able to go and ground truth every single one of these sites and see where they are or where they are not. And bear with us for a minute now as we're undergoing an ROV pilot shift change. But it's these organisms are, probably fairly widely spaced. They probably have a relatively narrow range of conditions that they're happy at. And the other thing is that they are fairly easily perturbed in terms of bottom contact of the variety that happens in deep sea fishing or, trawling or activities like that. And that's why it fundamentally underpins the importance of established marine protected areas to conserve these habitats where we know these corals and these sponges are observed to grow. Right now, we are actually diving within the Frank R. Lautenberg Marine Protected Area, which was established specifically for conserving areas where we know a lot of these corals and these sponges are known to be found from previous ROV dives conducted within this canyon, Veach Canyon, Oceanographer Canyon, and other ones.
Check, check.
And Mike, you mentioned the physical oceanographic aspect of habitat suitability.
It's like,
this dive that we're on right now
it's
is
up,
relatively close to what is
strafing,
called
if you
the
will.
Pioneer Ocean Cabled Observatory, which has
Copy
been
that.
operated, I believe, by the Woods Hole Oceanographic Institute for a really long amount of time. So this is south of Cape Cod, Woods Hole. Woods Hole Oceanographic Institute is one of the longest living and most successful oceanographic programs and communities in the country. And the importance of us doing this dive and doing this kind of baseline biologic community assessment is we can relate conditions here biologically and ecologically to the really long and continuous record of physical oceanographic data that we get from the Pioneer Array, which is taking measurements continuously at various depths in the water column and has been for many years.
All right, with the co-pilot.
So it's always good when we can tie our observations in with longer lasting ones.
And worth noting there that the video data that everybody is seeing on shore and on our live streams, and of course our mapping data, right? These are just, they're major data sets, but they are two components of data sets here on the ship, and we of course collect many others, including meteorological data, physical ocean data, of course, sea surface temperature and salinity. We're completing XPTs, and so all of this data, including the video and the mapping data, all of it is publicly available and can be shared in real time.
If we need to turn it back on, I'll holler. So
and would you check out this octopus that we're seeing
Sounds
here
good.
right now?
They might be able to
Oh,
see
these
some
are
of
great. This is the fourth or fifth one that we've seen on this dive, and not to be one-upped by
Heard that
their
might be
eight-armed
doing
brethren. We
thrusters
see
occasionally.
a couple short-fin squid coming into the footage, but yeah, we've seen an interesting range of behavior from these octopi on this dive so far. A lot of them are really happy to hang out in the really three-dimensional, very rugose substrate that the carbonate debris field that we were looking at provided.
Depth, I guess.
This one is kind of hanging out
What
on
was-
the
what was the last few
bentos
tracks?
and climbing up this
Yeah, we're
lower
going due
landform
south
in this
180.
consolidated
Just
mudstone
180.
So
right
they
now.
wanted to keep sort of in this direction
Appears to be doing
until we get
pull-ups.
sort
Never
of
skip arm day.
perpendicular to waypoint 2.
Okay.
Then they want to start moving back up the slope.
Okay.
So previously 20-meter moves.
So I think we're
at
going to take
180.
a zoom on this octopus right now just to see what he's up to. He or
Sounds
she,
good.
that is.
Bridge RV nav.
seen this octopus?
You want to get some headlights and still camera?
RV Nav's bridge.
Hey, Bridge, I'd like to put in a move request, please.
Headlights coming out
Range
video.
20 meters, bearing 180, speed 0.2.
Roger that. 20 meters, bearing 180 at speed 0.2.
Roger, thank you, Bridge.
Got you up there.
Yeah, and just as a quick addendum to something that I said earlier, the Frank R. Lautenberg area is actually called the Deep Sea Coral Protection Area. It is not a marine protected area. That is something similar but different.
So it looks like
so now we're getting
this
a nice,
depth, it's a couple of minutes, 3 minutes. It looks like it's noted that you guys you should feel that in serious.
I don't know whether those are
Okay.
bivalve shells
Coming in.
that we're looking at to the
Can you
left
frame it up
of
there?
the
Yep.
Yeah, I think they are actually.
Okay. Can you watch my thruster
I'm
raise?
gonna
Look like
come out
either
just a little bit to get
mussels
his
or
arm.
Sure.
Probably
I'll
not
let you know if I have to
scallops.
do anything. Oh, he's touching. Big stretch.
I love this just slow motion like climbing that these things do.
I'm going to tilt down. Can you zoom out a bit?
Okay.
I'm going to frame up the still camera, so it's not going to be centering your camera, but.
And I think this is
Just for a
the
little
dive
bit.
where
Okay,
we've seen
that's
the
full
most
wide.
active octopi in terms of just moving around.
Put his arms down.
Doing cool octopus things.
Oh, I just got hit, I think.
What happened?
So many cool octopus things.
They look okay.
So this is what the final dive of our expedition should be like. Full of octopods.
Yeah, I see a cloud above you.
And we got some corals and sponges as well.
Yeah, even better.
Port side.
And there's really good mac and cheese. Really good mac and cheese.
It's really stretching out there, huh? I want to get a still with the arms up. Just a little more video.
Do you want me to zoom?
I'm just
Or-
telling you, we're going to hold this framing for just
Okay.
a little bit more.
Gotcha.
I think we got it.
Just wave your hands in the air
Okay.
like you
Okay.
just don't care.
Okay, go ahead.
So everybody at home,
Holding.
you know, you stretch out like an octopus. Do some moves. This is your octopus interpretive dance time.
Right. Kill this
This
still cam.
octopus will lead us in our perfect cephalopod yoga.
Oh, again. Let's come out video.
Okay, coming out.
You want me to turn off the forward lights? Or leave them on?
Sure. Turn them off.
Hello,
Yeah,
Okeanos.
my port axial.
Hello.
Or port, sorry,
There's
port
Mike
vert.
down here.
Hi, Mike.
Trying to get my phone to show me the video and
Looks like it's
while
clear
I'm
now.
talking. I'm not being very successful at that. But I just thought I'd say a few things about the octopods you've been seeing.
Yeah, we'd love to hear more.
Okay, these are warty octopods.
Let's go full wide if
Genus
you're
is
not already.
Granulodonta.
I'm full wide.
Okay.
The reason I mentioned the genus is because it's the same genus that on the west coast has been shown to sit on its eggs for up
Just
to
had to clear
4
the
years.
thrusters there video.
That's
I
fine.
suspect the ones that you see out walking around are all males. I know I've seen the modified arm on a couple of them.
What's that?
And I
The
think
white
the
thing.
ones that you see tucked up in the caves might be females, and they may be on eggs. We've seen them
I'm going to move up a little bit. Check it out.
brooding eggs in places like that around these canyons.
The white thing on the left there?
Yeah.
Can we get to center?
Yep. I think I've got enough
That's really
tether to get out
cool.
there.
I know on a previous dive we spotted an octopus that was brooding eggs underneath a rock.
It's like a
4
rock.
years seems like a long time.
You want to go partial?
Clean rock.
Yeah, 4 years
Yes.
is a very
Coming in.
long time.
Gonna hold
Oh
here.
boy.
And
Okay.
so there's actually another question about these deep sea octopods that derived from exploration on the west coast, where they've seen aggregations of some octopods in areas where there was
Starboard bird. That time.
slow seeping of warm water, not hot, not the really hot hydrothermal water, but slightly
Should I
elevated temperature
come out, pilot?
water.
Come out a little bit, yeah.
Okay.
I suspect that involves
Let's full
the
wide.
octopods
Thanks.
actually actively choosing that because the temperature would
These
speed up
squid
the
had
development of the
an
embryo.
extra layer of challenge.
We're not seeing that in these guys.
That's really cool. Pilot, I think I spotted some sort of jelly. Something is blooping near the bottom.
Looping near the bottom.
To the
Yeah, it's real small, pink. It's coming.
Can you direct me to it?
Yeah,
left or
so
right?
it's left of the lasers and up a little bit.
Oh, in the sediment there?
In the sediment.
I'm
It's.
going to let you taste the jellies, and I'll hang up now.
Oh, thank you for
Oh,
calling in,
the
Mike.
one
It's great to
pulsating.
hear from you. It's
Gotcha.
definitely been an octopus and squid centric dive today.
very
Lots
small.
of different things.
Yeah, I saw a bunch of squid pieces. It looked like a squid went through the thrusters on the.
Okay, let's go in.
Okay, coming in.
Yeah, that might have happened. There were so many just swimming around us.
He's disoriented.
Okay, I'll say goodbye now.
All right, thanks. Yeah, this is a jelly. And I wonder why it's so close to the bottom. It seems to like be swimming straight at the bottom, which is unusual for a jelly.
Coming in.
It's like it's
Squid up report.
pulsing straight into the ground. There are some jellies that actually do stay near the bottom of the ocean, or it will lay on the bottom, but this one just keeps swimming down for some reason. But we'll be able to get some really good views of the structure of
go in.
Okay,
this
that's
animal,
max
so
zoom
you
right
can
there.
see the
Okay.
channels and the muscles
Excellent tracking,
contracting.
pilot.
And
It's
all
easy
the
when he's staying
tentacles.
still.
So this will be great for all of our midwater and jelly experts. They might be able to get a good ID on this animal. And it could be this might be one of those upside down jellyfish that does like to hang out near the bottom. It kind of looks like a beautiful flower.
ROV nav bridge, move complete.
Roger, Bridge, thank you.
It's almost mesmerizing. That was very cool, Pilot. Thank you.
Yep, it's
Okay.
come out video.
Coming out.
That was good timing.
Okay, that's full wide.
Thank you. Another octopus.
One more octopus. See, we were supposed to do this
At least
dive
this one is head on.
yesterday, but we had to put it off because the weather was really bad. So this would have been an octopus dive for Octopus Friday. But don't worry, octopods. I did represent wearing all of my octopus gear yesterday for your special day. But I'm really excited that we got to do this dive today because I would have missed out on all these cool octopuses.
Just going to take a peek over. I'm coming down slope a little bit. Maybe let's come back to this ledge, co-pilot. I'll come down here.
Yeah, the ledge is where all the really interesting stuff seems to be happening.
Back up just a little bit too.
So as I look on our mapping screen, I can see that we're right in this nice area of higher slope. So we're seeing a little more rugosity in this
Sea-Star.
area.
Any interest in the Sea Star?
Let's do a snap zoom. I think it might be
Okay, we'll do a quick zoom
Neomorphaster.
here, video.
Okay, coming in.
Yeah, that's what it is. It's Neomorphaster. It's pretty common in this area. We've seen a number of them from today's dive and previous dives.
Okay.
Okay, max wide.
Max wide. You can feel free to zoom past the skid whenever you'd like.
Okay. There we go.
Thank you. That's where I'll stop. You guys want another 20? Let's explore this for a minute.
Pilot, can you zoom that plate-like sponge on the left side of the wall?
Sure.
Some of you were also seeing some
partial video.
Anthomastus mushroom
Okay, coming
corals on
partial.
this wall. We're going to be zooming into this plate-like sponge here sticking out of the wall. I was curious as to what kind of sponge it might be, either a glass sponge or a dema sponge.
Squid coming in.
Copy. Okay, go in a little tighter.
Okay, coming in.
Hold that.
So this looks like it's a glass sponge. I'm looking at the structure of the sponge as we zoom in, and we can see the little holes and channels within the sponge skeleton.
Coming
And
in.
the difference between
Get a
glass
little
sponges
bump.
and dema sponges is what makes up their skeleton. So glass sponges, their skeleton is made up exclusively of silicate, so glass, and they have a very distinct set of spicules, those glass pieces, that determines their species. So a sponge taxonomist, somebody who studies sponges, will look at all these glass pieces within their skeleton in order to determine what species it is. And if it's a new species, they would describe those spicules in a scientific paper.
Am I free
Dema
to zoom, Pilot?
sponges, on the other hand, can have other kinds of particles or spicules made out of things that aren't glass within their skeleton.
You want to try the headlights?
Sure.
Coming on.
As we got a closer look, it might actually be a dema sponge. I'm not sure. It's really hard to tell. Some of these sponges can be pretty cryptic. And I've been learning more and more about sponges throughout our expeditions. Sponge experts from around the world have called in or written in our chat, letting us know what we're seeing. Can we get a full specimen view? Yeah, that'll be great. So for our deep sea animal guide, when these experts let us know what these animals are, we will take an image like this one and put an identification on it so that you and anybody else who's interested in these animals can look through the images on the Deep Sea Animal Guide, which is located on our website at oceanexplorer.noaa.gov, and learn more about a variety and diversity
Do they
of
need
animals we've
lasers?
seen throughout our expeditions.
Get lasers on.
And
Come
help yourselves identify what these organisms might be. So this sponge is just about 10 centimeters across. We know that because those lasers are 10 centimeters apart,
Okay.
which is approximately 4 inches.
Max wide.
And we're seeing small dema sponges spotted all over this wall face.
Okay.
That was a great view of that sponge.
Can you turn the lights off?
Sure. Any interest in this red coral, I think it is, on the left?
Yeah, Pilot, let's do a snap zoom of this one. I think we've seen this before. It's an Anthomastus
Okay,
mushroom
do a quick zoom.
Okay, coming in.
But this is a much smaller specimen than the one we've seen before. And there's a couple single polyps, young colonies, that haven't yet grown additional polyps next to it. There's also some small snails and other demisponges on this wall. That's good. Thank you.
Okay.
If you're just joining us, this is Dive 12 of our Deep Connections expedition exploring the canyons and seamounts of the Atlantic
All right,
in both
I think we can do another
Canada
20.
and the U.S.
Roger that.
waters.
Bridge, RV nav.
RV nav bridge.
Yes, another move request, please. Range 20 meters, bearing 180, speed 2 knots.
RV nav bridge.
So this is the final
20
dive
meters,
in our
bearing 180
expedition,
at
which makes
speed
me a little
2.
sad because
Roger that. Thank you, Bridge.
it's always hard to be around for the very last of something really awesome. But don't worry, there will be more expeditions throughout the year and many years to come.
See what these little wispy things are under this rock ledge.
Okay. The yellow ones or
The ones to the right of that.
beige ones?
Yeah, beige. Go ahead.
Okay.
So our pilot has spotted something that looked wispy. We're going to see what
Maybe
this
forward
is.
lights on.
Stand by.
I think so.
It'll light up from underneath there.
There
Yeah,
we go.
it looks like it's a
Yeah,
Go in
that
video.
helps a lot.
mucus that has accumulated some sediment. And on the mucus, we're seeing a small opossum shrimp, and maybe some amphipods hanging out in the small crevices within the rock.
Okay, you can come out.
Okay.
Next video. Actually, before we go, can you zoom in again? Right
Okay.
where the lasers are.
Oh, did you spot that squat lobster, pilot?
Yeah.
Yeah, there is a Munidopsis squat lobster that's found itself a nice little cave to hide in. We've seen this lobster hanging out in a couple different rock caves. For some reason, these little squat lobsters like to decorate their claws and keep their claws nice and muddy. And I don't think we know for sure why they do that, but for some reason they do enjoy having muddy claws. It could be that they have this natural pink color, and if they have mud on their claws, they can more easily blend in.
Okay.
So
Video's
that's a
clear.
Munidopsis
Okay, come out.
squat lobster.
Turn the headlights off.
Yeah.
So today's dive is at this box canyon, unnamed box canyon between Block Canyon and Alvin Canyon, is a really unique area because it's within the Frank R. Lautenberg Deep Sea Coral Protection Area. And that means this area is protected from bottom activity, so no commercial bottom activity can happen within this area. So that protects deep sea coral areas, the corals and sponges that we've been seeing from being disturbed, because these animals are very fragile and can take very, very long to grow. So we've protected the area, especially these canyon areas where we know corals and sponges grow and form habitat for a number of associated animals.
It's this little fan here down below.
Oh yeah, I think
We'll do a quick zoom.
that's a coral. We might want to zoom in on that.
I'll hold here.
Okay, thanks.
Oh.
Oh, little
There's a small sea cucumber swimming across our view. We've been seeing a number of small sea cucumbers swimming in this area, which
squid
is
storm
pretty
coming
cool.
Squid storm.
All right, go a little tighter.
Okay, coming in.
The squid storm is strong in this area. So
Yeah,
this
bridge
is a
move is complete.
primnoid coral.
Roger that. Thank you, Bridge.
It's very small,
Or I'll
so
stop
I'm not
pause.
sure if
Copy.
it's Thorelia like we were seeing yesterday. It's very small and delicate.
There's They're stirring up the dust, not me, I think.
Yeah.
No, it's totally
Is that
them.
me, Levi?
That's them.
No, you can
That's
see it.
them,
Look at your
right?
manip cam. They're
Yeah.
just hitting the ground all over the place.
The squid storm has kicked
You're out
up
to ruin your
some
video, Caitlin.
sediment.
It's okay. They're like the pigeons of the sea.
It's true.
So we'll wait patiently while this sediment settles out so we can get a better view
Oops.
of this coral.
We're on camera. Would you like me to come out?
Yeah,
Pilot?
come out. Yep. I'm going to invert the thrusters here real quick.
Yeah, the squids keep bumping into the bottom and kicking up sediment, especially right next to the coral we're trying to view. They're being very mischievous.
Could turn my lights on. It might take some of the attraction. I don't know.
All right, so we're still going for this coral, right?
Yes, please.
Okay.
I want to see if we can get an ID on that.
Yeah, it's right there. Yep. Okay.
There's some red corals at the bottom too, Pilot.
Copy.
So this is a small, bushy-looking primnoid coral with very delicate polyps.
Yeah, you want to try turning on your lights?
Yeah. I'll take some of the. Here we go, video coming on. Maybe some of the squid will come hang out with me.
Yep. Okay, let's go ahead and go in video.
Okay, coming in.
Jeez.
Silly squids. They're making this job very hard. Thank you, Pilot, for sticking with us.
No problem.
Yeah, I'm having a hard time deciding if we know what kind of primnoid coral this is. It has very small and delicate polyps. It's bushy, but it doesn't look like the Thorarella that we were seeing yesterday that had a very Christmas tree
Can I come
appearance.
in more, Pilot, or should I stay medium?
Let's stay here for now.
Okay.
Yeah, let's stay here.
Just for a second.
We might want to collect this.
Okay.
It's really small. Do you think you can get it with the suction sampler?
Yeah, we could probably try that.
All jars are free.
Okay.
Copy.
Okay. Should I come out, Pilot?
Yeah, let's come out.
Okay.
I might need to yeah, I need to reposition here a little bit.
Or I'll stop. Got a 19 Delta. I'm like
Copy that.
maybe 15 away from the wall here. From the starboard sides.
Okay.
Pretty close, but yeah.
Looks reasonable.
Yeah. Tether looks good.
Okay.
Yeah, this coral is so small, so delicate. I don't think we know what this is, but it doesn't look like Candidella because it doesn't have that lyrate branching. All the branches seem to be arising from the center. It doesn't look like Thorarella because it has a main axis where the branches seem to be coming off, much like a coniferous tree.
Hydraulics are up.
So like a pine tree.
Auto XY.
Looking
Okay.
through our deep sea animal guide to see if
Copy.
perhaps
Yeah,
someone
starboard
has
side.
seen something similar in another area. The polyps are too delicate to be Narella.
Squid storm.
The squid storms are really making it hard for us to look at this coral.
Do you want to go ahead and extend the drawer part of the way?
Sure.
Wait for this to settle out.
Enabling valve packs. Oh yeah. sure my sample box's pins are retracted. Alright. Coming out.
Though we have seen some Norellas that are very delicate.
Step of 1 meter.
I just keep thinking about what it could be.
Let's try it.
So one characteristic of the primnoid coral Norella is that it has a set of 3 body plates. And when the polyps close, they generally close downwards toward the main axis of the skeleton.
All right. Actually, Nav, could you put up the Sirius camera there? Just give me another camera to drive by.
And Norella has also been seen to have branching near the base.
All right, hydraulics coming on.
Good, pilot.
Yeah, that's great. Thank you.
Yep.
Force 5.
It could also be that it's a very, very young Calyptrophora, or Calogorgia. Calyptrophora has a lyrate branching. Calogorgia has more of a pinnate branching.
I can draw more if you need it.
So we're going to try to use the suction sampler to gather this coral because it is so small.
There we go.
Need to draw in?
Sure.
I'm in. The bungees are not pulling it back right now. See those lines?
Yeah.
Should I draw back out a little bit?
Yeah.
I didn't want to like go too far.
Yeah. Just enough to keep some tension on there. We'll have to see what's going on.
Maybe if you rip the craft up a little bit, maybe the friction against the tube is holding it back or something.
Okay, I can do that.
Retracting.
Still looks a little loose, huh?
No, it went tight.
Went tight? Okay.
Wiggled it.
So I just had the hose like angled too far down?
Yeah.
Okay. Good to know.
I think so. We'll keep an eye on these strings when we...
Okay.
Okay, I'm on jar 1. Be advised.
Copy that. Alright, it should be within reach there, huh? Be a little closer. Get auto depth. those settle out.
Okay. Mind if I pull up section cam?
Sure. Yep.
Let me know when you want me to turn it on.
Yeah, you can go ahead.
600. Increasing, increasing, increasing. Holding, 800.
I think we got it.
Check the jar. Some dust. Nope.
All
Yep.
right. That was neatly done by our ROV crew. Just
Right, you want to extend
instead
the jar?
of trying to use the big manipulator arm to try to get that little tiny coral sample, we just decided to use the suction sampler for it. Hello, squid. Yes. Yes, I see you. Or you're not being suction sampled.
Okay,
Yeah, I
that's
think
good.
that just made a lot more sense. So we're gonna tuck the suction sampler away and then continue on our way.
Nice. Drawing back in.
Okay.
Rigging looks
I
great.
do like having the squids as our constant companions. They're pretty great.
Take my monitor back.
Sure.
And index to jar 6, so there's no contamination issues.
Craft hydraulics are secure.
So with this collection, it marks our first collection of a primnoid coral. So a coral in the family Primnoidae, which is also good. It's also very small. And we have,
reduce the RPM of
and
HPU.
it's just like perfectly preserved since we
Leaving it
suctioned it
enabled.
up.
Copy that.
I can't imagine life on Deep Discoverer before the suction sampler. We've had so much use out of it this cruise already.
It's a really great tool for sure. And in the past, when trying to collect something small or something fragile, it's been very difficult. We thought of some many solutions for picking up these types of animals, but I think this is the best solution so far, because it means we can collect very soft-bodied animals like jellies, very small animals, and very delicate animals.
Yeah, and that just reiterates kind of the fact that OER, Ocean Exploration Research, the division of NOAA that runs the Okeanos program, is really a community-driven organization in that they respond to and build their
Bobby, pilot hears you on deck A, but it's really muffled.
Sound like you're in speaker mode, Bobby.
Sorry. Yeah, is really
Co-pilot deck. There we go.
driven by community,
Yeah, now I have you loud and clear too.
in terms of the data that are
We're
collected, the expedition
wanting to install
sites,
the rinser if now's a good
general
time for you.
area, as well as
Yeah, got a
equipment on
19
the
Delta.
ROV. If there is some sort of
How long are you going to take?
device that could really help with their
Minute.
science, and scientists get together and propose
Co-pilot
a
is
plan
off
for curation and
headlights?
preservation of the data.
We're
that can be
still setting up,
collected by that device,
but we'll
it
call
could
up
very
right
well
before we
be
put it
added
on.
to the scientific payload of Deep Discoverer and Sirius. I'm just shining our floodlights into this little crevice here just to see if anybody is home. It looks like this mudstone is fairly easy to burrow into, and a lot of organisms are using it as little hiding holes. We've seen some squat lobsters so
What
far
is this
using
right in
it.
front of us? You want to zoom in on the yellow-orange thing?
Yeah, I'm not sure what it is.
Rock with the dot on it or something.
This is a very strange yellow-orange thing.
Can I come in more?
I'm not convinced it is a live yellow-orange thing, but it's got some demasponge encrusted on it.
Yeah, this looks like carbonate rock of the type that we were seeing above.
Come out.
It's got some, probably some discoloration on it that makes
the
it
two
that
bulbs on the right
kind of yellow,
kind of looked like eyes from a distance.
Yeah. I was just thinking that.
Yeah, they totally
that
did
it
look
looks
like
a
eyes.
hell of a.
I thought it was like a really weird fish that I have never imagined in my life.
Okay, get headlights off.
Yeah. Just look at that.
Pilot, can we zoom that fish top screen?
Yes, we can.
I think this is a different fish, but we haven't yet gotten a good look at yet today.
Do a
So
partial zoom as I come up.
Okay. Gonna hold here.
Okay.
So this fish looks like it is a cusk eel in the family Ophidiidae. And maybe it'll be nice and let us see its face.
Seems to be aloofly ignoring Deep Discoverer. Oh, there we
There,
go.
it's coming around.
Okay, coming in. Hold here.
Chris, you can switch it to the aft deck cam if you want.
Copy that.
Okay. Gonna
We
come
might
in
be
more.
able to see it
Yeah.
better. I just don't have that one on my.
So this
Yeah,
is
that's
a
what I was
cusk
looking
eel.
for. I just didn't know what it was called.
Oh, okay. Yeah.
And you can tell by its fins. So it has the long dorsal fin that starts close to the head, just behind the head, and runs along the back. And then its anal fin does the same thing on the ventral side or the bottom side of the fish. So they have a very eel-like swimming form, just like the halosaurs and the rat-tail fishes that we've been seeing. And these fish also have these rounder, blunter heads. And I'm checking out its lateral line, seeing that it's running straight from behind the gill slits all the way down. Doesn't have a just jointedness to it. So it's not a live-bearing cusk eel.
And there's a couple of the other, what I've taken to calling in my head, the crunch cukes behind it. Those sea cucumbers that move by doing abdominal crunches. And as we're saying, those are all over the seabed.
Can I zoom in
We've
more
seen
on
a few
head?
of them
Go ahead.
swimming in
For
the water column
science.
before, but they're really widely distributed. We've only seen a couple of the really jazzy, bright-colored sea cucumbers on this expedition so far. Up and there goes one of the, what looks like a crunch cuke flying by right now.
Video's clear whenever science is.
Okay, come out.
Yeah, so this is a really good view of this
Okay,
cusk
coming
eel type of fish in the family Ophidiidae.
That's full wide.
It has a very small, sort of pointed head and that distinct lateral line. It could be in the genus Monometopus.
And we are racing this red crab downslope right now.
These red crabs have been on the move today. I think this one is just trying to find the ledge to hide under because it's like, oh my goodness, what's going on?
Yeah, it certainly appears that way, that Deep Discover represents an apex predator to it, and this is a good example of an animal taking advantage of the cover provided by these ledges. Oh, thinking about coming back out.
Go in if you like.
And these crabs
Okay.
are, as all crabs I believe, are primarily scavengers.
Gonna hold here since
we
he's
saw
moving.
one with an extremely acute interest in a piece of a squid on a previous dive, but all of the short fin squid just kind of circling around here, it probably doesn't have too much interest in because that probably represents way too much effort to get a meal.
Yeah, this crab will probably search out some of those squid pieces that we've been seeing. Oh!
Oh.
Watch
Poor
where you're
little
going,
fish.
man.
No. Okay.
It's really funny how I've seen that a lot, and it kind of is surprising how much just ignoring each other this deep sea life does. And it's hard to know whether that just happens naturally at these depths or whether it might just be a function of we're never
we can let 'em go.
actually
Okay.
observing
We've,
these things entirely under their
stressed 'em out
normal
enough.
Sounds
conditions
good.
because we have
Going
a
full wide.
really big ROV with bright lights pointed at them. The ideal way to go about this would be some way that we would be able to see everything and not, not modify the environment even temporarily just with our lights. But of course, as humans are visual organisms, we require these lights to conduct our research down here and bring you these great live streams direct from the seabed as we're getting them.
There's something on the wall, Pilot. It looks it's like kind of top left of center. Looks kind of like a shield or a fan.
Oh, I see it at the
something.
bottom of the rocky
Yeah.
part.
I can't come down too much more here.
Okay, it's not too far ahead. Just
I think we spotted another very plate-like sponge coming off this wall. And there's a sea spider. I do like sea spiders. They're sort of creepy and awesome.
Yeah, we're Team Sea Spider for sure. Then again, I was just talking to a member of our crew here on the
Lights coming on video.
who
Okay.
says he is adamantly not Team Sea Spider,
yeah,
and in fact
that's-
was chased
well,
around
that's
the
better.
vessel by a sea spider in a specimen jar on a previous cruise by one of the scientists
Coming
aboard.
in. Gonna frame up both.
That's not something I would do. So this large sea spider is in the family Colossinidae, probably in the genus Colossus, because it is so colossal. These spiders can get up to 30 centimeters across, so very large with their legs. And then the sponge next to it looks like a plate-like glass sponge.
Deck A, this is Nap.
Go ahead.
Just did a status check on the progress of your rinser.
We have not started
Sea
yet.
spiders are extremely
Copy that,
bizarre
thank you.
creatures.
We're still getting everything hooked up. We'll call up right
Do you see that?
right when we're ready.
That brittle star, like
Roger
on
that.
the
Thank you,
sponge?
Dex.
I
think I do, yeah.
That's
terrestrial
weird.
Spider
Wanna look
neighbors.
at it?
I
Yeah.
think that name is just out of superficial resemblance.
go for it.
Okay.
Yeah, they
I'll
do
bring the
have
head
the
over.
8 legs like the arachnids that we do see on land, but they are not. They are only very, very distantly related.
Huh. I guess it got behind
So
the sponge,
they
and,
belong
like in that
to
hole.
the Pycnogonida. And this is the largest family in size of the Pycnogonida.
Yeah, you can see what looks like those really kind of underdeveloped or modified
it in a minute.
claws that
Okay.
it has tucked
Sounds
underneath it.
Yeah, those modified claws are actually they're not all sea spiders have them, but this family does, and
You got
they
any more
use
zoom?
them
I believe I do.
for mating purposes.
Yep, that's max zoom.
Okay.
I was reading something about them, and they have a special name that I can't quite remember what they're called.
Everything has a special name in biology, Megan.
Yeah, I know. And everything has really interesting special names that if you study an organism long enough, it becomes part of your vocabulary, and you sometimes forget to explain them. Sort of like when we talk about the osculum in sponges, which is an opening, like this demosponge that we're seeing that has a round opening at the top. That would be an osculum.
Yeah, and on cue, Scott France on land is here to inform us that
Come
those
up.
specialized mating arms
Coming
are
out.
We're still
called
off
either
stick on the winch,
ovigerous
right?
or ovigerous legs.
We can use it if we need to. But
Yes.
Co-pilot nav deck.
Yeah.
Never
Well,
mind.
no. Now
Go
that
ahead,
I
Deck.
hear it,
Full wide?
Yes,
it
we are
rings
now
a
ready
bell,
to install the
but I just
rinser
couldn't-
at
it was
your
like
convenience.
something had to
We
do
are
with
go.
eggs.
Roger that. We're installing.
Because
The
ovum,
headlights off.
and that's why I was thinking mating.
Thanks.
So a lot of the names that we have in science are related to Greek or Latin words for things, and so a lot of the animal names we say have meaning because they are named using Greek and Latin roots. So sometimes for these animals, if you know the name, you can remember what it is based on what the name means. One good example, I'll try to think of some. It's hard when you're on the spot.
I don't see any squid. I might turn my light off. I think I can see you better. With
Sure.
it off.
The only
Video. I'm
The
gonna
only
turn my light
one that
off
I can
there.
think of right at the moment is
Okay,
not based
yeah.
on
It looks like it's got
a
a more
Latin
turbid.
name, but on a Hawaiian word is the gold coral in Hawaii. And in Hawaii is called Hula Mana Mana Haumea, which comes from Hawaiian words meaning branching and golden, because it's a gold branching coral.
If you can keep following us, Dan.
Yeah, I guess that's what they want to do. Just keep following this ledge.
There goes another crunch cucumber making its way across the water column in front of us.
Pilots, another 20 once we're done with the rinser.
Yeah.
Sure.
Absolutely.
And a xenophyophore, that basket-like or spaghetti-like mound that you see, which is a really small single-celled organism, but
Getting in the 12
builds
mark,
these
realized.
combs out of sediment and mucus or protoplasm
Ideal.
stuff to sizes much larger than the organism itself.
Our next move, we can either move it deeper or we can come up. I think
Let's
the two
get
options.
some hard returns there, or maybe it's just the gain, but we can go investigate, see if there's anything of interest.
Talking about like here.
Yeah.
Okay. Yeah. I agree.
If I stay kind of stretched out, I can come up a little bit.
Okay, another example of an animal name with the Latin roots is the Chrysogorgia, which is a type of coral. Chryso means gold in Latin or Greek, so that's why the common name for the group of corals is golden corals,
flat.
because their skeleton is golden in color. And that fun fact was supplied to you by Scott France.
That's cool.
This looks like a rubble field. Is this all dead coral, Megan?
It most definitely is. We've zoomed in on a few pieces, and it looks like hard coral, something like Lophelia, that sort of brambly branching coral, which we have been seeing quite a bit in other dives within the canyons. That suggests that this coral is growing here somewhere. And their skeleton has been transported likely downslope.
Go in video?
So
Sure.
as we continue our
this guy looks
journey
different.
during this dive, hopefully we'll see some of that
Hold in
kind of
here.
coral. Right now we're zooming in on an anemone and really getting a good look at the mouth of this anemone, which is in the very center of the disc.
Can I zoom in on
Yeah, we usually
mouth?
don't see them as fully open as this.
Okay.
Okay, go ahead.
Okay, coming in. Gonna hold
See, that's
here.
a really good screensaver. I actually, my screensaver is a slideshow of all the best images from Okeanos dives because the imagery is just so
Go a
beautiful.
little tighter if you got it.
Okay, coming in.
Yeah, every single one of these dives
That's full,
produces great
full zoom.
screenshots like this, and from all previous expeditions, including this one, Deep Connections 2019, you can view galleries of photos from the expedition, as well as highlight videos and long-form videos put together by our great video team at the Global Foundation for Ocean Exploration at
Looks like they're just about done with the rinse
oceanexplorer.noaa.gov.
And you too can be the proud owner of awesome screensavers by downloading some
Winch
of these
nav
images.
deck.
Everything
Nav deck.
on that we
Yeah,
view
sorry about
is
that. Took a little
publicly
longer than
available
expected, but we're
on
now
our
ready
website.
for winch to spin.
For monthly low payments.
Copy that. Thank
Okay,
you, Deck.
never mind.
All right, coming in.
$0.
Bridge, RV nav.
Copy, hauling in. Looks good.
Because we do these explorations for you and for
Thank you.
scientists all over the globe. This science that we do is
Hi, yes, look, put a mover question,
exploration,
please.
Looks good
building
out
the
here.
foundation
Winch at will.
for future science.
Thank you.
So much of our deep sea has yet to be explored,
want to
and
confirm that we have
the
total winch
Okeanos
control before we
is
call the move in, but it should be good.
America's ship for
sorry
ocean
about that, Bridge.
exploration. So
yes, please.
we are the only NOAA
range
vessel
20 meters,
100%
bearing
dedicated to
180,
ocean
speed
exploration,
2 knots.
and our mission is to go to
Our
places
video's
that
clear,
have never
pilot?
been discovered before
Roger
and
that. Thank you.
explored
Yeah,
before.
there they are.
So we're always finding something new, seeing new
Dan.
behaviors, and
Sorry
documenting
about that.
things for the first time. And that's the most exciting thing about doing these dives is that every day you never really know what to expect because every site has never been visited
settle
before.
back to you
And
and
so
head uphill.
I'm always excited to see for the first time ever what the ocean floor has to offer. And you get to explore with us too, and you also get to be your own explorer,
back to you.
exploring along with us, because everything we're doing right now is live, and everything we see is new.
Yeah, absolutely. It's thrilling in its own sort of way, as compared to scientific research, in which you're going to an area or running an experiment that you have a question that you want to solve. And there's definitely a thrill in that as well, and seeing whether you were on the right track or whether you need to revise your
180,
hypothesis
right? Yeah.
after you do some
It was,
experimentation. But just the idea here, where so
Thanks
many
for
of these
clarifying
sites,
on
when
that,
we're going
Winch.
there, we just really
Yeah,
don't
so
have
when
much
they
of an idea
asked
at all
what they'll
of
do
what we're
before
gonna
they give
see.
us
It's
total
cool.
winch control
That's
is
why a lot of
asked
times
to test
as
paying
the
out and
dives
to test
go on,
hauling
or
in
even from
to make sure
expedition
that
to
the
expedition,
rinsers
Just give it a bump.
we're really revising
and then
our
once
thoughts
they visually
on
inspect
what we
it and
think
everything's
is happening
good
in
to go,
these
then they'll give
areas
us full winch control
biologically,
back.
Gotcha.
geologically, in terms of physical oceanography, because all of this is just as new to us as it is to you.
Could just be one big slope of this stuff, I guess.
Yeah. Right, tilt up a bit, video. Well, there's a little ledge there.
Ledge there, yeah.
Are you in a safe configuration with this heading if I kind of push uphill a little bit? Yeah. Okay.
Yeah, it looks
I
like
run
there's a
our tether,
lot of littering on the seafloor of old broken off pieces of Ophelia, and we're seeing more and more as we're getting close to this ledge. So that could be really good sign for some excitement to come.
Yeah. So maybe our next move we can bump up.
And this ledge looks very white, so it's probably a slab of carbonate. I'm also seeing a lot of shells, gastropod shells, that have been transported from further away.
I think that's you. I wonder what that is.
Check this out, Jeff.
Take a look.
I think we're seeing that white carbonate wall.
Sure looks like it to me. And it looks like the source for a lot of those clasts and boulders that we were seeing below.
Maybe not.
And we're also getting right into that middle of the steepest slope area on our map.
To like that piece of the rock.
Yeah.
You know,
So it looks like a good sign.
Right.
And with all of this dead coral rubble around here, it also suggests that there's probably some really cool deep sea coral communities coming up soon as we head up the slope. So
Watchley, this is Pilot.
Go ahead, Pilot.
Right now we're still kind of moving laterally along roughly the same depth. Would you like us to start moving uphill again?
Yeah, I'm thinking it might be good to check out something a little bit higher, maybe not go too far up, but this ledge is really interesting, so we might want to check this out. It might lead to some good stuff.
Our next move will go uphill then.
Yeah, our dive plan today was to go straight up the slope, but we did land a little bit further away from our planned landing site, and we thought, okay, well, we'll just head up slope right here and see what there is to see. And as we got to this location where the highest slopes were indicated on our map, we started to see a lot of really cool boulders and rubble and outcrops of rock. So
Roger,
we
Bridge,
decided to
thank you.
move laterally
I'll stop
across the
pilots.
slope to see if there was something even more interesting. And I think we found some interesting geology. And usually with interesting geology, we have some really cool biology.
So you guys thinking 270 for next?
Yeah, our exploration model is fairly freeform, where we get
up on the slope.
community input for our dive plans months before the expedition. But there is latitude, especially for some of the sites that are identified based on these predictive models. The predictive models identify fairly broad zones of habitat suitability, so we do have some latitude within that to modify the dive tracks, both before the dive as well as during the dive, if we end up seeing something that catches our eye, like this crazy sea spider to the right.
Yeah, that looks like a much larger individual than what we saw just a few moments ago.
Go in video.
Check out the length of those legs.
Yeah, we have our lasers on right now, which are 10 centimeters apart. And
Gonna
so it
hold
looks
here
like
since
this
he's moving?
thing is leg to leg, well over 20 centimeters.
Going tighter if you got it.
So fear not if you are an arachnophobic, because these are not
Holding.
an arachnid. Totally just should not have any sort of concerns about them.
There's our squid fins.
Yes, and the squids are entirely obscuring what we were looking at.
Dang it.
Silly squids. Sea spiders are for scientists.
Deploying uppers.
Copy. Okay, we can let him go.
Okay, coming out.
You know, the more we're looking at this, the more I'm thinking maybe this is an old Lophelia mound. There's just so much of it, so much
move it up slope a little bit.
material here that's been sort of buried.
Copy.
But maybe not.
Inverting the thrusters.
I'm just speculating. It's fun to speculate sometimes.
It is. That's hypothesis making.
And if you speculate about sponges, you're speculating.
Ooh. And for those who may not know, that is a reference to the skeletal structure of sponges, which are called spicules. And so yes, that was a entirely face-palming pun that I highly appreciate and respect from your biology lead, Megan Putz.
I couldn't help myself. It's the last dive.
Okay.
I have to get all the puns I can in.
Holding here.
Now on this piece of rock, we're seeing some of these really interesting sponges that we made a collection of previously
Can I come
in our
before the squid storm comes?
expedition. These
Thank
really
you.
small, sort of spongy-looking sponges, but when we got them in the lab, they're actually pretty hard. So I'm not sure if they're a type of demisponge, but I don't think they're a glass sponge. And then a really beautiful single polyp scleractinian, a type of cup coral.
Okay, you can come out.
Okay, coming out.
Yeah, and these encrusting
Pigeons of
organisms
the
really
sea.
do seem to like this hard rock, which makes me wonder whether what it is that they prefer is the
I'm gonna settle back to
higher
you a little bit.
porosity or void space. And I mean porosity not in the sediment sense, but just in the fact that the carbonates are typically a lot more pitted and have a lot more holes in them, whether they like that or whether they like that the carbonate rock is so much more brittle and harder than that really over-consolidated mud and clay that we were seeing below. It makes me wish I could communicate with them.
Search starboard.
Copy that. Oop. Thought maybe I saw a fan. Maybe not. Let me check. Oh, never mind. Jelly. Swimming into the ground.
Yeah, it looks like the same kind.
Yeah, this is the jelly that I was telling you about, Jeff, when you were at lunch. It has these red channels in it, and it keeps insisting on swimming downwards.
There are some jellies that swim upside down. I think they're called upside down jellyfish.
That is right.
But I'm guessing that's not one of them.
It could be.
It's
I
just
mean,
waiting for us to zoom in on something.
upside down jellyfish are also known for sitting on
Stairs at the
the
bottom.
ground upside down and blurping downwards. Blurping is my term for what
240.
jellyfish do when they exercise their swimming bell.
We've really extensively used
be like
blurp
20
as a
meters,
verb
maybe?
on this
That's what I was
expedition.
thinking, yeah.
I think at this point it's
Roger
probably in the scientific lexicon and has probably been featured in several peer-reviewed publications by this point.
Bridge RV nav.
Coming up a little bit.
RV nav bridge.
Hi, another move request, please. Range 20 meters, bearing 240, speed .2 knots.
Well, that's how new words are formed anyway. Someone says it, other people continue it.
20 meters.
I'm just trying to
bearing
start some trends.
240 at speed .2.
Apparently, I've been quoted on
Roger that.
Twitter or
Thank you, Bridge.
something. Whoa. Found it. Some rocks.
Yeah, it definitely looks that way. And this is a steeper example of the carbonate stuff that we were seeing before, and it should be patently obvious the source of where this rubble field that we're looking at is located. And I'm really curious to see how the depth
You wanna zoom
range
in?
that we're
Okay.
observed these really steep
Yeah, it looks
carbonate
like there's
cone walls
some,
match up between
like newer
Beach
Blophilia,
Canyon
maybe?
and this intercanyon between Block and Alvin Canyon to know whether we're looking at the same geologic sequence or not. I suspect that we are.
Am I clear to zoom in, Pilot?
ahead.
Okay,
We
coming in.
might be seeing just the base of it today as opposed to beach where we saw a whole ton of it.
Holding here.
So we've zoomed here on a cup coral, and in the background behind it is some dead brambling coral, the Lophelia pertusa. So we keep seeing this, so I'm hoping we're going to see some live Lophelia as well. And we've been seeing a lot more of these brambles than we've seen on any other dive
so we can go find
thus
some live
far
stuff.
during this expedition.
Looks like a sea
so
star
I'm
on
really
your starboard.
hoping we see a whole bunch of it. I'm waiting for some extreme excitement here. The last dive, we have to finish strong, you know?
This is the Unimount auto heading.
Just going to come back to you for a little bit until Sirius gets moving.
Okay.
So we've got to move up. What's that? Quick zoom.
We're seeing a fastly swimming fish swimming away. Looks like a hake that's swimming away, possibly Antimora. So that is a fish in the family Moridae.
Coming out.
We saw a bunch of them at the beginning of our dive. The beginning of our dive was very fishy.
Sirius's feed is really interesting right now. You can see kind of amphitheater-like steps in this carbonate rock that we definitely didn't see in Beach Canyon, which is just one entirely vertical sheer cliff wall. So that might indicate different lithology, but it might also just indicate different post-depositional processes that carved the walls in this shape. Either way, it's notable how different it is.
Pilot, right where that squib swam by near the bottom of the screen, there was something that is pink in color, pink or red.
Yeah, we'll take a quick look. Go ahead, video.
Okay. I was just curious as to what this was.
Gonna hold here.
Oh, it's just a rock. Thank you.
Okay, coming out.
I thought it might be like a
Just
sea
a
pen. I was
rock.
like making everything into sea pens.
Just a rock. Oh.
It's not just a rock. It's just not what I wanted to look at.
It's a red rock, which is interesting. We're in a zone of a lot of white rocks. Why is it red?
I don't know. I'm not a geologist.
Camouflage.
Wish we would have gotten a better zoom.
Just the way that it is.
I don't know. It's only a singular red rock. If there's multiple, I think it'd be more interesting.
There goes some of those. Those are flame scallops, right?
Looks like
Yes, that's correct.
Step up ahead of us.
We've seen a lot of
that moves
shells
done,
of
right?
those before.
Yeah, it should be.
We haven't seen
Looks
any
like they're
living.
settling out right now.
Okay.
Come out.
Yeah, I think we might want to go laterally.
RV nav bridge move complete.
Hey, Pod,
Roger, Bridge, thank you.
moving
watch
laterally
lead.
instead of up and down.
Yeah, watch lead. We just finished a 20-meter move up slope. We're seeing what could be a pretty steep feature up ahead. We're just going to push out and take a look, and then we'll come back.
Okay.
Awesome, thanks.
Yeah, that's perfect. Yeah, I don't want to miss out on anything. But our pilots have sonars.
Not sure if that's the end of it or not.
Both
but
on the Deep Discover and on the Sirius, so they can get a really good
Yeah, it looks pretty
picture
sharp.
of what's going on up above,
I'm going to come up.
which is really important to the safety of our vehicles. We want to stay away from any overhangs and areas that could
Looks like
pose a
that's a
danger
plateau,
to our
huh?
vehicles.
Quite tall.
And this view is really cool.
Yeah, it is. This
Coming
is a lot more similar to the beach lithology that we were seeing the other day of just these really sheer carbonate walls. I would like
Your
to
altitude's
get
12.
a zoom of the interface between a couple
think we
of
need to
these
back up, or-
strata,
I think we're okay.
but
Well, and the interesting thing about beach was when we were coming up this wall, we were seeing sparse
maybe back at 5.
corals and sponges spotted around the wall, but it was at the last 5 minutes of the dive where we came upon a dense community full of a bunch of things that we didn't have enough time to characterize.
Are you going to settle
So
out here, you
my
think?
hope is perhaps when we get to about that same depth or near the top of this cliff face, we might be seeing the same types of communities that we didn't have the chance to characterize the other day, which would be really great. So we might want to use our time most efficiently to make sure we make it up there.
So this might actually be perfect, huh?
But I also don't want to miss out anything that we might have overlooked by moving too fast. It's a real delicate balance, trying to make sure we cover a lot of ground, but also make sure that we fully characterize this area.
What are our depth contours there? So D2's at like 11:30. So that's right what we expect.
11:40.
Yep. Seems like it should be pretty safe, huh?
We got a few more sonar updates to see if it gets closer or further.
Okay.
21 meters from it.
So in our chat room, we have some words of wisdom from Ken Sulak. He wants us to, critically think about what we've been seeing throughout our dives, the different strata that we've seen, the different rocks and rock faces, and areas that we're surveying. And not all hard substrate in the deep sea is created alike. Not all places are going to be highly suitable for deep sea corals and sponge habitat.
Watch this is Pilot.
Go ahead, Pilot.
Just letting you know, we were just wanting to make sure we were in a safe configuration. I think we're safe to proceed. This is at least a 10 or 15 meter tall vertical wall. We didn't see the top of it yet, so but we'll come back down to the bottom, take a look, and slowly move up.
Sounds like a good plan, Pilot.
I think we found that interesting thing that you wanted to.
Yes, interesting things.
I'll stay here unless you need me to come down, but...
Okay, seems good. Okay. Do you want to maybe let the bridge know that we're on a very steep feature?
Copy that.
And we'll need to just ensure every time that the center rotation is about the A-frame.
Roger that. Bridge RV now.
RV Bridge.
Hi, Bridge. Pilots just wanted to let you know that they're on a pretty steep cliff down below right now.
There's a fluffy
and so as we put our moves in, they just want to confirm center rotation.
I think it's a biology.
Okay, let's look at the biology.
Roger that. Center rotation.
I want to see the biology.
Roger that. Thank you, Bridge.
All right, watch lead. I'm sorry. Was there anything particular you'd like to see?
Yeah, let's see this toppled coral that's right here center screen. That's a pretty large coral colony, and it's very recently toppled over. It still looks like it's in pretty good shape. So something has happened here recently. It also looks like there's some fishing lines, so...
Oh, yeah. That eluded me. It's very transparent.
So as I was saying earlier, so lessons from today's dive and some of the preceding dives during this expedition, not all hard substrates are functionally equivalent. So not all hard substrates in the deep sea are going to host high-density communities. Some are more preferential than others for these animals.
Coming in. Hold here for partial.
And that could be due to the differences in the currents overhead and how much food is getting to the organisms in these areas and the productivity in the surface ocean.
Am I clear to come in?
Yeah.
Okay, coming in.
So this toppled coral is Acanthogorgia. So this was the coral that we were taking a collection of before, trying to get some of these aplacophorans that were living in the branches. So I'm seeing some brittle stars in the branches of this toppled coral. And there might be an aplacophoran. I see this small worm-like animal wrapped in the branches on the right side.
Not get any closer, so I'm comfortable here.
No, it's not a snake star. So the aplacophorans, they look kind of like that, but
Can I zoom
they're
in
short,
on the base?
and they usually just
We don't
wrap
normally see
around the
that.
branch.
Oh yeah, we're ripped up.
And it looks like the holdfast of this coral gave way. It was growing on that carbonate wall, and it just got too big, and it fell off. That it looks like it still has a piece of the wall attached to its holdfast, but there was instability in that wall there.
Okay, when you're clear, video.
Okay, I'm clear.
So I'm just trying to see if we might see any of those aplacophorans, because Kevin Kokot from the University of Alabama and his team are working on the genetics and taxonomy of that group, and they're not very well known. So getting samples of something like that could be very important to their work.
I'm sorry, Watchley, were you interested in a sample of this coral?
I was looking for small worms that live in the branches, and if they were there, we would want some, but I don't know if we saw any.
Okay. Pilot, could you pan up and to the right?
Okay. Stand by, Watchley. We're just we're getting our hydraulic lighting arms stowed so that we don't hit them against the wall.
No worries.
And then I'll get back with you. But what are you looking for exactly? This pattern here?
Yeah. Do you see that really smooth part in between, like the really holy stuff above and below? I want to look at that.
Sure.
Lower.
Thank you.
Thanks. That is a very different looking, isn't it?
Yeah, that layer.
Yeah, this is interesting what we're looking at right here. This is all, I believe, carbonate that we're looking at, but the actual material that is composed of this, you can see a real abrupt change between this layer between and above and below.
Okay.
And so this could be a post-depositional chemical change
Got a
that's
hold
causing
here?
the above and below to be really aggressively
little
chemically
overhang
weathered,
possibly, huh?
and this one not to, or this could just be an entirely different material altogether. And this stuff is really important to focus on stratigraphically, because it's these abrupt changes in lithology or the type of rocks that are really frequently failure planes of submarine landslides or instabilities. So, for example, if this one has a lot lower mechanical strength than stuff above and below it, this would be a nucleation point for a landslide to start along.
Could just be ink.
As for the actual material that's composed of,
let's go ahead and get the
I
uppers
don't
in
know.
as well. I might try to move to the side and look down the length of this layer.
You okay for me to stow them now?
Go ahead. I'll back up a little bit.
Yeah.
Looks like you might have a port side invert pilot.
Copy. Gonna invert the thrusters here.
Yeah. String arms are stowed.
Thank you.
I did turn to port a little bit, so now I'm kind of straddling that,
Gotcha.
if you will. Kind of looking along it.
I'll feel bad if we don't take another look. Can we look back at that coral real fast?
Sure.
All right. It was a really big colony, and it could be a really important sample, and it's already fallen, so it's already going to die.
There it is.
so I wouldn't feel bad about taking it. Because if it has those A placophorans, that would be a really
I'm going to
important
range my
sample.
sonar down to 10 meters, please.
Copy that.
So no one's going to be back here again. That's the hard thing.
So you'd like this whole colony?
Do you think we can fit the whole thing in the box?
I think so, yeah.
Okay. Yeah, let's just do it.
Hydraulics are up.
Okay.
It also has a number of associates with it, which are also very important.
Port-side sample boxes
And
are
I
full.
believe all of these associates were on it when it toppled, because this is a very freshly toppled coral.
See if I can set up so we can just get the drawer out as is.
Starboard side ready to go.
Okay. Might have to starboard wing inboard a bit, but I'll try to set up like on top of this rock here.
Okay. Sounds great.
Said hydraulics are up.
That's correct.
Okay, thanks. Okay, we'll get Mini Zeus up here. And Chris, if you can help us keep a close eye on the sonar and the Sirius camera, make sure we don't start drifting toward the wall. Watch lead, are we expecting this to be pretty brittle or flexible?
It's going to be pretty flexible.
Okay, good.
Looks like our little squid friends are joining us for this sample. We have an audience.
Thinking. Yeah. Thinking if I did. Were to grab it like. It's an awfully big colony. What do you think, Levi, for grabbing it?
I'd do like go for the base, have some draws between the two main branches and some draws on the base side and just cradle, maybe.
Cradle it, try not to break it off.
Yeah.
The only thing is that I have to go starboard box, so I'm trying to think about stowing it in the box, yeah.
Where you just go right on the base.
Yeah, let's try that.
How many rotate you down? On Zeus.
Sure. I think I want to grab it this way.
Alright, I'm ready in the drawer.
Okay. It's actually wider than I thought it was. I think we can do it. Is that all the way up?
I might be able to wing in and get you some more, but if you need it. That's it.
It's a pretty big colony. Thanks.
Yeah, you would pick a big one if you like spin it in.
I'll do that, and then I'll just kind of push it in.
Might be able to push in the outer box, yeah.
That's true. Straddling both, huh?
Yeah, might take both. So big. Go in and cut it.
Yeah, I might just do that.
And let it fall.
Yeah.
Let it fall on the other side.
Yeah, that's a good idea.
Cut it. Let it fall on the other side. Grab it out of that one. Put it back out of that. Just an idea.
Yeah.
Down a little further. Right. Oh, yeah, it's definitely sobered. Nice. Yeah.
Then I can, I could grab that one.
Yeah, grab it and put it in the other one.
Yeah.
Yeah. If it falls in, it falls in, but...
Right.
Really picked a big one.
I know.
Nice. Getting it. Yeah, and just push it down and you're good. Yep.
Okay. Sorry, watch leads. That ended up being much larger than I had anticipated. We got the full colony in the starboard outer box. We ended up having to cut it in half, but we got it all in there.
Nice job, Dan.
So Scott Francis' doctoral student James is actually working on the evolution of the family Acanthogorgia and the genus Acanthogorgia. So these samples are going to be really important to him with his work, and it's also going to be really great to catalog all the associates that we saw in the branches of that Acanthogorgia, and it was a really good sample because this had just recently fallen off the wall and would have eventually died. So now we actually are making good use of its life by bringing it back with us as a sample. So I'm pretty happy that we were able to get the whole colony in our bio box, and hopefully there will be organisms aplenty for all of the people out there that are studying associates, studying Acanthogorgia, and studying those aplacophorans. I'm pretty certain that we have at least one of those in that sample.
Okay. 240, so that's 240 minus 90. 160 something, right?
So watch lead, we're just discussing our next move. Did you want to move laterally along the base of this, or did you want to try to go up the face of this wall here?
Pilot, I think I'd like to go up.
Okay.
Going up.
You can do that.
I've got some FOMO from Beach Canyon, so I want to make sure we make it to the top.
Okay.
All right. I'm lined up at 240. Before we move, we'll come out. But yeah.
I can't recall.
Oh, I have your rotate.
Yeah, I was like,
yeah,
I was confused.
This really
That's fine.
Back to you.
Fine layer interbedded with the porous.
You can turn that monitor back to whatever you'd like.
Carbonate that we were seeing
Roger
before.
that.
That's an interesting observation. It looks like there's one band every 5 meters thereabouts.
What a nice sample. Look at that. Got the base and everything on the little guy.
These views of the rock ledge are just fantastic.
Take a peek to starboard.
A lot of the flame scallops I don't believe we saw any of when we were looking at Beach Canyon.
No, we didn't see them. Yeah, you'd think this looks very similar, and it's in a relatively close proximity, canyon-wise. But yeah, we didn't see any flame scallops during that dive. But we do notice that there is some sparseness of where we see these animals.
Yep.
So there's a very patchy distribution of animals within the deep sea. Sometimes you see a high density of something that you won't see for a long time, and then you'll come upon another patch.
And
that's where my FOMO comes in, because I'm like, well, maybe we just went a little further over one more ledge, and then we would have seen the thing that we came to see, you know? Right now we're looking at a synaptobranchid eel, a cutthroat eel. This one is Synaphobranchus gracilis. It's been pretty common throughout a number of our dives.
Okay, you can come out when you're ready.
Coming out.
There's a squid following us.
Yeah, one of the best moments for these eels was when there was a crab that had found a piece of squid and was having a really tasty snack, and 2 cutthroat eels came in and they started trying to steal that squid away from the crab. You can check out that video online. It is on our website, oceanexplorer.noaa.gov. I think I'm spotting some Lophelia on this wall.
Right there in front
Right
of us?
there in front of us. Yeah, right next
Take
to
a
those
look.
flame scallops. Let's take a brief look.
Okay, you can go in video.
Okay, coming in. Gonna hold here.
And it is. It's a nice living colony of Lophelia. So we know it's here. We know it's alive. And
Coming
if
in
this is
more?
any indication, there should be more somewhere. So this coral is a hard coral, meaning that its skeleton is made out of calcium carbonate. It forms these large brambling aggregations on a lot of locations. And these corals are related to the hard corals that you are most commonly familiar with in shallow water coral reefs that build up coral
Coming out.
reefs. Now, they don't make a real reef, but in some locations they do form large mounds that can provide a lot of habitat for associated
Try the headlights and
organisms.
see if it kind of evens up the lighting.
3, 2, 1.
I'm going to come up a bit. This is awesome. Definitely was not expecting this today.
I know. Huge wall out of nowhere.
Yeah.
Haven't even seen the top of it yet.
If you're just joining us, this is Megan Putz, your biology lead from the University of Hawaii. We are currently doing our 12th dive of our Deep Connections expedition on a box canyon that is located between Alvin and Block Canyon within the Frank R. Lautenberg Coral Protection Area.
Great view from Sirius, watch lead.
And this is Jeff O'Belts, your geology lead on this expedition. This is our 12th and final dive of Deep Connections 2019.
All right, we can turn off the headlights.
Yeah,
it's probably better without them. Should help you, Caitlin.
What diving zone? Got mini Zeus up, be advised.
Copy.
So you might be wondering a little bit about this Frank R. Lautenberg Deep Sea Coral Protection Area. This area is protected from bottom fishing, so commercial fishermen are prohibited from using most types of bottom tending fishing gear such as trawls, dredges, bottom long lines, and traps. But this rule doesn't apply to recreational fishing. This is only commercial activity within this protected area. That means that, you can come out to these locations, but, also keep in mind we are seeing these really vertical cliff faces. So bottom fishing in this area wouldn't be the best idea because you would most likely lose your gear. And so a lot of fishermen do target areas near here because these are really productive areas. Canyons in the deep sea are very productive, and we see a lot of biodiversity in these areas, which means that, you know, it's a high likelihood of
Do you
finding
want to mark this on my pack,
these
maybe?
really
Put like a vertical
sought after
wall.
commercial fisheries, like the fishery for the squids that we've been seeing, those short fin squids, or we saw a few
You have
flat
any idea on
fishes,
the height of it?
flounder, which could also
It's
be
at
good for
least
fishing,
40 meters.
and crabs and things like that.
We haven't reached the top of it yet, though.
I was just about to say as well that I haven't seen any fractures along this wall just yet. I
And
think
then
we're
here's one.
looking at one right now, yeah. So it looks like this is probably a normal fault cutting down into this material. And you can kind of get a range as well here of the different lithology within this entire carbonate sequence. So it's all carbonate, but due to a combination of, I'm not sure whether it's the organisms whose shells were used to construct this material, or if it was a post-deposition chemical change, you get a really different texture of the carbonate rocks. And you can see 3 different types of them.
I'm sure if you look on the sonar,
below
still returning.
Yeah.
The area that we're looking right now.
I think it just steps back.
And I saw something geologically interesting. Just Pilot, can you just pan to the left a little bit?
Sure.
It looks like there's some slippage where this layer is broken.
That is a fantastic observation, Megan. This is a fantastic example of a fault, and you can see it's a fault because there is vertical displacement of this thin layer that we've been looking at before. And this is one of the ways.
I'm just gonna leave them off for a few seconds for the video editor, and then I'll turn them back on.
Yeah, so this is one of the ways that geologists reconstruct motion of the Earth is by looking at correlatable layers, and then they can see when there's a fault like this how much it's been displaced.
So Copilot, does that
so this
return
angle,
fade out
what
once
we
we
can see
get
right now
we
is
see the
that
top?
to the right of your screen,
Yeah,
that's
so
called the
it's a big
down
shadow
thrown
area.
side of this fault,
Either
and
it's like a
the
wall,
left side
it's
is the
open
hanging
space
wall,
behind it,
and we can see
or
that
that's
there's
all I
about
can
a
see and I can't see
strata
over it, so
that
it's
really,
possible that,
you know,
like,
that thin
the
part
wall
in the middle
It
has
just
been displaced
it's
by
back
about
further.
its
I think it
thickness
continues up.
down the wall. And
straight up.
this varying
I think
lithology
the
offers a
darkness of
great
shell is the
opportunity
lack of light.
at looking at that displacement and providing a measure for it. So if we knew how long that
like once I start seeing blue back
that
here,
fault
then
has
I
been
know I can
moving
see
along,
over
we'd
it.
have a displacement
But right
rate
now I can't
at
see
how
over it.
fast this
I'm
down
above
thrown
things.
block has been moving
Roger that.
down the
Thank you.
wall.
Yeah.
So that was a great catch, Megan.
Getting kind of close on the port side.
Can I zoom in on the wall? That
Sure, I'll get
I'm
lasers
not
for
sure.
a few more seconds.
Okay.
Let's watch the dust, and then you can zoom
It
in.
varies on area to area what causes faults like this to move. In this case, this is probably a normal fault, which is also called a growth fault, which just means that the fracturing and the downward movement is related to just one fracture plane forming for some reason,
off, or
and
would you
then it
like to
kind of
zoom?
slumping along its own weight preferentially. So growth faults don't
Vertical.
need
The
seismicity or
structure,
earthquakes, and they don't need
like,
active
the-
tectonics or continental plates
like,
moving
looks
past each other,
like
pushing
it's
into
melting,
each other to
I
happen.
guess,
So
is-
it could
Can you
very
direct
well
me
be
to which
active.
part of the screen?
The stuff,
but
like,
I would
in front of us.
suspect It's
Okay,
not.
I can zoom in.
We would know from looking at the bathymetry above if it was really active because the throw
Just
would be
direct me.
evident. The displacement at the surface would be happening at a faster rate than the
This is
ambient
interesting here,
sedimentation, which is
with
really
the holes.
slow.
Yeah.
So maybe this would be an interesting area to look at the
we get
multi-beam
a few
or our
still
seafloor
images?
mapping data that we're collecting to see whether there is superficial expression
Trying to get another
at
view
the
down
top
the
of this
cliff
canyon wall of
face.
this fault that we're seeing at depth.
Copy.
And could this movement be an indication of
Okay.
seafloor instability?
When you're ready.
Okay.
Yeah, definitely, absolutely. And we got a really
just
great
to hammer
example
down.
of that on the Bear Seamount when we dove
Totally.
that, in which across the entire width of it,
Angled
you
the-
could see
housing.
what we
Probably
interpreted
could.
to be normal or growth faulting happened, and that can be indicative of future activity.
if this works.
Or a lot of times, what that indicates is that upslope from the area where you see the faulting or the displacement, that area is going to be unstable at some point in the future.
Yeah.
Oh, that was great. That was some great normal faulting. I haven't We haven't seen that so far.
You gotta go above and beyond for this final dive.
Yeah, we see that a lot in seismic data or sub-bottom profiling seismic reflection data in the marine environment, but it's really uncommon for us to be able to look at submarine outcrops and see fractures and faults like that.
Yeah, okay. Happy with that.
I also find this layer really interesting, that this really thin layer is very, it seems stronger than the layer below and above. Ooh, and there's some purple coral on this.
Take a look at that.
So, yeah,
it's really beautiful, huh? I like the smooth rock face.
This layer is really, really smooth. It's not, doesn't have as many pits and holes in it, and it's jutting out more from the wall.
Yeah, and again, I'm not sure whether that is a result of different carbonate shells or tests that were that composed it, or whether it was different chemical alteration in that interval that formed that rock.
Hold here.
I got to talk to some carbonate experts who can weigh in on that and get the answer for us.
And here we've zoomed in on this purple coral that's
coming
been growing
into purple
over
coral.
this really interesting layer in the rock. This purple coral is a stoloniferous coral, likely clavularia. And so this coral doesn't create its own skeleton, but grows over the rock, using the rock sort of as its skeleton. So it's an encrusting style coral. And it is an octocoral, you can tell, because it has 8 tentacles, and those tentacles are pinnate, meaning they look sort of feather-like. And that's characteristic of all octocorals.
Do you think the headlights
I also really like the purple corals because they are beautiful.
3, 2, 1.
And then next to this purple coral, we're seeing some flame scallops, some cup corals, and a few small sponges.
Ooh, that's nice.
You know what's also sort of interesting about this rock? It looks a little bit like rhinoceros skin.
Yeah.
You know? It looks crisscrossed and wrinkly.
Yeah, I can see that, and there doesn't appear to be any sort of orientation for those marks. I'm gonna assume that's probably not something to do with the rock itself. Do you think that could be something like something pecking at that or scraping along it?
I can't think of anything that would be making gouges like that in such a crisscrossing pattern.
Let's take a look at what they're talking about here.
Okay.
Zoom in.
Zooming into rhino skin.
Yeah.
Pilot, can we get some sort of hardness test of this that we're looking at right now?
Yeah, I think so.
Yeah, so we've been looking at this
coming out.
this layer, which is about 20 or 30 centimeters in thickness, and we notice what looks to be some differences in terms of its strength as well as its texture. And we just want to get a relative idea of how hard this might be, whether this is a really crumbly or friable material, which is what I strongly suspect above and below it, or whether this might be a stronger rock-like formation.
Yes, we are.
I think I could put in my port toe. Just give us.
So we're going to do the same scratch test that we did before, with just trying to get a toe of the ROV on it and kind of scraping down to see whether it just immediately crumbles away or whether it's more harder and can support the weight.
Okay.
My guess is it's harder than the other substrate around it. It just, because of the way it's jutting out,
Got that
it doesn't seem to have dissolved away.
Yeah, we'll keep them on.
All right.
Also spotted what looks like a bamboo coral growing perhaps from the shell of one of these flame scallops.
Okay, can you put the Sirius view on your monitor now?
Roger that.
it. Okay, that seems stable.
Copy.
Okay. Hydraulics are up?
Yeah.
Okay.
You want me to bring minis this time?
Yeah, sure.
I got it. Kind of looking at the ceiling right now. Boom.
Thanks.
Assuming that's the...
Yeah.
Or the one kind of in front of the Zeus. Come out.
So we're going to pull our claw out here. And maybe you can start the scratch in the overlying stuff and then go down so we have a comparison point.
In the top layer, you mean? Up here?
Yeah, please. Yeah, so we're going to do a scratch test starting on the top layer and then moving down into the different one. So this won't be a scratch test where you're doing it against reference materials, so you actually have benchmark things to compare it to. But we will get a relative hardness difference, maybe on how crumbly this one is compared to the lower layer.
So it's pretty soft.
So this is our soft crumbly stuff. So this might just be a limestone or chalky like stuff. You can see Deep Discover's manipulator arm scar in there.
Yeah, that was very soft.
Yeah.
Very lightly touched it there.
Yeah, do you mind moving down to the lower one? And the great thing about Deep Discover's manipulator arm is this has force feedback for the pilot, so they can actually feel through how much pressure has to be applied, how hard a material is, even if we're not getting like a sample topside, which is important information for them to be able to recover samples, and for this case with our impromptu scratch test that we're conducting here.
Actually, maybe I'll go for this outer one. Okay, video, you can zoom in as far as you can to frame it.
Okay.
Actually, I guess maybe I will just do this front face so we can see it better. Okay, you can zoom in.
Okay, zooming in.
Okay, hold that. Also, well, just scratch the surface. I'm not sure how much I dug in there, though.
That's moving the vehicle.
Yep. So that's quite a bit of force. It actually seems harder than the top layer, to be honest.
Woo! I was right.
Pilot, could you zoom in on, where's the bamboo whip? I didn't see that. Sorry,
So, Pilot, right below this ledge, there's a bamboo coral coming off the shell of one of these flame scallops.
Sure. Do you want to look at that first
Yes.
or finish our scratch test?
I think as long as you're confident that the top layer was a lot more crumbly than the lower layer, that's all we were curious in.
It seemed that way to me. I could try scratching in a couple other places just to verify, but...
yeah, if you wouldn't mind grabbing, like, one more,
okay.
that would be awesome.
Let's come out full wide video.
Okay.
Yeah, so our pilot tells us that at first blush, it appears that the upper layer was a lot more crumbly than the lower layer.
Let's put...
we're going to
back up and reset here. Don't want to hit the craft on the wall. All right.
So fish to your left. See that minute can? What is that?
What is that?
I would look left, check out the fish.
Very large fish coming into view.
It's like a
There you go. Watchley, do you see this?
Yeah.
Just above you.
Can you put the Sirius camera on the right monitor, please? Can you take my rotator?
Yeah. There you go.
Oh, it's an Oreo fish.
Big Oreo, though.
Say what?
It's an Oreo.
Okay.
Never, yeah, seen one that
We've
big.
seen these before.
Can I zoom on it?
Yeah.
Oreo is
Okay.
the very first part of
Sorry, the arm's in the
name.
way.
Oh, it's a family name of this fish.
That is bizarre looking.
They're kind of cute in their silly, sort of flappy sort of way.
Okay, I think we got an ID.
Okay.
Come out.
All right, so Oreosomatidae is the name of the family. Possible neositis. That's the first of that fish we've seen during this expedition.
It is. Wow, and you can get a zoomed out view to see how far above and below that
Still at the,
normal fault
right side?
propagates down. And it looks like it's an arcuate
yeah, I
fault
can keep
in
them on
that
for now.
it goes
Back
straight down and then it starts to deflect. And you'll notice that the exact area where it's deflecting is right where we've seen this rock that we know is harder. And we've seen that a lot happen, and that's dictated basically the fault's path of fracture through a material is dictated by the mechanical strength. So that change from just a direct up and down fault motion to an arcuate one is probably influenced by just the increased mechanical strength of this layer that we've been scratching at.
And before we leave, we would definitely want to take a look at that small coral that was growing off the shell of one of these flame scallops.
Sure.
Because that's a really strange thing to see, a coral growing off a bivalve shell. A really interesting association, and I know Scott's really interested in taking a nice look at that.
Sure. I'll tell you what, we'll go for that first this time, and then we'll come back to our second scratch test. Okay. All right, you can zoom in video.
Okay, coming in.
So we're trying to
Gonna
figure out
hold
if it's actually
here.
growing from this
Okay,
scallop
coming in.
shell, or,
Okay.
or it's growing out of the rock next to it. Because it would be really strange to see it on the scallop shell, and I think that's what we are seeing.
Yeah,
I
I've
think
never
I
seen
see
that
the
before.
holdfast that's
Me
on
either.
the shell.
It's interesting.
That's really neat. That's not something you see every day.
Very young. It's pretty small.
Yes, this is a very, very small colony. It'd be interesting to find out, like, how long these scallops can live and if this coral will continue to grow off this scallop shell. As it gets bigger, it might be able to send out some roots to attach to the rock wall, but it does seem a bit farther away than it should be if it was going to do that. So, really interesting life observation.
Okay, when you're clear video, we can come out. We'll do
Scott
our
France
scratch
says
test.
he
Coming out?
would not suggest this larva picked a very good place to settle.
And Scott France is a legendary real estate agent for corals everywhere, so
That's
that
full
didn't
wide.
consult with him before settling.
Okay.
Yeah, he predicts that as this coral grows, it'll likely pull this bivalve off the wall. So, oops.
Location, location, location.
Okay, let's zoom in video.
Okay, coming in. Gonna hold here.
Okay, so this was our first scratch that we did earlier, and our ROV pilot told us that it took almost no resistance to put the tip of the manipulator arm into this stuff. You can just see how crumbly it is, and it looks like it's going in a couple millimeters, and you can see the remnants of it on the manipulator arm of the ROV. Now,
it's pretty soft.
The manipulator arm is titanium, so that's why we can't do this as a true scratch test, which would be testing it against a range of materials with known hardness. But we can, from the dynamic feedback that the ROV pilot gets from the manipulator arm, get a relative hardness of that above rock versus the one below.
You know how I did my hardness test?
How's that?
All the corals seem to be growing on this part of the wall where they aren't really growing on the softer part of the wall. The harder parts seem to be more preferential substrate for these animals.
Okay, coming in.
Hold that.
Holding.
Perfect. Yeah, I can scrape just like a thin layer off the top, but it does not dig in as much.
Yeah, barely leaving a mark. Wow, cool. Okay, thank you, pilot.
Yep. And my co-pilot's noticing that when I was scraping the top softer layer, the vehicle did not move, but as I scratch this this thinner layer here, it moves the vehicle, which means it's taking a lot more force.
Yeah, that was another interesting notification that it moves the vehicle when they're digging in with the manipulator arm on the lower level. So I think we've gotten a lot of context clues as well as our relative scratch test that we conducted here to know that. And I'll be interested to take this information back to some carbonate or karst experts that I know to figure out exactly what this material is and what could cause that variation in mechanical strength as well as texture.
All right, craft hydraulics are secure.
And it's just another example of the interaction of the various subfields within oceanography.
Okay.
Can we turn the forward lights off?
Sure.
All right, coming off. 3, 2, 1.
Why don't we come up this face a bit?
Copy.
Yeah, and Scott France back on shore pointed out that once that coral gets to be a certain size, it's probably going to take that scallop off the wall with it. And so his wife pointed out that perhaps the corals prefer a mobile home property.
This little overhang here with some bivalves and a
which is
crinoid, maybe?
No, it's right in line with the awful puns.
Yeah, let's check out this layer too with all these things.
Okay, let's zoom in.
Okay, coming in. Gonna hold here.
So this is the type of star that definitely summons Chris Moth.
What is it?
That's a very precarious situation to be in.
Now this is a type of Brisingid sea star. And they're really interesting because they have these long arms that they curl up and very
zoom in, pilot?
sticky.
Okay, coming in.
2 feet. I know that Chris has been doing a lot of work with these stars in the past, and you can see how sticky those little spikes are. There's also some stoloniferous coral growing over
I'm moving in slowly, video.
one of these scallops as well, and then there's a scalpellid barnacle on the scallops. Looks like lots of things are settling on these scallops.
A squat lobster in that hole up there too.
Oh yeah, you're right.
Be nice to get a good view of those like tube feet holding on.
Sure. Should we try the forward lights again?
Yeah, it's a
Yeah.
good thing
Some
these
stills.
stars
Yeah,
have
yeah,
those
sure.
really
Yeah.
sticky suction little tube feet because that's quite a sheer drop, and obviously the fall velocity in water isn't anywhere near as high as it is in air, so it would probably be okay if it fell, but definitely looks intimidating.
Well, if it did fall, it would take 1 hot minute for this star to get back up. Oh, it's on a coral. You see, there's a coral in there.
Yeah.
Oh, it is.
You're right.
Once the scallops closed, you could actually see the coral that
Can I
this
zoom in,
star is
Pilot?
actually
Yeah.
hanging onto.
You can see the coral right there.
Well,
This
that's
was
cool.
a coral growing on the bivalve and a sea star living on the coral.
This is definitely a precarious place to be living. Just like, oh, I'm just going to make my home underneath a ledge. It kind of reminds me of those that Airbnb you can rent somewhere in South America where it's on the side of a cliff.
I have not heard about it, but it certainly appears to be a similar arrangement.
Looks
but
like the
if
coral
you could
is
just,
missing
there
its
was
polyps.
like a ladder
On the
on
left
the
there?
side of the house that you could rent.
Let me see
That's
the
what that
stick.
sea star is in right now.
Get on the coral on
Yeah, this coral that it's on is actually a
scallop
bamboo
on the
coral.
veg
I
on
can
the bottom.
see the nodes,
Yep.
those
Zoom in a
brown
little
parts,
bit,
and
see
it
if we
looks
can
like
see
it's
where the-
branching internodally, where there's a branch point before the node. So there's a node, and then it branches. So it's not branching at the node, but like just past it. And this sea star is in the family Bursingidae. Could be in the
Can we
genus
tilt down?
Novodynia, which I have seen before in great numbers on shipwrecks.
I'm going to try to frame it up for the still.
Oh, I think I got a nice view of the central disk. And you can see these sticky little tube feet coming out. And so that's how this star feeds. It's a filter feeder.
Okay,
It holds
I'm
its
gonna tilt
arm
down
in
just
this
a
sort of
little more,
curly
Caitlin.
pattern.
Okay.
And
come out if
things
you
will
want, if
come
you'd
by
like.
and will get stuck. Oh, and there's a bamboo coral
There's a bamboo
growing
coral.
just off the
Yeah, you
wall,
can zoom in
too.
there.
Okay.
Try to get it on the side of the frame. I'm almost done with the still. Yeah.
Nice.
Down just a touch more. Get one more, and then we'll be good. Yeah. That's a good one.
Do you want me to turn it off?
Sure. Okay. Okay, let's come out. I see a purple coral right below me.
Okay.
That was cool seeing all the bivalves close. It was right when I touched the wall.
Yep. A little scared.
That might not be a coral at all.
That was really cool. Talk about mobile home property.
There's a purple cup coral to the right, though.
Yeah, that was just everything piling on top of everything else.
Well, you know, when you find a really good place, everybody wants to live there.
Okay,
And
can I come in?
that ledge is
Sure.
just
Is
that
Just
place.
the pilot cam to look at the range between the mini Zeus and that little ledge. Seems like it should be clear, but yeah. Okay.
So we're now zooming in on a really neat looking anemone. So this anemone is in the family Actinernidae, is in the genus Actinernus. And you can tell that because it has these really interesting lobes that seem to fold inward toward the mouth, and it has sort of a puckered mouth right there in the middle of the disc.
Oh, a coral there. All right, let's come out.
Okay, coming out.
Levi's scanning the wall for us.
Ooh, that's a
Identifying new
cool
targets.
coral.
Yeah.
It's the underrated camera. Right
Yeah.
Tight.
There's a lot of stuff here.
All right. Let's get a Sirius back up there, and I'll move over there. That's a good find.
I think that was a Sirius cam zoom.
Oh, it was D2s.
It's
There
like
is a lot of stuff
a
on
spy
this wall.
camera.
Yeah. Recon.
Went into recon mode.
I think what we spotted is a type of black coral.
Okay, you can zoom in.
Okay, coming in. Oh, jelly going by.
So we're looking at this black coral, and we're noticing that it is
Holding here?
branching off of its main branches from the main axis. So I think this could be a Stauropathes black
Coming in.
coral. And that'll bring us back to our conversation about how when we talk about the names
very
of corals
soft.
and animals, scientific names, they're based off Latin and Greek. And this is a good example of that. The order name for this group of black corals is Antipatharia, which means against disease, because people thought that the skeletons of these corals made good medicine and could cure disease.
Can they?
Yep, you're right.
Probably not.
Oh, that's a
I don't think there's any research that supports that they have any medicinal properties. I do believe the Greeks used some coral in medicines, including pink coral. They said that there was some good evidence that pink coral, which is calcium carbonate, could cure some tummy troubles. Which makes sense because we do know, like, if you take a Tums, which is mostly calcium carbonate, it'll help settle your stomach, reducing the amount of acid and neutralizing that acid in your stomach.
Which is the same reaction that creates a lot of this pitting that we're observing along this carbonate wall. Acid-base reaction turns calcite into carbon dioxide and water, and off it goes.
There.
Forward lights on. Sure. I'm gonna come back to port.
Okay. There is a coral or something on the edge of that ledge where the percentages and stuff are. It's kind of dark, yeah.
Yeah, you can do a snap zoom.
Okay.
Oh, and here's another type of black coral. This one is like the one that we saw right at the end of our dive at Beach Canyon. This is a Teleopathes. I meant Parantipathes. Parantipathes.
Coming out.
Teleopathes is just the name of what I was just reading.
That's full wide.
And you'll notice that all these black corals end in pathes, along with the order name Antipatharia.
Okay, I'm normal to the wall.
Okay.
It's interesting to see that
Why don't we make some
pretty
vertical
big anemone
progress here?
growing on
Come up a little
the
bit.
weak area that we've identified. Now, of course, that doesn't mean that they can't.
Kinda like
it's
having
just
minis
that
who's
on
up.
average, as
point it
the
up.
organisms
Yeah, sure.
get larger,
Point it up.
they're more likely to end up as one of the cobbles
at
or
the brow.
boulders that we
Yeah.
saw
Situation.
lower down in the canyon.
Nope. Something just fell down. Not sure what it was.
Ooh, a cave.
Cave of Wonders.
What's in the cave?
There's a fish.
Surely we can fit Deep Discover in there, right?
Well, that looks cool and serious.
Deep Discover
It does
is the
look
linking
cool.
time engage.
Forward lights.
3, 2, 1.
Oh, is that a sea star on the right?
This is the very
I think
epitome
so.
of exploration right
Like the
now.
really fat one? You see that?
Yeah, I see it.
I think he's like
Might be
Oh
eating
yeah,
something.
that is a sea star.
brow.
And it's
Brow.
it does look pretty
Port
fat.
brow.
Yeah. Crab.
There's a lot of interesting stuff right here.
Crab falling to starboard. Oh, flying by.
Fly by.
It's not called Cave of Wonders for nothing.
Yeah. Is that what is that on the right
Is
side?
it called the Cave of Wonder now?
See that kind
Yeah,
Okay.
I named it.
I haven't got the memo yet.
The right side?
It'll be on my desk in a couple
I
minutes,
think that's a skate
Well,
egg.
memos take time
It's a
to move
skate egg?
backwards
I
and
think
forth.
so.
Why don't we do a quick zoom?
Okay. Yeah, I saw one earlier during the dive.
Yeah.
But I didn't point it out
First
because we were going
wonder
over it really quick.
Is shark egg case.
Or shark egg.
Cool.
Called Mermaid's Purse.
Okay. You can come out.
Okay, coming
And I
out.
would totally carry one.
Should we zoom
you know,
in
if it was a
on-
little bit bigger,
Yeah,
I need to
sea
be able
star.
to
Sea
fit
star.
at least 2 or 3 things.
Just
Might
like
be
the ones
just a
that you would
smidge
see on the
over, huh?
beach. Well, probably
Okay.
not just like. They're probably
You can help me
from a
watch
different
that.
creature, but
Try to size it out.
Okay, you can go in partial.
Okay, coming in. Okay, gonna hold here.
He's well fed.
Yes, it definitely looks. Well, so starfish like this can actually inflate their bodies with water, so they have what's called a hydrostatic skeleton, and they can inflate that hydrostatic skeleton, which is probably what this one is doing, and making it look quite fat.
are you just saying that to flatter it?
Yeah, we turn forward lights off.
It's not. It's rude to call
I'm gonna
starfish
bring
fat.
lights off video.
Okay.
I think it's pretty far away. It won't shouldn't change. Reduce the move
It's
back
just
there.
puffed
Yeah,
up
that's better.
for reasons. It also could be feeding on something.
Can I zoom in,
it
pilot?
looks like
Quite get a
it's
good
over
footing.
on
Hold on one
top
second.
of something. Seems like an awkward position, and a lot of times when these animals are feeding on, say, a coral, they'll extend their stomach out of their body and wrap it around what they're feeding
Okay, go
on,
ahead.
and
Okay,
that would
coming
be
in.
underneath this animal. So it definitely looks like it's over top of something.
You're getting like 40 to 50 meter
Yeah, I saw
altitude pings, so it's another tall wall. Yeah, watch leads, just so you know, we're getting 40 to 50 meter altitude pings on Sirius, which means that that's at least how tall this vertical wall is. You know, obviously, we haven't reached the top yet. So it's another very steep feature that we were not expecting to see here.
Awesome. Let's keep climbing.
Okay, coming out.
You guys saw some squid ink on here?
Okay.
There
What's
is a
first
worm in
finish
the back.
investigating the cave?
Let's come out.
Okay, coming out.
What else do we have in
That's
our
full
little
wide.
Cave of Wonder here?
Well,
A
there might
worm?
be an octopus in the cave, maybe sheltering with eggs.
I would
If
cer-
I was an octopus, that would be a nice place to, you know, hang out with your eggs.
Yeah, it's a great crevice.
And we just learned that the octopus that we've been seeing during this dive can shelter with their eggs for nearly 4 years before they hatch. And they never leave their eggs. So that's a really long time.
Okay, it's stable.
Okay, are we gonna
Mother of
zoom
the
into
year
the
award.
cave?
Yeah, go ahead. Maybe
Yeah,
get the
mother
forward
of
lights on
the
here.
Yeah, I'll bring them on.
half-decade award.
Looks like a fish in there.
Knew there was a fish in there. So there is a rat-tail fish in this cave, hiding from us. That's a fish in the family Macoridae, also known as grenadiers.
It's really interesting as well how far this cave goes back. And you'll notice that it is carved mostly into the harder substrate that we noticed. The roof of the cave is that softer stuff. The actual walls of the cave are the harder substrate, which shouldn't be surprising. We know that stuff has the mechanical strength and can probably
I see
bear
the
the
fish.
weight, or can obviously bear the weight of what's above it. Otherwise, this cave would have collapsed by this point.
Okay, you want to come out?
Okay, coming out.
You can turn off the headlights when you're ready.
Coming off. 3, 2, 1.
All right, anything else here, watch lead, or should we move on?
I think I'm good. Megan, you good?
Just one more thing. Right at bottom screen in the center, there's an open shell, but then there's also a pink-looking
go ahead,
thing.
video.
Just a snap zoom on
Okay,
that.
coming in.
I'm not sure what that is. And it might just be another
Portbird.
acesta. Yeah, that's what it is.
Oh.
It's just another flame
Let's go on
scallop.
video.
Okay, coming out.
I thought it might be something else.
Thanks, Levi. Yeah,
Also looks
that is
like
big
there's
cloud.
a bunch of brambly Lophelia around here. There's some live stuff on the outside of
Come
this
up a little bit.
cave wall.
A little bit of an overhang, like you said.
Yeah, and I don't know if this was relayed to the stream from our pilots, but the sonar from Sirius and Deep Discover is reflecting another 40 to 50 meters of hard pings above us. So this is almost certainly, I would bet my paycheck, the same formation that we were seeing on Beach Canyon the other day, just based on lithological similarity as well as the vertical extent of it,
It's still
and
not
depth
clear.
similarity.
I got a fault. Can you clear it? Thanks.
And
Still not clear.
that's an example of one of the really
There we
first
go.
order geological principles known as Walther's Law, which just states that formational environments tend to be similar in lateral areas close to each other. Beach Canyon isn't too far from the canyon that we are right now in geological distance, and generally speaking, depositional environments change laterally fairly slowly, so over fairly large areas, I should say. So it wouldn't be too surprising to see the same formations this fairly close together.
Wow. It's quite the wall.
Yeah.
Yeah, and this part of the wall is really encrusted in
See something
flame
with it.
scallops
Yeah,
and
a little bit.
anemones, as well as Lophelia, and just very sheer vertical cliff walls, very similar to the ones that we were seeing in Beach Canyon the other day.
We're also seeing quite a few different colonies of stoloniferous coral, which tend to grow over this rock face. So we have some yellow and some dark purple and some light
Zoom in
purple.
video.
But this center
coming in.
colony is interesting.
Holding here.
Oh, so this is Parantipathes, a black coral with a number of associated organisms. And the bottom area of this sponge, or of this coral, excuse me, is dead and covered with sediment, but the end that's out into the water column is still alive and well with a number of associates, brittle stars, and perhaps there are a few, yeah, I think I see a squat lobster. And some of these squat lobsters aren't as well described. These chirostylids that sit on these corals are still being worked on.
Oh yeah, the tiny little squat
They're
lobster
very
there.
small. It's hard to collect them.
Oh, hold there, hold there.
Should I come out, pilot? Just a little bit?
It's very squee. So I just wanted to put out there to Scott, France, and others who might be listening in, do you think that this would make a good collection? Okay, so Scott's saying we've collected several of these from the Northeast Canyon, so we'll pass on this collection.
Okay, coming out.
Okay. Right when I'm stable. That's okay. All right. Stand by, Caitlin.
Yeah,
We're
and a
gonna do
really
little shift
dense
here. Shift
Sounds
change.
good.
assemblage of those flame scallops that we've been seeing all over this wall.
I'm just going to put a toe in here.
And I think it's suffice to say that Mike White has done it again.
He's never let me down. Oh, there's a cool sponge and a bunch of Lophelia. See, that's why we had to make it all the way to the end. We're seeing better and better things. And it looks like we're getting near the top of this high slope area.
Oh, wow. Wow. No, now I'm looking at our HYPACK map, and I can see an area of higher slope at the top that's arcuate, and our track line is right along the edge of it. So we were probably seeing the hanging wall of this entire normal fault that is down-throwing this entire wall of the canyon. Wow.
It's pretty cool. We could put it on feed number 3. I think that's...
So nav pilot.
Yes.
Are the surface conditions going to move us towards or away from the wall right now if we lose DP?
So which way are we pointing? Pointed
We're
west?
currently doing a pilot
Yeah,
change.
the currents
That's why
would
we're sitting
actually,
still.
because you're pointed west right now, right?
But we're also discussing
west.
Pointed west, yes.
the geology
Yeah,
of this
so
area and
that
what
would
we're
take
seeing
you-
on our map
Yeah, right
and
into
what
it.
we're actually
So-
observing
Okay.
with our
Bailout
cameras.
is southeast.
What are the currents doing?
So it's pretty
It's
cool.
towards the west.
We
Yeah, but
did
that's
notice
surface
that
current,
fault
so-
in the
Yeah.
wall.
Oh,
That might correspond
at
to
depth now?
um,
That's
a
different.
fault that
yeah.
we can actually see
Just if the
But
ship.
yeah,
on our
it would
map.
take the ship. Yeah, right.
Yeah. At depth.
And our geologist is
well,
really thinking hard about
southwest.
this.
So really, even at depth, it's still about the same direction, even at depth.
Oh,
Yeah,
okay.
so if you go
until you get to
feed
about
number
500
3
meters
right now, video feed number 3,
then it
if you're
goes
back on shore, you'll see
northwest.
Fleeter Mouse, which is our
Okay.
Yeah.
three-dimensional
Well, if,
program that we
Andy,
use
if
to
something were to
display
happen,
the
you,
seafloor
I would
mapping
keep
data
my
that we
head and
collect.
you would just come
That
out
orange
under me and
line
just
that you see is our
zoom out
track
to the
line.
east, I
The
think.
floating
Copy
red
that.
square is
Yeah,
where the
bailout
ROV
is
is right now.
southeast.
And as
so-
you know, as we moved up this track, we saw that fault propagating across the
and
wall.
there hasn't
This
been a move
map,
for
the colors that you're seeing are
over
gradient.
an hour.
So the
so
brighter
everything
colors
should
are
be stable.
steeper areas,
Copy.
and the cooler colors are less steep areas.
Pilot, I think you
so
got
to
all
the
the
right
notes on
is
the
the canyon
samples.
floor.
Yep.
To the left is
So-
the wall, the top of the canyon, and the bright area in general has been going up the canyon wall. But I want to draw your attention to
Yeah, it looks like
at the
everything's
top you'll
pretty
see
stable,
kind of
so-
a arcuate kind of comma-shaped loop around the top of the canyon wall that seems to terminate at the canyon
And
wall directly where our dive track is,
based
where
on
we
our
saw
depth right
that
now-
fault cutting across the wall. So what we might be seeing is on this
It's
side
like a
we
little over
saw
an hour
the
bottom
downward
time.
propagation of that
Copy.
fracture, and on the
Little current
bathymetry
going
right
to the
now,
south.
that high area of slope, that loop
Close
going
link.
across the top of the canyon, might be the superficial expression of that normal fault where the canyon wall is kind of slumping down relative to everything
Yeah, like I said,
else.
there's
And
a
we're going to have
south
to look at some
depth.
bathymetry transects and all to confirm that. But
And-
if that
and
is
it's not
the case, that
real
would be a really
accurate
cool
based on the-
observation.
The accuracy, but southwest.
I'm going to turn on a few lights so you can see the tether. I think you might be feeling that upwelling that we had the other day,
Copy.
or at least a little bit of it. But maybe not.
So I just came up a few meters in elevation, and I got softer returns here, so I might be close to the top of that wall.
Oh, copy that.
But
Yes, we just had a
Yeah, I'm just kind of slowly coming up.
yeah,
pilot ship change.
Go ahead, watch lead.
And we're observing a lot of the same fauna that we've been seeing, and pretty high density. Those flame scallops, anemones. I think those little purple guys are stoloniferous coral.
I'm pretty certain that sediment that's going by isn't me.
Those are the clavularia stoloniferous corals. They're octocorals that grow over these areas.
Are those ones that grow their own substrate?
No, they do not. Those are the ones that grow on top of other substrates.
That's how I remember their names, because they're stoloniferous and they steal their home.
Well, they usually don't steal it from other corals, but they usually grow across rock faces, so they steal your rocks.
Which ones are the ones that grow on other corals?
Those are the parazoanthids.
I was just testing you.
Uh-huh.
Do we have a lot of time to get to the next waypoint, or?
So I guess I should have updated you on that. The waypoint is, let's see here.
it's down to the south, I think.
Yeah.
So the objective is just to get to the top of this feature?
Well, they've been working just along the feature, so.
Like on a contour?
Yes, just following it. So the last move was at, like I said, it's been an hour and a half ago, and they moved at 240.
So if you're just joining us, welcome to Dive 11
All the
of our Deep
previous
Connections
moves were 180
2019 expedition. This is
So
our
we're just
final
kinda moving
dive
along the
of
contours.
our expedition, which
Okay.
is
Yeah.
a sad time,
Oh, well,
but it's
okay.
been a really great
Like, I
dive so
meant
far,
with
and we
respect
still
to the wall.
have
Like
an
240
hour
would
or
have
so
brought us
left
right
in
up
our
to this
dive.
wall.
So we
Yeah,
are going to continue
but
to
on the
explore
wall,
this really neat carbonate wall that's been crusted with corals.
I think the previous moves were 180,
scallops,
moving us south, and then
anemones,
240 was just inching us a little closer.
sponges,
Yeah, 240
and so many more things.
Just moved
so
it a little bit, yeah,
keep
a little bit
exploring with
closer.
us as we make
But
our
all the
way
moves
up this
prior to
wall
that were
and
180.
start
So just moving south.
characterizing this community.
So,
another really interesting
like I said,
thing
it looks like
about
it's just
today's
following the
dive is
contour.
it's located
Okay.
a few kilometers south of the Pioneer
looks to
Cable
me like
Observatory. And so the
as you're coming
data that we're
around,
collecting today can be
yeah, that wall
compared
must
with
be almost
the
due
data
west.
collected at the Pioneer Array and
In
matched
relation
with that
to,
array data.
G2.
So this is a really cool observation of
Yeah, I would say the strike
coral
is like,
community,
southeast,
as well
right?
as
Yeah.
data
Southeast,
collected like CTD data that
northwest.
can be compared to
Yeah.
what's collected at the
That's
array.
what it looks like. Yeah.
Another interesting thing about
So,
today's dive is it's within the
I'll
Frank R.
find out with
Lautenberg
the,
Deep Sea Coral Protection
waypoint
Area,
2 is a hard
which protects areas
waypoint
like this
or
one that
just a,
host a number of deep sea coral species. And
move
these
towards it.
Yeah, that'd be
animals
good.
are
I'll
very
find out.
fragile and
Alright,
slow
on the next
growing.
move.
I'm just coming up slowly, co-pilot.
Watch lead now.
Copy that. Looking good.
Yeah, I just wanted to check with you, kind of get an update on waypoint 2. Is it a hard waypoint, or are we just kind of working along this feature? Okay, that's fine. Just as a heads up, we got about an hour of bottom time. Roger that.
Looks like it gets a little more barren just ahead.
Yeah, I think so. Yeah, I'd say next move we probably want to make, like, if we're going to stay at this depth and just go along it. I think it's more like south-southeast, a little bit more south than southeast. If that makes sense.
Yeah.
So something
so
like
maybe
150 or 160 or something like that. Because 240
Yeah.
has got us lined up pretty well with it.
Yeah, 240 got it right up against it. And then
So 90 off.
take,
Be 150.
well, so D2's looking
Or 160.
west-southwest.
We'll back up a little bit and look to the left and right.
Yeah. Strikes about something like that.
Yeah, I think you're right. South-southeast.
Yeah.
So the one.
Oh yeah, looks
50,
I think this is the
160
top
range.
here.
Oh yeah. You see it in a minute. Just coming up slow with you. Still
Okay.
got you at 30 degrees, so you're looking good.
Copy.
Yeah, it looked like if we move along it, like you said, between 150 and 160.
Yeah. So exactly. Yeah, so I just came up and lost sonar contact, so that seems to agree.
Watch lead nav, go ahead.
Your call cannot be completed at dial.
Okay, so we're
Please
going to head
check the number
north,
and dial
not
again.
south towards waypoint 2.
The number
Okay.
you dialed.
So the
6 6 1
the wall
7,
here, it looks like we
5,
may be kind
8,
of coming up
6,
to a shell.
0.
So the current is very slightly co-pilot from starboard to port.
Okay.
Looks like it's not really very much. I was just wondering, I thought we'd do a zoom on this coral, and if you would
Okay,
recommend
received.
to
So don't worry about waypoint 2. We'll head
I guess
on along
down
the north
current
route.
or
then.
up current.
Okay, received.
Uh,
So pilot shall copy that.
Probably the way that you're headed right now.
Okay.
Down current, yeah. I did copy that. I think that's that. We might want to talk about that a little more.
Roger that, watch lead, working on that right now.
So
How many moves?
not the south move. So
How many moves 180 did we make?
So everyone on the
Pilot,
line
that
listening,
fish has
we
something in
have
its mouth.
approximately 1 hour left
Oh yeah.
in
So
our
there
dive.
was
partial in one moment here video.
And we are
6
seeing some
moves at
fish
20
activity
meters.
at the top of this ledge. Ooh, and there's lots of cool stuff on this
And
part of
that's
the
Okay, you can go in video.
Okay, going in.
over 3 hours. So
Okay, I'm going to pan to the right.
So it looks like some eels have found some spoils.
and then the 240 move, I think, was just to bring it in a little
Are
closer.
those squid remains that we're looking at?
Yeah.
If I was going to make a guess, I'd probably
Okay,
guess
there's a little bit of
yes.
a current that I just got caught up in.
And they don't have any crabs to fight over it with this
Might have
time.
to clear one of your verts.
Okay.
Yeah,
All
this
right.
this
Come back
eel is
wide,
definitely
video.
making
Okay,
its
coming
way.
I'm gonna
Photo
clear the
bombing a
verts.
squid.
Oh yeah, I've got. Okay, that got it.
Perpetually photobombing squid.
Looking good now.
So many silly squids around here.
Whoa.
Yeah, once
All
it
right.
cleared it. You can still reach that one with the something in its mouth, though, I think.
Oh yeah. All right.
You got a little bit of tether. Sorry, I should have caught that one earlier.
That's okay. Okay.
Welcome to the conferencing
All right,
center.
let me get
At
a
any
little bit
time
more
during
stable
this
here,
message,
video, and then you can go
please
in
enter
partial.
your passcode followed by the pound sign.
All right, video, you can go in partial. Okay, I've got a little bit of tether, so maybe I'll get a little bit closer.
Yeah, you've got some.
Okay, video, would you come back out then? Okay, I'm going to run over a little bit closer.
Your passcode has been confirmed. If you need technical assistance during your call, press *0. After the tone, please state your name, followed by the pound sign.
Yeah, he's got enough. He's got plenty as we look at it now.
Okay.
I'll say it looks like you're up
Thank
on
you.
top of this thing now.
Okay,
You are
video,
the
you can
first
go back
caller
in.
to this conference. Please wait while others join.
Sorry, pilot. I'm back with you now.
That's okay.
Yeah, so we're definitely observing some
Can I-
cool
Yep.
Zoom in on
scavenging
it?
action
sure can.
from one of these cutthroat
Thank you.
eels. Looks like it's going after what we suspect is some. Is that squid? That doesn't look like squid, actually.
No, it definitely doesn't look like squid. That looks definitely like fish.
Okay, I'm going to pan over, tilt down.
Yeah, and it's certainly an interesting eating method.
Yep.
The
Coming out just to
suspended
frame him.
feeding
Okay.
going on. There we go. Now we're settling back onto the bottom.
Yeah, well, this eel doesn't want anybody to steal its meal.
Can I come in on
they
on eel
often
again?
will wiggle their body quite strongly in order to rip off a piece and try to shove more down its throat.
Okay, sorry.
Let him go for
Yeah.
a second. Okay.
All right.
As you
Coming
said
out.
before, the name of the game down here is how do I get food into my mouth?
Indeed. So these cutthroat
Okay,
eels
he's
are
underneath
some
my
of
poor toe.
the apex predators in
Gonna
this
back up a little
environment.
bit.
They're the ones feeding and scavenging in this area. So them, along with the rat-tail fish, are some of the apex predators that we'll see here in the deep ocean. It's using its twirl method to help rip off a piece of this
was cool.
fish it's trying to feed upon.
And of course, a fair amount of squid in the area. We can see
Okay.
the ink going around along with amount of debris, organic debris. This certainly looks like another one of those feeding zones that we've seen in previous dives,
Once
where
you,
just a lot of
never mind. Cleared.
that twirling method is really cool. I hadn't
Did it
seen that
happen again?
before.
Yeah.
Okay.
Yeah, they'll just wiggle
Yeah.
their body in any way to try to
I've
rip
definitely
off a
been
piece.
noticing a little bit of strangeness. I was kind of thinking it was the
Mhm.
changing current as we got to the top.
Yeah, the one was definitely a sabotage.
Okay, I'll come back in on this fish, or are we done with the fish video?
It's up to science. I mean,
Okay.
it'd be cool to get more, but like we're also looking for, like corals and
Copy.
and other organisms.
So, watch lead, I haven't forgotten about that coral, which is right behind me at this point. Would you like some more of this fish,
A
or?
fish at the bottom got food too.
I personally am good with the fish.
Okay.
I'd like to look at the coral.
Okay.
Are you seeing a ledge coming up ahead of us, or do you think this is it?
I think this might be it.
Yeah, then let's go back to the ledge and
Okay.
take a look at that.
Okay, I'm coming backwards. Go, pilot.
Sounds good. Yeah, you got plenty of room. So now my only hesitation with going north now is that we just came south, so going north would be backtracking a little bit.
Yeah.
So if that's what they want to do, then that's fine.
Okay.
But I know we came up in elevation.
Yeah, that's what I was wondering.
But, might want to...
Well, that was exciting. We just saw a cutthroat eel feeding on a piece of fish that it's found. It looks like there's a number of fish up on that flat. Just speaking with the pilot, we're going to go back down to the ledge to check out some of these really neat corals that are here on the rock face.
Nice.
And it looks like this is the end of the rock wall. So we want to make sure we characterize this area as best we can before we make our final ascent to the top of the ridge.
Do you think I should go straight into this guy?
I think you could.
Co-pilot?
Yeah.
Okay.
Watch lead now.
Probably touch your toes just right underneath it. Tilt camera just... Yeah, exactly.
Go ahead, Nav.
Yeah, I just wanted to confirm. We were discussing, you said you wanted to go north along the wall. We just moved south. I'm assuming that the previous moves were up the wall and now we're going along the top. Is that what the discussion was?
Yes, yeah. So now we're sort of at the top, so let's just go along the wall.
Okay, so we're going to zigzag basically. We went along it south, we've come up, now we're going to go back north.
That's correct.
Okay, cool. Thank you.
Yeah, sounds good.
Okay,
Yep.
that's what it is. Yeah. So we just want to follow this top part north now. So cool.
Sounds good.
So that would actually change our plans from the 150, 160 to looks like, well, it kind of bends a little bit, so we'll have to watch that.
Yeah, it does. Maybe like 330 is inverse of that, so something like that.
Yeah. Kind of looks like it has a little bend to it, though.
So we've come upon a Parantopathes black coral that has a number of associates.
We were to move.
Stand by with headlights if you want them.
Copy. Okay, when you're ready, video.
Okay, zooming in.
Yeah, I think that might be, I mean, I defer to video, but I think probably
Yeah.
we need some additional lighting.
Yeah, I think the headlights would be good.
Okay, coming up. Oh, nice. Yeah.
Okay.
Tilt down to the base first.
Okay.
So there are just
And,
a number of these brittle
Co-pilot, I
stars
think that you're already
hanging out
a
in the
little bit
branches
north
Can
of
I
this
zoom in, Pilot?
black
Yes,
coral.
yeah, as you will.
And
Okay.
this is a really good example
Gonna hold here.
of black
Okay.
coral
That way when
because
we
you can
tilt
see
up,
that black
it'll be
skeleton through the
in
tissue.
frame.
So a lot of
Copy,
times
and
these black
I'll
corals,
adjust
they
the heading, try
are
and
like
center it
different
up a little
colors,
bit.
yellow or
A little
brown,
bit better.
golden. And that's because you're seeing the tissue, which has a different color. But this Parantopathes has very lightly colored tissue, allowing you to see through it and the color of its skeleton.
Okay, whenever you're ready for the
Okay.
tilt out video.
I'm ready.
Okay, coming up slowly.
And if you haven't gotten that pilot, I think we're planning to collect this sample.
Oh, copy that. I didn't hear that. Thank you. Okay, we'll do.
Yeah, we were just discussing on shore whether or not this would make a good collection. So this might be well known, but it does have a number of
about as
associates
slow
like
as I
this
can
squat
go,
lobster
video.
and
That's
these
fine.
brittle stars, which could be good for genetics, since there's only
Looks good, Pilot.
been one collection of this particular species from these canyon areas.
I'm assuming it would go in one of the bio boxes.
Yeah, so I was just about to ask.
Starboard inner.
Starboard inner is, I think, the only one left.
Correct.
Yeah. Nice tilt, Andy. That's hard to do.
Thanks.
You could also send it through the suction if you take a small clipping, since
Ah.
it's pretty small.
Yep,
Oh.
that sounds good, Watchley. That's what we'll do.
Well, so the... Yeah, okay. So
Yeah, that
you
way we
clip it
make
and then
sure to
bring
get
it
any
through- bring it
associates
down
that
to the
we
side. Gotcha.
Gonna come
pick
out,
up
Pilot,
off this sample.
Copy video.
for full specimen.
Just a little trim off the top. Yep. In the background, you can see our eel
Oh, yeah.
really chowing down on that piece of fish. What I think is really interesting
could
that...
probably deploy
Oh,
the
wait, that's a
uppers.
squid. It's
Oh.
found something else to eat.
Yeah, sure.
Or that could be another one. We've seen quite a few of those
All
cutthroat
right.
eels in
And
the
in the
background now.
meantime,
Well, it's definitely lunchtime.
we'll get the hydraulics spun up.
Gotcha. All spun up.
Yep. Okay.
And Mary Wixton
So,
from Texas A&M,
think
who's
we'll
an
have to-
expert in crustaceans,
once you
has
take
called
the
in
clip
to
and
talk
putting
about
it
some of
in
these
your,
squat lobsters we've
jaws.
been
Yep.
seeing. And there's been
Okay.
a lot of
You'll have to
revision
hold it.
in
Oh,
the
yeah.
Chirostylodia.
Would you go
the
back
group
wide,
of
video?
squat
Okay.
lobsters that
Thanks.
is currently
Sorry,
living
co-pilot.
on
No, that's
this
I think you have to hold it.
Parantopathes
and
black
then,
coral.
come
Bridge
off
nav,
the
go ahead.
wall. You can
Okay.
extend the
Hey, Kurt's
drawer.
moving around
So-
a little bit.
and then
If
do
you come
A
to
collection
the
of one of these squat
about
Okay.
lobsters
10,
could be
maybe
really
15 degrees.
informative
Okay.
to that
I could
work.
also go on top and sit down.
Stand by one.
I think you're okay where you are.
Okay.
If you can, just stand by a
Because
second.
otherwise you have to reach down, and that's kind of hard.
Copy.
with it.
Hey, pilots, they need to move about 10 to 15 degrees starboard. Is that okay?
That should be fine, yeah.
Yeah.
As long as it's
Hold on, I slipped
still
off
about
the wall.
A-frame, yep.
Okay,
Is that
bridge
full
nav.
wide video?
ahead, Nav.
I guess it is.
Just want to
Thank
confirm we are on A-frame rotation.
Center rotation is on the A-frame.
Okay, then we're good for that move. Just let me know when you're done. Thanks.
Okay, we'll do.
Appreciate it.
Okay, hold on. Let me just see if that's stable. No, it's slipping down a little bit. Oops, slipped off.
Yeah, you might just have to play around with
Okay.
good footing there.
Are my verts good? Okay, yeah.
I think so, yeah.
Okay, I'm going back in. Just want to aim for more forward than down here, just a little bit of down.
Yeah, I think you were kind of teetering. And maybe that oscillation knocked you off a little. Here, I'll kill your headlights for now.
Oh, thank you.
Video, I think Mini Zeus is extremely dark.
Oh, is it? Let's see here. Oh, you're right. Sorry about that.
That's all right. Just want to get it ready for...
Yeah. Thank you. Yeah.
Yeah.
We were like against the wall a bit, so...
Yeah, I'm sure it was blown out.
Yeah.
Yeah, great. Thank you.
Okay, that might be a little bit better. Okay. All right, so then take a little off the top.
Yeah.
Either go back out into the water column or just run up and sit on top. Oh, because then we wouldn't be able to. Well, it might be harder to draw out.
Yeah. I think if we just came up and into the water column. I mean, you can go sit up on top then. As long as you're sitting on somewhat flat ground, we can draw out.
Sure.
That's no problem.
And then orient it into the tube and turn on the suction.
I'll turn it up. Yep.
Which cans have we used so far?
So jar 1 is the only one in use.
Okay.
So.
Okay. Would you mind moving the Mini over so I can see the arm, Copilot?
No problem. I'm gonna bring it up. We know Sirius is stable, so.
Copy.
Coming up. Yeah. So your right toe is off.
Yeah.
But you look super stable there.
Yeah. I think I might. That's kind of close, don't you think? I might try and auto head out a little bit.
I think you'll be okay there.
Okay.
Yeah, because you can always just, you have a good view of it here.
Yeah.
You can just come at it from the side, and then your arm's not in the way of the cameras.
Copy.
Okay. We have a visitor on our port side here.
I'll keep an eye on your thrusters too.
Okay.
Yeah.
Hydraulics coming on.
Oh. Photobomb.
And this squid is emitting copious amounts of ink directly behind our sample target.
Not happy.
Really nice example of them pushing water through their mantle to move.
Okeanos Nav bridge.
Up those clouds of sediment.
Bridge nav, go ahead.
Right
yeah, that
now.
heading change is done. We're steady on 145.
Okay, cutters are
okay,
on the bottom.
appreciate it. Thank you.
Yep. And if you
Grip force, am I on 3?
Yeah, 3. Yeah. If you come at it from the side, you can just kind of bring it right on in at that same orientation. So you don't have to do too much maneuvering to get it to the nozzle.
Uh-oh. Kind of going off a little bit.
Yeah, you're...
okay, I'm indexed in.
Vert's got...
I was going to say, I'm not sure that I think you got
sabotaged.
Yeah.
Okay. Gonna set down again.
Go ahead, Copilot.
Hey, I was just going to say it's just defending the pilot here. He's dealing with some outside forces that are making it hard for him to stay on the wall, so we'll reacquire and get back with you.
Yeah, no worries. Thanks for the heads up.
Okay, I think that should be good. Maybe a little bit more forward.
There's both your toes there.
Oh, got it. Yeah. Okay, I think that's a lot. It's probably even better.
Yeah, looking good. Yep.
All right, going back in with the arm. Okay, that looks like a good amount. What do you think, co-pilot?
Yeah, I think that looks good.
Looks good to me too.
Nice.
Okay. So go back out in the water column?
Yeah, I think I can bring up Sirius again here. Actually, you're okay without that view. I think you're... I'm not sure how well XY will hold you, but I don't think you were doing a whole lot of moving.
If we get over this bottom here, XY would probably be a lot better.
Yeah, agreed.
Okay, I'm just going to push forward a little bit.
And you got plenty of tether to get up there.
Copy. Okay, I don't have bottom lock just yet. DVL should be coming over the cliff edge pretty soon, though. I'm surprised I don't have bottom lock yet.
Yeah, I might just give it a few seconds here.
Well, I think it's pretty stable with auto depth and auto heading enabled. I think we'll be...
I agree.
Okay.
Yeah.
All right. So...
Yeah, so I will draw her out if you're ready for that.
Yep, stand by. I'm going to tilt down a little bit. Okay, standing by.
Okay. For now, I just want to make sure the buy boxes aren't open.
One moment. Let me just elbow up a little bit. My
Sure.
shoulder up.
Oh.
Up.
Nice catch.
save.
Sorry about that, watch lead. Okay, I'm ready for drawer out then.
Copy. Drawing out.
It was a good recovery.
And you're on jar 2. And I'll start the suction now.
Okay. So,
Yeah, I see suction in there.
looks like I got it. What do you think?
Yeah, I think you're pretty close there, yeah.
There we go.
And it's in the jar.
Copy.
Killing section. Yep, it's in there. Nice.
Thank you. Would you get the mini on the, so I can check
Sure.
stow?
Yep. Vehicle's looking good. Holding pretty much stationary.
All right.
George coming
So
back
we
in.
just got another sample, and I think we are going to continue climbing along the lip of this canyon where it's still really steep, just to see what else we can see.
So should I shoulder in a little bit?
No, I think when you kill hydraulics, it'll just kind of lay down nicely.
Copy.
Yeah.
Okay, hydraulics coming off.
Nice. Okay, suction's back on jar 6.
Copy.
And I'm going to center out mini Zeus.
So watch lead, now that we've collected that sample, would you like me to go back to the edge or?
Oh, yes, please. Right when we were looking at that black coral that we collected, there were some rather large colonies off to the right from
Copy.
there. So that'd be great.
Okay, I'm gonna back
And as
up
a
here.
Heads up, guys. 30 minutes.
Copy 30 minutes.
Watch lead nav.
Do you want some time on the bottom?
No,
Copilot,
I'm
or-
okay, but you got
Go ahead,
join
Nav.
lock in, I think.
Just updating you.
Thank you.
30 minutes of bottom time.
Thank you, Nav. So we have approximately 30 minutes of bottom time left in our dive today. We're going to go back to that ledge and check out the few large coral colonies that are off to the right from the one that we just collected. Characterize those, and then we'll check out this fish community before we leave.
Yeah, I got eyes
Nobody's
on the
going
ledge
to talk
there.
us off the
Copy.
ledge, Megan.
No one's going to talk me off the ledge. Not this ledge. It's got corals on it. But I'll walk on by to check out some fish later.
Did you want to range out? Well, Ceros isn't seeing anything out to 50 meters, but I didn't know if D2 would see anything. D2's at 10 right now.
I think let's keep
We're
D2 in.
We're just going to
'cause
stay
when
along
I get
the
back
ridge.
to that ledge.
Yeah. Okay.
Yeah, I think that's fine. I mean, 50 on mine.
Yeah. I just didn't know if there may be something further up,
Yeah.
but they want to go along the ledge anyway, so
Yeah, I think we'll just stick with it here.
yeah.
So we can head in either direction, watch lead. Oh, here's some corals. Or
Yes, those are exactly the ones I was looking for.
copy.
There was this very large pink coral, and then there's these really beautiful purples and another sea star. And now that we have reconnected our telecom line, Chris Maugh can tell us about some of his favorite stars.
So Nav,
I
when
bet
they
he has as many
made that
favorites as
heading
you do.
change, they translated
Probably.
10 meters or so.
Yep.
Yeah.
Yep.
So
But A-frame
So
stayed.
we are seeing a number
Yeah,
of
stayed,
these
so.
flame scallops, the Acesta
Oh, Sirius
scallops that
Tell me, how big
seem
of
to
a
be
head
lining
change
this
was it?
wall.
Sirius didn't move.
Okay.
Those purple corals, the
hardly at
Stolenfrus
all, but-
coral clavularia.
Yeah, I was just saying. Like,
Also
you can
spotted
tell
a
when
yellow
they're about the
coral.
A-frame or not.
Yeah.
That yellow coral is likely
Well, I just made
Paramuracea,
sure
a type
before they
of
started
plexaurid
moving.
coral.
So
Yeah.
Then we have
That's all I
these
was
neat
asking
sea
is
stars.
if they're
And
going
then
to make
this
that
pink
move and
coral.
you're this
As we
close,
zoom
Yeah,
in, I
right.
think
Watch
it will
it.
be a
Yeah, never hurts to
Paragorgia,
double
type
check,
of
yeah.
bubblegum coral. I didn't want to miss these beautiful fans. There's so many right here. Such
Sorry,
a
I
neat
said
ledge.
translate, I meant rotate.
Yeah.
Okay.
And 30 minutes is plenty of time to characterize these different species. And we do have 5 collections today, so we have filled up our boxes. We have plenty of great things from this dive, and a lot of work cut out for us for this evening. But you gotta go big or go home, because tomorrow we have to go home. So tonight we have to go big.
I think I'll keep your swing arms in for now.
Yeah.
Like this coral.
I think that's a good idea, co-pilot. So is this one that you'd like a more detailed zoom on, watch lead? Or?
Yes, please, pilot.
Copy.
I would like to see this pretty up close.
And it's oriented itself along a pretty small fracture on the rock face. I wonder if that helps it get a hold.
Yeah, this is a very large coral, and I think it's really interesting how the axis of this coral is bent downwards. You can see where it was first started growing, and instead of growing upwards, it has decided to grow downwards. So that suggests that the current lower down is stronger, and it's growing toward this stronger current, which is a really interesting observation. So, and might be the reason why we're not seeing too many corals further up. There's current acceleration at this point on the wall, which is why we're seeing so many more organisms here than we have in the other areas that we surveyed today. So this is a bubblegum coral, and you're seeing a number of associates in the branches. Those are snake stars wrapped around the branches, and the bodies, the main bodies of those snake stars are actually on the opposite side of the fan than what we're looking at. So we're just seeing the arms wrapping around the branches.
Okay, video. You can go in when you're ready.
Okay, coming in. Gonna frame up the coral really quick.
Now we're zooming in. It looks like there's a small squat lobster on this coral as well. Okay. And at the base,
am I free to zoom
there is a cup coral.
Okay, coming in.
Ah, there's Waldo.
Hold here for a second.
Couldn't spot him until now.
He was blending in with the rest of these snake stars.
All right.
Okay.
Okay, come back out partial, please.
Okay, coming out.
And then we've got some Acanthocordia coral, that yellow, light yellow coral that was right next to this Paragorgia. And I think I spotted an anemone in its branches, along with some brittle stars.
Another little community unto itself.
Yeah, there are quite a few animals right here in this small patch.
Copy.
And you can see that the holdfast, that base of this Paragorgia, is very wide and covering a lot of the rock face, which is going to be very important since we did test the hardness of this rock and discovered that it was relatively soft. So at some point, this coral could end up like the one we collected earlier in the dive that we found just laying on the ground at the base of this cliff face, where this holdfast gave away and left the coral down at the bottom with all of its associates.
Just waiting for some giant RV to come by and pick it up.
It was a really advantageous sample because that coral would eventually have been buried in sediment and died, so at least now we know it's going towards a really good use in furthering our study of the development of these corals. There are a few scientists that are studying them, one being Scott France's student, and he's looking into how these corals have evolved and the genetics and taxonomy of these corals. So it's really, really interesting work. And the more samples, the more we can learn about them.
Yeah,
so even though it would have perished on its own, it's going to a good use, and all the associates along with it will also be of a lot of help
Okay,
to
good.
people who are
Little
studying
down, get my
associated
toe back,
organisms with deep sea
laterally
corals, especially if we do find one of the aplacophorian mollusks that we spotted in that first Acanthogorgia that we saw
Okay,
during the
getting
dive. We tried
pretty
to
good
make a
lateral
collection of
going
that,
in.
but I think we
Pushes
lost the
me
piece of it
back.
that had those aplacophorans on it. But it's likely
Not
with
that
this
much.
second collection of Acanthogorgia that we might get one of those aplacophorans. And Kevin Kokot at the University of Alabama has been doing a lot of
Might
work
level out for
in
you.
describing those species of aplacophora and doing genetics on them as well.
Okay, I'm trying to creep back in a little bit here.
As a LSU
So
alumni,
in and down.
I may have taken objection
as soon
if
as
I
I
knew
give
these
it any
samples
down,
were
it
going to
just,
somebody at the University of Alabama, but
Probably just weird
I'm sure he's
geometry
doing
on that
great
edge
research,
there.
Well,
so I'll
yeah,
make
it's
an
kind of a
exception
slope.
for this one.
All research is great. Well, a lot of research is great research, and I'm sure he's doing some great research. And the name of Scott French's student is James Aubrey, who's doing the study on the Acanthogorgia. So part of our mission here is to help scientists all over the world who are making studies of these animals. So we're collecting as much data as possible and hoping to help out everybody in the community.
Okay, I think I might just have to.
Yeah.
All right, video, you can go in.
Okay.
Copilot, would you grab the rotator?
Sure. Got it now.
Okay,
Holding here, pilot.
copy.
Coming down. Holding. With the headlights on video?
I think we're good.
Okay.
To be honest with you. I think we're good. Okay, going to come in a little bit.
Okay, I'm trying to counter that pan.
Nice.
So as we get a nice zoom of this paragorgid coral, we're looking at all the polyps along this coral.
That's
And what
max zoom.
characters that we look
Copy.
for when we're trying to identify this coral is the branching and how dense the polyps are near the end of the branches. They tend to form these sort of bubblegum balls, and with the pink color, it really does look like bubblegum coral.
Okay, coming out, pilot.
Copy.
Gonna hold here for a
And
second.
as we look at the base of this
Copy.
coral, we notice that there
Bounced
are a
off a little
few
bit.
branches
Okay, coming out.
coming off from the main one that we're seeing. So it could be that there were multiple fans in this area,
Okay, that's
but
full wide,
only
pilot.
this
Copy.
one remains.
Thank you.
I'm going to touch it back up just past your skids and give it back
I
to
was
you.
about
Copy.
to say squid
Okay.
impudently
Rotator's
going
back to you.
into our view, but
Yeah, sometimes you just
a
got to...
partial squid.
Yeah,
go
well, we did
manual
learn from Mike
oscillate with it.
Vecchione that these short fin squids, when they get into a feeding frenzy, will attack each other. And
Who knows what's going
their
on with that
insistence on
geometry over
just
there,
being
the
up and
wing and
around our
everything,
vehicle
Yeah.
can be a little dangerous for them, because we do have many moving parts, mainly our thrusters, that spin quite fast.
Then the other side is an overhang. Do you want another shot of that coral watch lead or one of these other ones? Or?
Yes, one of these other ones would
Okay.
be great. I think we saw a sea star just below this coral, if we can go down just
Copy.
a little bit.
Yep.
I brought one of your uppers down when you were in tight on there. You might have to.
Yeah, this
clear
definitely
over
seems
here.
like the place where it's at.
You can see
There
the serious
is
view.
a nice big bramble and this cool sea star.
You're gonna have to clear your verts.
Yeah.
Copy. Doing that now.
And Scott France has
Yeah, it looks
spotted a yellow coral just below this Paragorgia
Did
as
that clear it?
well.
I think so. Yep.
Okay. Yeah, it was verting down at 50% and barely moving.
Yeah. I think that.
Yeah, I think
cleared
it's
most
just
of it.
at the
You might
bottom of
wanna
the
give it
screen
just
coming
another
into
little
view.
touch. I think you got it.
We're dealing with a temporary thruster issue, watch lead.
Understood, pilot.
And is this the one that you were talking about? The one in view?
It's just off screen, right at the bottom middle.
Oh, copy.
You got enough tether? I can come down with you too.
Copy.
And it actually does not look
Bottom
like that
middle.
cleared.
Look at all
So-
those
Okay.
acesta
All right,
clam,
I'm gonna
there's
come up to-
Some short
scallops, and a bright yellow coral, and then there are all these
There you go.
really cool
Just came out over here.
sea stars, the
Copy.
Brasingid sea stars, possibly
That's
in the
cool.
genus Novodinea.
Okay, I think we resolved that temporary thruster issue, watch lead.
Oh, perfect. Yeah, we'd like a zoom of that yellow coral first.
Copy. So, what do you think, co-pilot? Should I set up to the right of it?
I would maybe just try to hover on this one.
Copy. Will do.
You could put in auto depth now. It'll creep one way and then come right back.
Okay.
I mean, it's totally up to you. I think your Z bias is nailed down pretty well. But then once your auto depth sets, it's pretty locked in there.
Copy.
And then just adjust it as needed. And I don't think you have really any good places to set up on the wall.
Yeah.
Unfortunately.
Be kind of far away.
Yeah. Yeah, that auto depth is like super solid.
So we're coming to the end of
Okay,
our
you can
dive
go in
today
partial video.
at this box
Okay, coming in
canyon that's
partial.
located between Alvin Canyon and Block Canyon.
Gonna hold here.
Copy.
And this canyon is located within the Frank R. Lautenberg
Okay, I can get a little
Deep
closer.
Sea Coral Protection Area.
Yeah, it's right under Darcy.
And looks like this is
Would you like me
another
to come out, pilot?
Acanthogorgia
No,
yellow
you're
coral.
good.
Okay.
You can go in more if you want.
Okay, coming in.
Yeah, that's like.
It just looks really fluffy.
That's cool.
And that's my gestalt
rotating out.
to Acanthogorgia, as they tend to look really fluffy, and then the polyps look very skinny and unable to pull in towards the skeleton of this coral. In the branches, I'm spotting a number of brittle stars and amphipods
Okay,
as
rotating
associates.
down a little bit.
And then all around this coral are these acesta flame scallops.
Okay, rotating down a little bit more.
Some
I
with
should
encrusting
be.
sponges.
Indeed, some do have encrusting
Not
sponges, and I
moving
saw a
very much
snail on
pretty
another
soon.
one. It's just
Yeah,
a
you
hot
can see
spot
the-
of life.
Heather's still rotating your head a little.
Okay.
But that's still, like, that's a great shot.
Mike White just knows how to
I think
pick 'em out.
sometimes it works in your favor.
This has been a pretty cool dive.
Rotation.
There's also an anemone in the branch as well.
Yeah, can't really ask for a better last dive
Yeah,
than
sorry
this
about
one.
all those.
It's been pretty good.
I think the auto depth is still
Lots
kind
of
servoing in a little
cephalopods,
bit.
lots of corals, great collections. Hello.
Hey, this is Chris Ma.
Hey, Chris. You ready to see your
Hey.
really cool star?
Yes.
Okay, come back wide.
Okay.
Video, please.
It's a
Coming
happy
wide.
coincidence that because
Actually
this area
close to your left toe.
so
Yeah.
close to the lab
Copy.
of my Canadian
All right, I'm gonna come
colleagues
at auto depth
that
now.
I actually have identified specimens of this species before, of the Brasingid that you're looking
Yeah, if
at.
I back
You
up a
are
little bit.
correct that this is indeed Novodinea, and the species is appropriately enough Novodinea americana. The animals have pedicelaria, little small claws, if you will, that if and when we get to zoom in on the spines, all of those club-shaped structures, those are actually spines covered with a sock full of these little kind of claw-shaped structures. And so those are what capture they can actually capture small crustaceans like little amphipods and other things. And when they're captured, they're actually moved by tube feet to the mouth. And so even though they're very graceful and flower-like, they're actually pretty big, deadly animals. And I've actually seen these. I think it was actually on one of these
gonna hold here.
These North Atlantic dives. I've seen little cyclothony, those little fish, bristle-mouthed fish, run into the arms and get caught by the pedicelaria, and they don't. The one that we saw didn't have enough to get loose. So at least not for a while. So some of these Brisingids can really even capture not just mobile prey but vertebrate prey. It's not clear if it moved away, if the fish got loose after the ROV moved on.
Okay, you can go in video.
The Brisingids are some of the first animals that I studied, and they're really kind of cool.
And is that under lasers now?
The names are also really interesting because
lasers.
You were talking about how some of the names might actually reflect sort of their behavior. This one actually doesn't have anything to do with their functionality at all. These are all named after Norse gods, because the original one, Brisinga, is named after Brisingmen, a brooch that was thrown into the abyss by Loki. And so this used to be called, this genus used to be called Odinia, which was named for Odin, the god, the Thor's dad. And unfortunately, there's already a beetle named Odin, or Odinia, so it turned out they had to rename it. And so they named it New Odin, or Novodinia. So and these can always be recognized by that sort of really sort of club-shaped spine and
Am I clear to zoom in
the
on
sort of
sea
really big spiny structures
Okay.
that you see on the disc and around the proximal part of the arm. That's the part of the arm that's next to the disc. So
okay, should I come?
They're
come
usually
back
more
out?
shallow water than where we are today.
I'm gonna hold
But but they get to be to be pretty deep. Yeah, you can see how very pronounced all of the spines are. That's very sort of classic Novodinia. Although I think Novodinia antilensis is one that I typically see when we're looking at video from the tropical. And this is obviously farther north, and there's some question as to how closely related they are, or if they're possibly just the same thing. So interesting animals. So been fun
I think I can hold this one pretty good.
listening to the dive. Thanks very much.
Thank you, Chris. It's good to hear from you all the time about these really neat stars. I'm sad that this will be the last time you get to call in during our expedition.
Okay, coming down.
Well, I'll come back.
I know you will. You'll be on for the next expedition, telling us all about these sea stars.
Indeed. So have a good trip home and a smooth last dinner, and enjoy the first drink when you get ashore.
We will.
Good night.
Good night.
Coming out,
I
pilot.
slipped off. Thank you. So, should I try for more zooms on this one, Watchley?
Video is clear.
Thank you, video. Okay, I'll head over there.
Coming up. And rotators back to you.
Copy.
Yeah, just enjoying the probably the
Okay, can I
last
get-
call in
10 minutes, go.
Deep
Copy,
Connections
10 minutes.
2019 from Chris
Watch
Ma.
lead nav. Yeah, about 10 minutes.
Invaluable
10 minutes left.
echinoderm consultant.
I think that reflection video
So
is
now we're
just
going
off
to
the
take
wall.
a good look at these bushy, I'm going to guess they're black corals here growing on the wall. That'll also be a new thing that we have not yet seen today or during any of our other dives.
I think you
And
can
as we
reach
zoom in,
that,
I'm going to look at the branching pattern on
down with
this
you.
coral, because
Okay.
that'll tell us a lot about what this might be. And also, we have about 10 minutes left on bottom today.
Yeah, so you can
and I'm
see,
totally
like,
okay with not making it to the top.
Tether
This
really
is way
wants
cooler.
to rotate your head to the right. So
Okay.
it's
We
just a
did make it to
constant
the top. We
rotation.
just went back down.
So you've been
Yeah,
battling
because
that,
all the cool stuff is here.
Okay.
along with everything else.
Yeah, but I'm not going to get that, FOMO like we did on the other dive at Beach Canyon where we just started to see the really cool stuff and had to leave. We saw some cool stuff, and now we're seeing even cooler stuff. Lots of cool stuff.
You're going to jinx it. We're going to see the coolest stuff right before we come off bottom again.
That's totally how it works. But so looking at this black coral that we have in view, seeing these branching patterns coming off and how that it's branching in a similar way to what a Bathypathes would do, but it has all these sub-branching on it. So I think this is a Teleopathes. And let's see if someone's written anything in the chat. I see that Scott France has said that this must be Teleopathes magna. So it is pretty large, so magna does fit with this Teleopathes.
Yeah,
Okay,
definitely. It looks like kind of looks like a
stable
fern
here.
that you might see on land, but suspended on a canyon wall hundreds of meters above the axis.
I think I might just get a floating shot of this one. And what do you think, co-pilot? Should I just try and port toe back in?
No, I
float?
think you could float. Yeah.
Okay.
Yeah, you can just do a snap zoom pilot.
Copy. Okay, video, you can go in then.
Okay, coming in for partial.
Yeah, a nice good framing of
Holding
this
here.
whole colony will make a really good image for our deep sea animal guide. And then I'm also seeing a cute little squat lobster with very long arms.
Nice. Look at those arms, man.
It's on the
So that
right side of the
squat
coral
lobster
pilot.
is a
Copy that.
chirostylid squat lobster. They're usually characterized with
pirouette
these
around.
long,
Okay.
skinny arms. And we've seen them a number of times in black corals.
If it was just a little more flexible, it might have
Tilting
a future as an
down.
underwater wacky inflatable
Drifted
tube
back
man.
just ever so slightly.
Copy.
Yeah, nice.
Oh, yeah, I can see that.
That's a
Drifting
very
back.
beautiful example of a black coral. Let's go.
Can I zoom in a
So
little bit?
Yes,
Scott's
you can.
got some
Thank you.
information about this
Coming
coral that we're
in.
looking at. The holotype of this specimen, or this coral,
Okay, I'm getting a little
was
closer, so I'm
actually
gonna tilt down.
archived in the Nova Scotia Museum collection in Halifax, Nova Scotia, which is where
Gonna
we
come in a little
began
bit,
our
Pilot?
journey for
Copy.
this expedition.
Get polyps.
Yeah, that's taking us full circle. It's been quite an expedition. We started in
a little less
Halifax.
than 5 minutes.
We
Copy
remained
five.
in Halifax
that.
while we had some weather and equipment related
That's a nice
delays.
shot.
We got some great dives in the Canadian waters, including the Gully Canyon Marine Protected Area, a couple of other Canadian canyons. Transited into U.S. waters, went and hid from Hurricane Dorian for a couple days in Newport. Got back out, made a beeline for the seamounts so we could explore both the seamounts and the submarine canyons that we were planning
look away for a
on
moment.
exploring
It's
for this Deep
pushing me
Connections
way off.
expedition.
Yeah.
Got back for a few canyons and
Video's clear, Pilot.
now we're
Copy that video.
getting
Okay,
ready
coming
to
out.
head on home.
Thank you.
It's been a great 12 dives.
So Jeff, what would you say was your
think
favorite
some
thing that you've
accelerated
seen
flow
during
there that's kinda
these
just, like,
expeditions
pushing
and
you
these
up
dives?
and off.
Got a crab coming.
Yeah,
That's
we've
full
been
wide,
putting
Pilot.
together
Copy. Thank you, full wide.
highlights and dive summaries and the report and all, so that kind of stuff is coming to mind? I don't know. It's hard to pick just one, but if I did, it might honestly just be Magnateuthis magna. I really like those things. Those are really cool.
I'm going to have to say my favorite thing was the Spongapalooza on Retriever Seamount.
Oh man, sponges beat out the corals. Don't tell them that.
There was just so many sponges. I just
Or-
couldn't stop saying sponges.
I could not help but notice.
So if we could just end our dive looking at a sponge.
I am enjoying this crab rappelling down the very sheer rock face.
I'm really impressed with this crab's skill
It's
at holding onto the rock.
Truly impressive.
It really helps that it has all these small little holes. So Pilot, I spotted a sponge actually down and to the right.
Copy that. Got a little bit of time left.
Couple minutes left. Yep.
Copy.
Watch the nav.
There you go.
Go ahead, Nav.
Yeah, 2-minute warning.
Thanks. Just want a nice quick look at that sponge, and I
Copy
guess that'll
that.
be that.
I'll come down just a touch.
Copy.
So that was our 2-minute warning.
You can go in partial video.
Okay, coming in partial.
There's a little bivalve under there.
One last sponge for
There you go.
posterity.
With a
Holding
flame scallop
here.
wearing
I
that
do have more
sponge is
zoom.
a very fashionable bonnet, I think that would be called.
Yes, it does look like a bonnet. So fashionable. We have seen
Can I
quite
get,
the deep sea
zoom in more,
fashion
Pilot?
throughout
Yes,
our dives.
go ahead.
Okay.
Is that the
So
headlights
many
video?
beautiful colors.
Sure.
So many beautiful shapes.
Might
The
help with
great
the underside there.
deep red pancake urchins come to mind. I think those win my deep sea fashion award for this this expedition.
Sorry, this current's.
It's okay. Here, let me zoom out just a little bit for framing.
Yep.
It is quite fashionable, so it's good to get a good shot of it.
There you go.
Okay, I'll
And
try
we've been
hold
seeing
that.
There you
these
go.
small structures within the skeleton
Nice.
of this sponge, and they look like they might be reproductive structures, those little white dots throughout the sponge. And so we've actually preserved some of our specimens in formalin in order to preserve those structures for further study.
Yep, and all of this is
Okay, I was just
Okay.
kind of drifting
Coming
away
back to
because I
land
could keep it more
to,
stable.
That's fine.
hopefully answer some really
I'm gonna
neat
come out.
scientific
Copy.
questions in the future.
It was a good shot to end on.
Yeah,
So
sounds
we're
great.
doing
Yeah.
a slow pan
Headlights
out
going on.
right now.
Copy, headlights off.
Just
That's
So I
to
could-
wide.
get
think
our
maybe
last
I'll
look.
boogie it back up to the edge.
Okay. Yeah, I'll come up with you as you do that.
Yeah, I was talking to Dive Super, and he said
What a dive, what
pretty
an
much
expedition.
if we just come up when you guys are ready.
It sure has been a
the
great
ship's just gonna
expedition,
stay
and
on
it
the
has
current
been
heading.
a wonderful dive.
Come
I
0.5
can now
over
identify
ground.
more than
And
one
then,
biology.
when we get to about
thanks
500
to you.
meters,
You've
we're gonna
definitely
do
caught
0.5
on
through
with
the
a lot
water.
of the biology
So-
that you've been
Okay.
seeing,
That sounds good.
commenting even more than me some
And I
days
think-
on some of the
so, I think
biological
Pilot, all
aspects.
we have to do is
Just because I'm wrong on more than
For
half
me
of
to
them
come.
doesn't
port.
But the confidence is
Copy.
really good.
Okay, so
the
you go
effort.
port
A
to.
plus for effort. That's all
Yeah.
I want.
Yeah,
Yeah.
Yeah.
some of
And
the
then I would
best
go to starboard
biology
right.
by a
Right, exactly.
non-biologist I've
Okay.
heard in a long time.
Thanks, Megan.
So get a little bit up farther above this
Yeah,
wall.
and as we're ascending
and I
above
guess we can
the
call it off the
lip
bottom
of our
there.
box canyon
Yeah.
here,
Yeah, I would
taking
just let
in
them clear
the
this.
Okay.
connection
here.
Yeah, that's
between
fine.
the
I was just going to
continental
mark it.
shelf
Oh,
and
yeah,
the
I
deep
think so, the
sea
mark,
that
yeah.
the submarine
Yeah.
canyons
Just,
represent.
I thought you meant a call
I
bridge.
think this
No.
is
I'll just
a nice
I'll mark it.
opportunity for us to wish deep connections well.
I feel very deep and very connected.
Deeply connected.
So connected.
Okay.
So deep.
All right.
I just
that's
auto
delta.
depth in. It's going to servo up
Magnificent
a little bit.
connections.
Great.
Connections to the surface water, connections to the deep sea, connections
Okay,
to Canada,
so-
connections to the U.S.,
So should I back up a little bit more, or
connections between canyons, connections between
I think
seamounts.
you're probably pretty good
Everything
there.
is
Okay.
connected.
It is.
I think, yeah, the easy way to
Everything
do it
is
is
connected
for us
in the
both
deep
to
sea.
stay in
The deep sea is
autos.
connected to all of us.
Actually, you might want to back up because you're going to have to come 90, I
So,
guess.
okay?
With that, I believe
Okay, I'm coming back.
Copy
we are
that.
going to
Okay,
begin
I'll,
our ascent. So, with that, I would like to wish everyone
should I send
happy
her out?
rest of your weekend. And please tune in for the next Okeanos expedition, which is going to be in November. Details forthcoming. And for the last time on Deep Connections, this is Jeff L2, Geology Lead, signing off.
All right.
And then one last piece of poetry before I sign off. There are no boundaries in the ocean. So we want to, we came here to study and explore and find out more about how the ocean is connected to all other locations within our oceans. And as stewards of the ocean, it's a responsibility to do these kinds of explorations. And I'm so happy that all of you got to join us with these explorations, and I hope you had just as much fun as we have, because we've had a blast out here in the northeast canyons and seamounts, and exploring all these new places that have never been seen before. So for the last time on this expedition, Deep Connections, this is Megan Putz, your biology lead from the University of Hawaii, signing off.
All right, that's close.
Yeah, and as we're leaving the seafloor, I just want to take a few moments to thank all the people that made this possible. So obviously it's a big team. We got close to 50 people on board, everyone that played a critical component to making this all happen. Of course, the North Ship Okeanos Explorers officers, led by Commanding Officer Nicole Manning, Executive Officer Faith Knighton, Operations Officer Rosary Abbott, Joan Cachonago, Kevin Tarazona, Nico Osborne, and Brian Caldwell. I just want to thank the crew that did an outstanding job at keeping the engines running, keeping the deck safe, and making sure that we could dive every day. Vincent Palazzolo, Rick Abona, Dave Blessing, Chris Ramley, William Royo, Peter Brill, Mike Collins, Nate Sapien, Jared Hasendorf, Chris LeBron, Pedro LeBron, Sydney Dunn, Ray Capati, Ellie Pacheco, James Scott, Celeste Mori, and Randy Collins. Also like to thank all the wonderful personnel from the Global Foundation of Ocean Exploration, our engineers that worked on the remotely operated vehicles, and the video engineers, led by Shan Kennison, Chris Ritter, Bobby Moore, Dave Wright, Mark Durbin, Chris Wright, Dan Rogers, Levi Onema, Andrew O'Brien, and Lars Murphy. Also, big thanks to our mapping team that was hard at work whenever we're not diving.